difficulties that we have faced during the COVID, and can we do better? God forbid if there's another emerging pathogen that will take on the next pandemic, and then the way forward. Right. So we have seen over the last 10, 20 years that we're seeing a lot of emerging diseases and also pandemics that's occurring every 5 years or so. And according to WHO data, between 300 to 400 infectious hazard events or public health concerns are being reported every year. And of course, recently the COVID pandemic has shown the world that there's a lot of gaps in many of the public health infrastructures in many countries across the globe, including countries which has a huge income, i.e. high-income countries. So not just the low and middle-income countries, but also high-income countries. And of course, the progression of COVID has indeed proven that no one is safe until everyone is safe. So equity is important. We have to make sure that public health is covered across all regions and all countries across the globe. So COVID came and left, and actually has never left, but we're still seeing cases of COVID, but much milder compared to what we used to see in the past. But there are many lessons that we have learned from COVID, and these lessons should make us stronger facing future pandemics. So the lessons that we have learned, especially when it comes to respiratory pathogens, we have to be prepared, and preparedness does work. So countries have to work on preparedness plans. And health is everyone's business. So we have to work with the community and convince the public that working together is going to make an impact on health. And also we need to make sure that people know that no one is safe until everybody is safe. So you cannot have a pocket of people who are not immunized or not vaccinated and expect that you're going to have no outbreaks and no continuation of the pandemic. And of course, the response must be agile and adaptive, and you should change this response based on how things progress over time. Now, all of us, including us physicians working in the field of public health, have this pandemic fatigue. So after 5 years of dealing with COVID, changing policies, home isolation, quarantines, testing, retesting, and so on and so forth, people need to have a break. And they don't want to hear anything about the disease. They don't want to get vaccines. And unfortunately, this has actually gone beyond COVID, and now people don't want to get any vaccines altogether, including influenza, measles, and other vaccines. So we have to realize that this is really a fact, and we need to work with it. And we have to work transparently with the public. We have to be fair in our judgment and our analysis. We shouldn't be angry. We shouldn't be sort of reactive. And we should be consistent in our messages so that we should all say the same things and not be hesitant or not sure what we're doing. We have to coordinate our efforts, and also we have to be predictable so that we say the same thing and not change our mind if things change over time. We also know that most of the emerging viruses that come to us as a new emerging or re-emerging pathogen that will be the future of pandemics are coming from the animal side. And the mixing between humans and animals is always the source of future pandemics, or at least 60% of the viruses, the emerging viruses that we're aware of. And we have an emergence of a virus that spreads very sporadically between animals, then causes outbreaks among humans, causes outbreak and epidemics among humans, and then eventually becomes a pandemic when it actually spans more than one region of WHO, 6 regions. And of course, we have factors that tend to amplify these infectious diseases, whether it's weather. We can see the mosquito-borne viruses, and you can see the pink shaded area, which basically are impacted by the weather change. And also you can see the blue area where they impact ticks changes in the environment. And of course, pandemic is a crisis among many crises. So pandemics don't come alone. It actually gets magnified and intensified when there is natural disasters, whether it's an earthquake or wars, also by displacement and migration and people who don't have any homes, and also by changing in the climate, which we're seeing very actively, causing floods and hurricanes and fires everywhere. What else is happening that is going to make things even much more complicated is the political changes that we're seeing on a global level. And when I say political changes, the biggest change that we have seen over the last few months is the decision of the US to withdraw from WHO. And does that really have any impact? And the answer is absolutely yes. Okay, so many articles has been written on the withdrawal of WHO from the withdrawal of the US from WHO. And the reason is because the US have not paid its dues to WHO for 2024 and not planning to pay for 2025. So that's 2 years in a row. And that's 1/5 of the budget of the WHO. So that's a huge impact that's going to have on the strength and the ability of WHO to handle emerging diseases and future pandemics. So this is the detailed resolution of President Trump that was released on January 20th, 2025, to pause all the future transfer of US funds to the WHO, recall and reassign all US staff who are assigned to the WHO, and identify a credible and transparent US and international partner to assume the functions of WHO, and also withdraw from the negotiations on a pandemic agreement. So these all have been taking place. So the minute that decision was made, almost, I'm not going to say the numbers, but a lot of staff who are secondees from CDC in WHO were asked to go home, not responding to emails and not responding to phone calls, which was very, very stressful for the individuals and also for the organization. In addition, there's been a lot of other changes on the US health system, including the funding stop on NIH, and also the issue with the climate change withdrawal, and also some vaccine challenges that we're going to be seeing when it comes to vaccine coverage in the future. So this will lead to a lot of emerging viruses and pandemic potential. We have seen this graph from Anthony Fauci, published in the New England Journal in 2022, but these are not up to date. We're seeing a lot of things happening over the last year or two. The bird flu is causing a lot of chaos, and we think this is going to be the next epidemic or pandemic, especially with the US being sort of entrenched now with all the farms across multiple states having the virus in their animal population, and the virus being able to spread from different animal species within the US herd population. So we need to use whatever we have learned from COVID to be able to address the avian influenza issues. And there are not that many tools that can be used except doing good research early on and be able to sort of apply new therapeutics, new diagnostics, and new preventative strategies. The struggle that we've had with COVID should not be repeated again with avian influenza. And of course, monkeypox is still like a wildfire in Africa and also spreading to many countries across the globe. And of course, other pathogens like Group A strep, which started in Japan, but actually is in every country now across the globe. If you just put Group A strep on PubMed, you can see that almost every country has shown a significant increase in the numbers of infections caused by Group A strep. And the reason that most scientists put together is because of the 3 years or 4 years of isolation and loneliness that's come with the quarantine has led to sort of sensitivity to the Group A strep again. And this is some, significant severe cases of Group A strep in children that's been reported, showing, orbital cellulitis, necrotizing fasciitis, and also brain abscesses. So what has changed over the last many, many years of pandemics? When you look at the viruses, you can see that the list of viruses are changing, but the time to discovery is actually becoming much shorter. When you look at the 1918, it took 14 years for the virus to be identified. For SARS-CoV, it took only weeks. And now we have rapid diagnostic tests. The therapy was made available very quickly with antiviral, monoclonal antibodies and vaccines, while for other emerging pandemics, these were not possible. And of course, dealing with a future pandemic like avian influenza, we have to use what we have learned from COVID. And since COVID has declined in number of cases and also severity, we're seeing a change of pattern of viral infections. And again, you see influenza becoming much more severe. You're seeing much more RSV, much more TB, mycoplasma, and metapneumoviruses. And that's happening in every country across the globe. And unfortunately, we don't have any local data to show it, but we're seeing it everywhere else, and we should not be any different. So that's why WHO came up with this preparedness and resilience for emerging threats. And Mike Ryan, who's the deputy DG of WHO, said using a mode of transmission approach can help countries to prepare more effectively and efficiently for future events. So we should not be just targeting a specific pathogen, but we should be targeting a mode of transmission. And we know that all respiratory pathogens spread through inhalation or contact. So we should be basically focusing on these areas. And we should have good surveillance, community protection, emergency coordination, access to countermeasures, and have advanced clinical care. And they need to be coordinated very well in between. WHO is hoping that by 2025, 30% of the countries will have applied these prepared and monitoring system, and 80% by 2031. And I don't think we are there yet. So I think we're probably behind when it comes to these measures. And these are some lists of different pathogens that could be the future pandemic viruses, whether it's adenoviruses, coronaviruses, metapneumoviruses, and other viruses. And we have to work hard to protect people who are at high risk, the older population, people who are immunocompromised, and people who are at risk of becoming infected due to exposure to sick people, which are the healthcare workers. So research during pandemic is very critical to making decisions and moving forward. What went wrong during the COVID? Basically, a lot of research was done, but it was not a large number of cases because people were doing research on their own and in small groups and not working together as big groups. So people needed to know what are the symptoms of COVID, how can we detect the virus, how does it spread, how should it be treated, and how should we someday vaccinate against it. These were the key questions that came up early in the COVID pandemic. And we need to have answers to these questions, and that's why there was a lot of speculations and hypotheses made by scientists and researchers, and they needed to have some data. And of course, the best means of settling these questions is to conduct research on unprecedented scale, i.e. large scales with hundreds of thousands of patients. And despite having millions and millions of cases of corona, we could not have that done in many countries in our region because each hospital, each university is doing its own sampling and studies. So research sample size is everything. So you need to have large coordinated sampling, and that's why we have the famous UK recovery trial, which basically had coordinated centers in the UK that were able to recruit patients very quickly and randomize them and then give them therapeutic agents. And of course, they have been very successful in discovering diagnostics and discovering therapeutics. And then there are the other platform of researchers, which is REMAP-CAP. Again, this is involving almost 200 sites in 15 European cities. And now the US is trying to assemble a similar system in the US to deal with future epidemics and pandemics. And it's only when you have those numbers of people being involved in the study that you can actually randomize and you can remove biases. Otherwise, you'll have results that are not reliable, like we had initial data on hydrochloroquine, which gave a lot of hope for a lot of people, including us in KSA. We bought a lot of hydrochloroquine because we thought it was going to work. And when the data came out, it showed basically it doesn't do anything, only does more harm than good. So international collaboration is important, but also national collaboration is important. And I'm hoping that the RDIA or WIPAIA or KAUST or any of the new entities or Saudi NIH would be leading these initiatives to sort of bring researchers together so that we can be prepared for future pandemics. And of course, observational studies must be reformed so that we can deal with the next pandemic. And these are some of the ideas of how we can reform our observational studies in the future. We have to have master protocols ready to go and not think of a new protocol and get IRB approvals from 15 hospitals when the pandemic has started. Basically have standardized data collection. You have to collaborate and consolidate data and basically lay the scientific grounds very early. When you look on a global level, only 5% of the clinical trials that were done on a global level for COVID were actually randomized controlled trials. Most of the other ones, we've seen a lot of stuff done published in different journals. It's all one center, one site data, and that needs to change. Also, we need to have more data on health policy trials, closure of schools, closing work, and then basically putting people at home and isolation. We need to have more data on that. And of course, we need to work very hard with academic centers. So the Ministry of Health, the local public health authority needs to work with academia to be able to get the impact. So the question now is the world ready for a next pandemic? And the answer is simply no, it's not. And if you look at the scoring system that was done by an independent body that reviewed post-COVID pandemic, it shows that there's a lot of deficiencies on a global level and even at WHO level that needs to be addressed before we can actually address a future pandemic. So we need to have political leadership, we have to have country preparedness, we have to have a new financing system for WHO, which is now destroyed. We have to have independence of WHO, and we have to have surveillance, and we have to have access to multiple community measures and accountability. And when you look at the countries and their accountability, I like this diagram because it shows you, when you look at the mandatory requirement for countries, 99% are doing what they're supposed to be doing. But when you look at the voluntary requirement, you can see that the numbers decrease significantly. So when you look at the GEEs, only 35 countries are doing it. After action review, only 18 out of the 196 countries are doing it. And simulation exercise, only 35 countries. So basically, when it comes to voluntary activities of the countries, they need a lot of enhancement. Otherwise, they will not be able to do it. And this is based on civil society organization assessment that the world is not ready for future pandemic. We know how much money we lost. We know how many people died during the pandemic, and we know how many children became orphans because they lost their parents of the disease. And I hope that this will be the message for countries and for WHO to work harder to prevent such a disaster from happening in the future. And also, economically, it has a huge impact. And I just want to bring up your attention that 40% of the budget of WHO is dedicated for emergencies. So now with 1/5 of the budget of WHO lost by the US leaving WHO, where is that money going to come from? So we need to look at alternative support for WHO to be able to continue its effort. Vaccine manufacturing, you can see that the distribution does not include our region. And now with the new Crown Prince initiative, we will have huge manufacturing plants for vaccines in KSA that will be able to support many countries across the globe. And also, when you look at the CIPTI projects, looking at new vaccines, most of these projects are actually targeted for high-income countries and not low and middle-income countries. And when you look at countries that have vaccinated their population for COVID, look at the African region. It's all in red and pink. While you look at the advanced countries and high-income countries, their coverage was almost 90%. So there is a lot of inequity when it comes to vaccine supply. So what are the future recommendations? We have to create structures and partnership that facilitate clinical research, simplify clinical trial delivery, develop digital models and procedure for data collection and sharing, develop a mechanism rapidly leveraging pandemic funding, so we have to have money available for research, invest in clinical trial networks, and basically be able to sort of quickly recruit patients. For respiratory outbreaks, we have to focus on wastewater surveillance, and this is really critical, and I know that Wiqaya is doing it, but we should be more intensive in doing it. And there's many data. If you look at wastewater surveillance, you can see multiple applications. You can actually predict what's going to happen in the future when you look at the wastewater supply. And there's some data from the CDC in the US during the COVID time from Cindy Freeman saying that basically you can detect the change of the COVID virus almost 6 weeks, 8 weeks before the virus actually becomes detectable in the humans through surveillance. And of course, metagenomics, that's something that's very important and needs to be enhanced. Artificial intelligence for infectious diseases. And if you look at the Saudi CDC, these are the priorities for research when it comes to the Gulf CDC. It's all almost 90% infectious disease, whether it's antimicrobial stewardship, COVID, influenza, MERS, and so on and so forth. 90% of them are actually related to infectious disease. And also, Wiqaya has a list of things that needs to be applied and enforced. And we have the experts here who can ensure that these are being done. So basically, we need to sort of have a One Health system where we monitor the animal side and the human side and be early in detecting these changes by doing sampling at different levels, natural, urban, industrial, so that we can see if there's any change in the results. And if you look at some of the European countries and what they're doing as far as wastewater surveillance, you can see that they're testing for many, many different pathogens, many viruses, including RSV and influenza and other pathogens. And in conclusion, I think the next viral pandemic is not a question of if, but when and how, because it's going to happen. International collaboration is the only way to protect ourselves from the next pandemic. And of course, we need to work on new vaccine generations where we can use inhaled vaccines to protect ourselves from acquiring the pathogen, and making pandemic planning part of the everyday business of work. And a lot we have learned from COVID, we have to apply to be able to sort of secure our future, inshallah. Thank you very much for your attention.
Thank you, Prof. Ziad, for this is nice, presentations. Really, I myself, I enjoyed, listening to that one. I was looking for you not to stop, but we have to proceed with the other presentations. But really, you have touched base all the, what the researchers they are thinking, especially from the preparedness plan for the next pandemic. Thanks a lot, Prof. Ziad. So we'll keep the questions after the sessions. If that's okay with you, Prof. Ziad. Question after session. Thank you.
Hi everyone, it's my pleasure to introduce Professor Peng Hong. She's a professor in the BSE in KAUST. She leads an environmental microbial safety and biotechnology lab, advancing innovative solution for safe wastewater reuse. So Professor Ziad just spoke about wastewater, and you're actually lucky to hear it from the best here. Professor Hong advances innovative solutions, just like I said, for wastewater and wastewater management. She's a recipient of the 2019 James Morgan Award and Latin Prize 2023 finalist. Dr. Hong is also associated editor in environmental science and technology. We're looking forward to hear your work. So.
Thank you, and I'm not tall enough for you to see the whole of me, so. Oh, yeah. I need the mic. So, good morning, and I want to thank the organizers for inviting me to KU and also to this particular conference. This is my first time here, so I'm glad to see all of you here. So, we've just heard from Dr. Ziad about the importance of wastewater surveillance and how we should be prepared for the next pandemic. And this really aligns very well with what we are trying to do over in KAUST, where we are trying to somewhat work towards establishing a national defense strategy through wastewater-based surveillance, particularly using aircraft to sewage. And so in this presentation, I'm going to show some of our latest results where we carried out a proof of concept together with Dr. Sharif Hala as well, to demonstrate a proof of concept of how we can possibly use aircraft sewage to maybe work towards developing a national defense strategy. And the main motivation behind why we started this work, of course, is to prepare for the next pandemic, but also because Saudi Arabia is increasingly becoming a very, very important global hub. I mean, of course, we all know that, especially in Jeddah, which is the main point of entry for 90% of the international travelers that are coming here to Saudi Arabia, majority of whom are the Hajj pilgrims. So we are in Jeddah receiving actually a lot of international travelers who are mainly using it as the main point of entry to then subsequently move around Saudi Arabia. So in 2023, based on the stats that we have, is that Jeddah alone is already receiving about 43 million travelers in the world, in the whole year. And this is projected to increase to about 114 million by 2030, especially because of the Vision 2030, where Saudi Arabia aims to be a very active tourism site. So we are expecting to receive a lot of international travelers now and also in the years ahead, which then therefore leads us to think about the fact that all these global travelers can potentially be carriers to bring in new emerging contaminants into Saudi Arabia and then subsequently be disseminated within the local environment. So if that's the case, perhaps the first question that we want to ask is that how can we then develop surveillance strategies, maybe at the airport itself, to defend against new emerging contaminants that are brought in by the global travelers. So specifically, if we collect the aircraft sewage from the flights in which the travelers are coming in, we should therefore be able to maybe identify these emerging contaminants at the direct point of entry, and then perhaps come up with intervention measures to stop further dissemination of those new emerging contaminants. And if that's the case, some of the questions that we want to ask in order for us to optimally design our surveillance strategy instead of trying to survey all the aircraft, which is not feasible money-wise and labor-wise. So the first question is, which flight route should we be focusing on? And then the second question, for instance, is what parameters? For instance, is there a particular geographic origin? Are there certain socioeconomic factors that contributes most to the relative abundance of emerging contaminants and therefore should be focused on? So those are some of the emerging contaminants that I'm going to touch upon in this proof of concept study are antibiotic resistance genes and the escape pathogens. So the idea behind why we want to formulate this is because if we have a better understanding of the answers to the above questions, we can consolidate our sampling efforts instead of a broad-based approach of surveying all, especially if we have limited resources in certain low, middle-income countries as well. So that's really the motivation behind why we want to think about that. So in our proof of concept study, we sampled about 130 samples in the Jeddah International Airport from 27 countries, and that majority of the credit in terms of sampling comes from Dr. Sharif Hala and his group. So we sampled about 130 samples during the June 2023, and that's a consolidated sampling effort at that time, and we managed to get about 27 countries that comes from different regions, including Africa, America, primarily from Asia and Europe. And we can also classify these flight routes based on the distance at which those flights were flying from to Jeddah. So it can be classified as medium distance, medium haul distance, long haul distance, and ultra long haul distance. So these are classification based on geographical origins and also based on the duration of the flight for all the 130 samples that we have collected during June 2023 at Jeddah International Airport. So for all the samples, we actually processed and filtered them for DNA and RNA extraction. So we did both DNA metagenomic and RNA metatranscriptomics. So these are some of the things that we looked at when we were doing the DNA metagenomics, which is basically, again, the ARGs, the antibiotic resistance genes, and the escape pathogens at the DNA level. And then at the RNA level, we also look at somewhat the same, but of course we know that there are some differences in terms of DNA and RNA. So RNA would be providing information on those that are active and viable and transcriptionally active and so on. So DNA, however, may include those that are already dead, but the DNA persists for quite some time. So there will be some differences in terms of the data that we are seeing in DNA and RNA sequencing, which I will show you later. So in the first results that we get from the metagenomics, when we were looking at the different geographical origins, we saw that when we look at the antibiotic resistance genes diversity, originally we were thinking, oh, maybe from the Africa regions we might be seeing a higher antibiotic resistance genes diversity. But when we look at the Shannon diversity index and across the different regions, it's actually different from what we originally thought. There was, in fact, a lower antibiotic resistance genes diversity from those flights that originated from Africa compared to that from America, Asia, and Europe. So there was a lower diversity in ARG. And then when we further look into what are the main differences, from the DNA-based perspective, we saw that there was a much higher relative abundance of those antibiotic resistance genes that are conferring resistance to carbapenem in America, Europe, and particularly America and Europe, compared to Africa and Asia. And then there's also a higher antibiotic resistance genes related to the aminoglycoside and other beta-lactam in both America and Europe. So that accounts for the higher antibiotic resistance gene diversity in America and Europe from a DNA-based perspective. And then when we look at the RNA-based metatranscriptomics, we see a somewhat slightly different story where those transcriptionally active genes that are conferring resistance to carbapenem and aminoglycoside are actually much higher in Europe, perhaps because of the shorter distance of the flights from Europe to Jeddah compared to from America to Jeddah. So there are some differences in terms of what we see, but from those results, we see that there is a higher antibiotic resistance diversity in terms of of what we see in the sewage from America and Europe and Asia compared to that on Africa. And then if we focus on the metatranscriptomics reads, the mRNA reads that are associated with antibiotic resistance genes, we saw that they are much higher in the long haul distance, so significantly much higher in the long haul distance, not in the medium haul distance, but in the long haul distance, and then it's kind of drop in the ultra long haul distance. So how do we kind of explain about it? So one possibility, one reason to explain for why there may be a higher antibiotic resistance genes, particularly only in the long haul distance, is perhaps because of the longer flight duration. More people are using the toilets during the flight. So there is more discharge by the travelers, by the people who are inside the plane during that period. And then these genes remain transcriptionally active. Whereas in the medium distance, people sometimes don't want to use the toilet that often, so there's a lower antibiotic resistance genes. But then when it comes to the ultra long distance, then perhaps there's a less transcriptional active or decay of those antibiotic resistance genes, and therefore lower antibiotic resistance genes in the ultra long distance compared to the long distance. So this kind of makes sense in this case, which is why we see from the RNA transcriptomics, the Europe one has a much higher transcriptional active antibiotic resistance genes related to carbapenem and aminoglycosides. But we see a somewhat different pattern when we are looking at the mRNA reads associated with the escape pathogens. Instead of what we see just now for the antibiotic resistance genes, which is significantly higher in the long distance, the escape pathogens for the mRNA reads, they were consistent. They were not significantly different, regardless of the distance of the flight. So it's the same mRNA log reads for the medium distance, long distance, as well as the ultra long distance. They were consistently detected almost at the same level for the mRNA. So what this means is that regardless of the flight distance, these escape pathogens remain transcriptionally active and viable. Of course, in our study, we did not do cultivation and isolation, so we cannot be definitively sure that they are viable, but we are able to detect the mRNA reads, which at least suggest that they are transcriptionally active. And when we look at these escape pathogens, they include, for instance, escape pathogens like Acetobacter baumannii, Enterococcus faecium, Klebsiella pneumonia, Pseudomonas aeruginosa, as well as the Staphylococcus aureus. And so they were consistently detected throughout for the flights, as regardless of the flight duration. And what is more interesting, or perhaps interesting, or maybe a cause of concern, is that these escape pathogens correlate with different types of antibiotic resistance genes, and including, for instance, carbapenem and extended spectrum beta-lactamase, like for instance, Klebsiella pneumonia, as well as the Escherichia coli that were detected in the aircraft sewage in terms of transcriptionally active and mRNA sequencing, and that they were possibly correlated to these late antibiotic resistance genes that confer resistance to the last line of defense in terms of antibiotics therapy. And then when we dig further deeper into what socioeconomic factors can correlate with the escape pathogens, because we saw that the flight duration doesn't have any impact on that. So then what other factors, socioeconomic factors, will correlate with the escape pathogens? So again, if we look only at the DNA sequencing, we found that at the significance level of less than 0.05, which is at 95% confidence level, only the climate index correlates with the significantly with the relative abundance of the escape pathogens, and it's a negative correlation of 0.46. So what does that mean? That means those places with hotter climates, more hot climates, we see a lower relative abundance of the escape pathogens. But those that are with, let's say, colder climates, colder temperaments, we see actually a higher relative abundance of those escape pathogens in the sewage. So this is DNA-based. And then when we look at RNA-based sequencing, we now see even more parameters, socioeconomic factors that correlate significantly with the escape pathogens. The first is in terms of the antibiotic consumption index, education index, the GDP per capita, as well as the health expenditure index, infrastructure index, and also the number of Hajj attendees. So we see that majority of them, or in fact all of them, are negative correlation, except for the last one, which was the number of Hajj attendees. So just example, for instance, how can we relate to that? So in terms of education index, and which is also related to, let's say, the government index and health, or the infrastructure index and health expenditure index, these are all negative correlation, which means the more the education it is, or the more investment in terms of health expenditure and infrastructure, sanitation, and other kinds of infrastructure development, then the less we are seeing relative abundance of escape pathogens in the sewage, in the aircraft sewage. Okay. But then in terms of number of Hajj attendees, it's a positive correlation. So, which means that in certain routes, where we are expecting a huge number of the travelers to be Hajj pilgrims, we are going to expect seeing perhaps a higher relative abundance of the escape pathogens in the aircraft sewage. So this is what the current socioeconomic factors correlation is inferring to us. So if that's the case, how can we then consolidate what we have just obtained from our proof of concept to answer the two questions that I've mentioned just now? So for instance, which flight route should we be focusing on? So if your concern is on antimicrobial resistance threats, perhaps those flights that are originating from America, Europe, and Asia should be more focused in terms of the aircraft surveillance and less so for the flights from the Africa regions. And then the long haul distance, not the ultra long haul or not the short distances. Those long haul flights should perhaps also have a more intense surveillance than that of the ultra long or the medium to short distance. So if that's the case, perhaps if AMR is the main concern, the European originating flights should perhaps be more sampled more frequently than that from the American and the Asian. But if the concern is on escape pathogens, those countries that have low education literacy, low GDP, low antibiotic consumption rates, low health expenditure, and infrastructure investment should be more intensely surveyed and sampled for their aircraft sewage. So that would be, for example, countries that are what we classify as the global south. So Africa regions or those places that are low to middle income places. So it depends on really what the main focus and what type of emerging contaminants that one is looking for. And then some other optimization strategies is that perhaps during the Hajj seasons, we want to focus on those flights that are carrying with them per flight a high number of pilgrims. So we want to then focus on those to really look at both the AMR and also the escape pathogens as well, because there's a positive correlation with that of the escape pathogens. And perhaps RNA sequencing is much better than DNA sequencing to answer some of the questions that we have in terms of defending against new emerging contaminants, because we do see differences in the data that we can get from DNA and RNA sequencing but perhaps RNA sequencing is going to be much more effective to tell us what are transcriptionally active and which ones are the viable ones. So with that, I would like to acknowledge, of course, this study is not possible without help from the King Abdulaziz International Airport and the ground crew SGS, acknowledgement from the funding from CAUS, as well as the sampling logistics and the sequencing from NewDX. And with that, thank you very much.
Thank you, Professor. We will hold questions until the panel at the end. Speaker.
We'll move forward with the next speakers. I'm really honored to welcome Professor Muhammad Yasser. He's a professor of microbiology at the Special Infectious Agent Unit at KFMRC. And he obtained his PhD in molecular biology from South Korea and the postdoctoral training in US. His research pioneered metagenomics and culturomics to explore the antibiotic resistome, human microbiome, and emerging zoonotic infections, advancing the One Health approach. Prof. Yasser, please.
Thank you, Professor Holmes, and thank you, the organizing committee, for this invitation. And good morning, everyone. So I will share some of our research findings about AMR that we obtained to the metagenomic approach from a kind of diverse Saudi ecosystem. So sorry, I think there's a mess up in the. Anyway, so antimicrobial resistance is considered a silent pandemic. If you can look into the scientific literature that is published over 2 decades. So if you look into the literature, it's really supported by that antimicrobial resistance, the silent pandemic. WHO and some of the very good research articles forecast a very horrible picture that forecast around 10 million deaths from AMR by 2050, and that will cost around $66 trillion cost to the world economy if we didn't take up antimicrobial resistance appropriately. If you look into the figures on the top right corner, the highest deaths are expected from the Asian continent and African continent, around 4 million plus, followed by around 300,000 to 400,000 deaths in the North America and the European. And if you look into the per 1,000 population death, the highest expected is from the Africa, followed by the Asia, and if you can see, Saudi Arabia is also in the Asian continent where we see more death from the AMR. But one of the interesting study that is published in 2019 in Lancet, they recognize a little bit low attributions of death stated to antimicrobial resistance in the Middle East region, while high in the Sub-Saharan Africa, followed by the South Asia and Eastern Europe. So the important thing, we need to tackle antimicrobial resistance to prevent this horrible picture. And the best approach is the One Health approach, as Prof. Ziad mentioned in his previous presentations. So we need to take up all the human, animal, plant, and environment together. We call it One Planet, One Health. So, and if you look at the Saudi Arabia is a part of the most of the global initiative. It is a part of the class, and the Saudi Arabia has announced its first AMR action plan in 2017, and the revised plan in 2022 to 2025. While one of the most important events placed last year in 2024, November, the fourth ministerial conference, we call it Jeddah Commitment, that puts Saudi Arabia as on the front lead in antimicrobial resistance. So antimicrobial resistance is an ancient phenomenon, it's not a recent one. Antimicrobial resistance evolved from the very beginning. And several research articles published in a very prestigious journals all reported. For example, one of these good studies published from the Canadian group, they collect a 14-meter scores and they perform the dating and divide it into sections in the actualia and the permafrost layer. The permafrost layer, based on the carbon data, was about 5,000 old. And when they did the microbiome analysis, they identified some of the bacteria that were related to some of the recent bacteria, like the Bacillus species, Bacillus, streptococcus, and pneumonia. And when they do the functional metagenomic analysis, they identified antimicrobial resistant genes that were related to beta-lactamase that are shown in the red color, and the tetracycline resistant genes in the green color, and the monoglycoside resistant genes in the blue color, and that 5,000 old course samples. Well, if you can look into the current scenario, we know that then the antibiotic was introduced by Sir Fleming in 1928. While when introducing the clinical practice in 1940, we see the emergence of resistance against penicillin. However, we see the good thing that between 1950 to 1980, we get a large number of new molecules. That is called the golden area of antibiotic. But if you look into the down figures, we see the emergence of microbial resistance parallel against this molecule. What changed after 1980 that the number of new molecules has been decreased while antimicrobial resistance was increasing in the same pace. So what we are facing today in 2025, that we have several superbugs that we know we have limited antibiotic options or we have no antibiotic against those superbugs. So if you look into this graph, what exactly I want to establish the point that usually the antimicrobial resistance accelerated and it has been in a high pace in the human resistomes and the human pathogen, while it was evolutionarily low in the low pace if you look into the environmental system and the cleansing system. But what changed to that because of the increased human interactions with the environment, and as Professor Hans worked, she already published before, there was an increased discharge of the wastewaters and the antibiotic discharge into the environment. They might be changing this slow evolution to the accelerated evolutionary pace. It will be not surprising if we get some more new antimicrobial resistance from the environment now. So it's really important that we should not only screen the clinical side, but we should also look into the environment. And metagenomic is one of the best approaches to screen the antimicrobial resistant genes in the environment. So if you look into the studies, especially from Saudi Arabia and the PubMed, we hardly see 30 studies that reported the AMR from Saudi Arabia. But if you really look at those studies, you hardly see 10 to 15 studies that work on the Saudi ecosystem. But most of the studies are review articles or they are working on some other geographical regions. So our research group in special infectious unit, we are working on the ARGs from a metagenomic point of view. Some of our work has been already published, so today I will share some of the findings from the published work and some of the ongoing work we are currently doing. So what we do is we usually screen we're working on the MR from the clinical point of view as well as from the environmental point of view. We are working on the hospital microbiome, marine ecosystem, wastewater, soil microbiome, and the food chain and human microbiome. We're using the both approach, metagenomic approach, as well as the culturomic. As in metagenomic, generally we extend the delay from the samples. We do the next generation sequencing, shortened sequencing. Know why we are adopting the long read as well in our facility. Do the taxonomic analysis, identify the bacteria, and identify the resistant genes. While in the traditional way, we isolate the bacteria, identify them by the molecular, do the sensitivity analysis, and for the important bacteria, we do the whole genome sequencing, identify the resistant genes, virulent genes, and some further other functional analysis. So this is the major study we conducted with the support of King of the City for Science and Technology. As you know, the Red Sea coast that is around 2,000 kilometers long, a lot of development has been occurring in the past few years. We see some beautiful recreational beaches being built on this site. So in this study, we collected the sediment samples from the extreme south, Khorasan Iceland, to the north, Hakkaz. We collected samples from the beaches, we collected samples from the mangroves, we collected the samples from the island, and then some of the public study, part of the study has been already published. While from the metagenomic analysis, we identified around 708 resistant genes from those Red Sea coast sediment samples, and most of them produced was from the coastal side as expected, followed by the mangrove and the offshore side. And those antibiotic resistant genes were producing resistance against 28 different antibiotic classes, mainly against the fluoroquinolones, tetracycline, glycopeptide, and peptide and of course the multi-resistant efflux pump. If we further go into deep, around 15 genes were related to the fluoroquinolones, and 59 genes were related to the tetracycline, mainly belongs to the TEC48, TEC-A, TEC-B, and TEC35. While among them, 26 resistant genes were associated with the aminoglycoside, while 150 resistant genes were associated with the beta-lactamase, including the carbapenem resistant genes. Well, from the further analysis, we identify around 21 novel RF when we compare with the non-resistant genes. They show less than 70% sequence similarity and candidate for the novel ARGs. While among them, a good number of genes have less than 92% sequence similarity, but the non-resistant gene is a candidate for the novel variant. And if we look into the phylogenetic analysis, interestingly, most of these genes are linked to the environmental bacteria, and most of the marine bacteria like Epimerina, Henomonas, Echinobacteria, and Bacillus species. While only few of the isolate were linked to the Pseudomonas sturgeri, that is the clinical bacteria. So originally you can say most of these genes originated from the marine bacteria. So overall, I conclude this study that, of course, still we see that most of this thing from the environmental argument, but because of the increased interaction of the human mind, we will see a change in this resistome in microbial community in future. So we should look eye on it. The second is study we conducted on the hot spring that has a temperature from 89 degrees centigrade to 56 degrees centigrade. So, and important thing that hot spring is considered like an initial time. Their properties are more like the initial time with the high temperature. So, of course, in that time of collection, we see some of the human contribution. People were taking steam baths on the hot spring. So, of course, there was a human infections on those, or human contribution on those sites. But when I visit those sites today, and like not today, means like in the February, we visit the same site, we see a beautiful, the tourist site is being developed, a very nice table is being developed for the steam bath, and the people are swimming. So is that indeed more interactions and contribution from the human. So it's really important that we should follow those sites for the continuous microbial monitoring. So we did a very deep short-run sequencing, more than 160 million read per sample, and we characterized them for different functions. We identified some novel CRISPR, but my focus is on the antibiotic resistance that we found out of this hot spring. So we see a lot of protobacteria and then echinobacteria and chlorophyll, and of course we see more than 100 novel contigs that are candidate for the novel taxa. And if you can look into their antimicrobial resistance, we see the beta-lactamase resistant genes, we see the fluoroquinolone, erythromycin, and vancomycin resistant genes. And if you link these genes to the taxonomy, so most of these genes are linked to the thermophilic bacteria. For example, the beta-lactamase gene was found on the Thermus thermophilus, it's a pure thermophilic bacteria. The fluoroquinolone resistant gene was found on the Bacillus legiformis, which is commonly reported in the hot spring worldwide. And similarly, we see the vancomycin resistant genes were reported from the reflex in different bacteria that are the hot spring bacteria. So what is today, we see in the previous study that most of these genes are from these hot spring bacteria. But when the human interaction will increase, there's the maximum chances that there's a transfer of these genes from these environmental to the human pathogen. It will be not surprising if you see some new resistance coming out from this ecosystem. The third study we performed on the soil microbiome from the Western Highlands, supported by the DSR of the Kingdom of Brazil University. We performed the culturomic and metagenomic analysis on these samples. We see the traditional microbiology of these soil samples comprised of Rhizombium, Cytophaga, Burkholderia, and Pseudomonas and Rhodopseudomonas. But if we look into the antimicrobial resistance, we isolated some of the clinical isolated or resistant to the clinically important antibiotics like the azithromycin and the cephalosporin group and the carbapenem group, especially C and the Bacillus species and the Pseudomonas species. But when you look into the metagenomic point of view, we get several genes that were mainly suited to the multi-drug resistant efflux pump, some of the macrolide resistant genes, and also we found the sulfur resistant gene, tetracycline resistant gene. And if you look into the heat map, most of those resistant genes would belong to the efflux pump, that is the intrinsic resistance, but we also see some of the acquired resistance genes, including the beta-lactamase and the tetracycline resistant genes in those soil samples. And we confirmed some of the clinically important resistant genes by qPCR, and majority of them was related to the tetracycline, aminoglycoside, and the quinolone antibiotic. While we also know that the food chain is one of the emerging sources of the antimicrobial resistance. So this study was funded by the Ministry of Education. I will share a part of our finding from this study. We performed the metagenomic analysis of the fermented food in these samples, and we identified more than 800 species of those fermented food, including 28 known probiotic bacteria and 33 pathogens, but most of the pathogen was present in a very low abundance, and most probably these pathogens will be of environmental contamination, because those were present in less than 1% abundance. So if you look at the resistome, we identified 285 resistant genes among them, which was common among them. And if you look into the resistome profile on the heat wave, most of them was the multi-drug resistant and the efflux pump, so it means those are intrinsic resistance, and likely these are not required. But we see, of course, some of very few acquired resistant genes as well in those fermented food. So overall, and we further characterized those one by the card analysis. We identified 60 resistant genes, and most of those resistance were associated with the Alkinobacterium, and some of them were the Pneumonia Acinetobacter. So if I conclude my talk, so the metagenomic is one of the best approach to characterize the ARG and the environment, and it will be helpful to see the shifting of the microbiology in the resistome with the type and with the development. So profiling ARG from the ecosystem would be helpful for the health risk assessment of AMR, while metagenomics is particularly suitable for the antimicrobial resistance surveillance within a One Health perspective, support the development of effective policies to combat AMR. Thank you very much. So I'm acknowledging all my team from SIE and being part of the session.
For those nice, presentations about the food, environmental, and human from the One Health perspective.
So, thank you everyone for staying awake. And what we want is right now is to switch gears a little bit from metagenomics. So it's my great pleasure to introduce Dr. Bandar Al-Asami. Dr. Bandar is currently working at King Fahd. I've lost you. Okay, King Fahd Medical City, and his master's is in infectious disease. He did his PhD on oncovirology. Both masters and PhD from Manchester, so UK graduate. He currently leads the virology and vaccine research team and supervises several PhD students. It's probably the wrong thing to say this in King Abdullah University. You'll have a lot of students coming your way. And his work focuses on viruses and oncoviruses, area of critical and future global health and preparedness. So welcome.
Thank you very much. I actually have a student from King Abdulaziz University and two from King Saud. Thank you very much for the organizers, to all the organizing committee and the scientific committee at the KFMRC conference. It is a pleasure meeting you today, and it is also I'm honored to share with you our story about MERS coronavirus. The previous talks have driven us to AMR. I'm bringing you back to viruses to talk about MERS coronavirus. It is a story of a 10 years long-lived cellular and humoral MERS coronavirus immunity that cross-recognize the wild type and other variants of SARS-CoV-2. So let me first of all talk about MERS coronavirus as a quick introduction. There is around 2,600 confirmed cases of MERS coronavirus, and there are more than 950 deaths, which means that there is at least 35% of people who have been infected, they passed away. As of last year, 2024, there has been many reported cases, a total of 4, and 2 of them passed away from here, from Saudi Arabia. This year, there has been also 3 or 4 cases so far. So we are in Saudi Arabia, have at least 83% of the global health burden. Of those cases are taking place here in Saudi Arabia. So we are the most suffered country from this virus. And people who have been infected, they suffer from cough, fever, pneumonia, and in worst cases, they develop an ARDS and then pass away. Before the pandemic, we were thinking why there are 35% mortality, why people die from MERS coronavirus and not the previous viruses, and not the previous coronaviruses. We studied the virus and we described that in an inflammatory loop concept. So what is inflammatory loop? As you can see in this picture, we have the interleukin, the C5a induces several pro-inflammatory cytokines. Those cytokines, they include the interleukin 8. So interleukin 8 calls for further inflammatory cells and recruits neutrophils and monocytes to the site of infection, which is the lung. This accumulation and activation of those inflammatory cells trigger C5a. I wish I can point. Would this be possible to point? If there is a laser or something. Or maybe a mouse to touch the... because it's an interesting figure, and as I described, A triggers B, B calls for C, cells from C, and C again produces A, and so on. The loop continues, and the acute respiratory distress syndrome completes itself and repeats itself again and again, and the patient drowns with his or her own fluids, and they pass away. So the development of ARDS, or the cytokine storm as it described, is a result of this inflammatory loop that has been described by our team in 2019. I mean, during the study. So when we described that, that was very interesting, and it's led to an interesting finding during the pandemic. When the pandemic hit, we were thinking, what is special about Saudi Arabia? What can we provide special than anywhere else in the world? So it came to our mind that MERS coronavirus is affecting the country. It is the oldest brother of this current virus, and there is a 50% sequence homology between those 2 viruses. So why shouldn't we not study this virus and go deeper into its analysis? And there was an interesting question. There is a cross activity or cross reactivity between those viruses. There is a potential. Why we should study this cross reactivity? And there was only one study by Anwar Hashim, by the way, who found the MERS coronavirus immune responses lasting for 7 years, and no studies beyond that. So we said, okay, we have 10 years. Since 2012, so why we should study this and see if those immune cells would last for a longer period. We didn't stop that. We studied at a humoral and again at a cellular level. So across 10 years, from 2012 until 2022, we collected the samples, and we wanted to make sure that we wanted to study if there is a cross recognition between SARS-CoV-1 and MERS coronavirus. So our main goal was to see if the immunity of MERS coronavirus would last longer, or at least for 10 years, and if they would cross-react, and immunity from MERS coronavirus, from SARS MERS coronavirus, would give a protection against SARS-CoV-2. As I said, we are privileged in Saudi Arabia to have, it's a privilege and disadvantage at the same time, but we had this high number of MERS infected people. In my institute, there were around 540 reports. We managed to call those people and convince at least 70 people to come and give us a blood. We called them by all means, by the national pride, by we struggled to convince those people to come and give us blood. Mostly they were from rural areas and it was difficult to convince them, but we finally managed to collect the blood and extract the VVMC within the first 24 hours. We conducted several humoral investigations. First of all, we conducted a direct ELISA to see if there is long-lasting immunity since 2012. Another direct ELISA also to study the cross-recognition. But was it functional? That was our main question. We went to study an inhibitory effect, and also in a BCL3 facility, in a collaborative research site, we conducted a neutralizing assay where we grew the two viruses in a BSL3 facility in a 0E6 cell line, and we mimicked exactly what is happening in the lungs, in the human body. We grow 3 SARS-CoV-2 variants and 3 strains from MERS coronavirus, and we studied the reduction neutralization assay. We moved to also other cellular investigations. The most important one was ELISpot assay, which I will be talking about in a few minutes, and a series of flow cytometry. Let's move to the results. The most important finding was that during the sample collection, I was asking data collectors, have you seen any reports of people who have been infected by SARS-CoV-2? And the answer was no. I was relaxed and happy because if someone reports that they were actually positive for SARS-CoV-2, this would not really make us confident about our findings. So during sample collection, there were no reports of SARS-CoV-2 positivity from those MERS-infected patients. The first finding is that this infection led to a production of a long-lasting neutralizing antibody, and it is persistent of a decade of specific antibodies from MERS-specific coronavirus. We found also those antibodies were specific and still functional, bound and inhibited the binding between the receptor binding domains and ACE2 receptors. It was the neutralizing was as high as 60%. When we moved also to a direct ELISA, a competitive ELISA to study the cross recognition, the cross recognizing between the two viruses, we came up with I would say a clever idea. One of my research team came up with a clever idea. He said we can use an ELISA plate that already pre-coated with the SARS-CoV-2 spike proteins, and we add MERS coronavirus antibodies. It's a competitive. Let's see if those antibodies from those recovered patients would attach to the pre-coated spike proteins at the bottom of the plate and prevent the binding between SARS-CoV-2 and the pre-coated proteins. And this what was really happening, and we have found that those recovered MERS coronavirus patients, their antibodies could not bind to their antibodies actually bound to the spike protein of SARS-CoV-2, and they were competing to the same receptor. Lastly, in the humoral investigations, we have also, as I said, in PSL3 facility, confirmed and found the same findings where SARS-CoV-2 and MERS coronavirus antibodies produced a neutralizing antibodies. We moved then to a series of cellular investigations, and the most important finding here is the ELISpot assay. We have found that the CD8 T cells that produces interferon gamma and the CD4 T cells that produces TNF alpha, they were actually formed as spots, and those spots cells have produced a long-lasting MERS coronavirus memory T cells, and they were cross-recognizing SARS-CoV-2. So following that, we repeated the same finding using flow cytometry, and the finding from flow cytometry is that we have found a detectable amounts of CD4 and CD8 that were specific to MERS, and they cross-recognize SARS-CoV-2. And again, when we did the vice versa, we found again a detectable amount of cellular responses that was also specific to SARS-CoV-2, and they cross-reactive to MERS coronavirus. So we came to a number of conclusions. As I said, the first and important conclusion at the beginning is that none of our patients reported positive SARS-CoV-2 infection. That was a relief, and they might have a silent infection, but the disease severity wasn't that high or has not been reported, which gave us a confidence that our finding were legitimate, and this could be attributed to the antigenic similarity. The cellular and humoral immunity persisted for at least a decade post-infection. This is the major finding. The neutralizing also was confirmed by our study. We have found that MERS-specific antibodies inhibited the interaction between the receptor binding domains and the ACE2 receptors. We have also concluded that there is a cross-protection between the two viruses. It is a one-way cross-protection, which means that it is a protection conferred from people who have been infected by MERS towards SARS-CoV-2, but we did not study the way around. We don't know if you were infected by COVID, would you be protected against MERS? That's a question which we don't know. Maybe a potential study for a new PhD student. MERS reinfection, we can maybe arrive to a conclusion that MERS reinfection is not really possible, or unlikely. I would say unlikely, and that's because of the amount of protection that MERS coronavirus confers. This study has a potential further studies on pan-coronavirus vaccines, and as Dr. Mamish said, this virus has a pandemic potential, and it is reported, and it's still circulating in Saudi Arabia. So too much attention should be paid, and too many studies should focus on studying the antigenic and the genetic changes in MERS coronavirus. Many thanks to my main collaborators on MERS coronavirus, Prof. Jin Khan from the Shanghai University, Dr. Abir Shkeri from King Faisal Specialist Hospital here in Jeddah, Dr. Daniel Duke, also from the American NIH. We have a work on Hajj, and parts of it is on MERS coronavirus. Also, many thanks to Professor David Wissler. He's he's trying to produce a monoclonal antibody using MERS coronavirus. Dr. Hadi Yassin from Qatar University also trying to develop a pan-coronavirus vaccine. Also, I have a work with Professor Sarah Gilbert, who is developing a coronavirus, specifically a MERS coronavirus vaccine. And all of these projects are ongoing. Many thanks to my research team, the volunteers, the 3 PhD students, and the more to come from King Abdulaziz University, and to KFMC staff. This work has been published in the Journal of Medical Virology and was supported financially by King Fahd Medical City. And thank you very much.
Oh, this is nice. I mean, presentation about the study that you have commenced with your collaborators. I would like now to ask the speakers for the sessions to come please forward to the stage for the panel discussions. Please, Prof. Sihan, Professor. So we'll start having questions from the audience. Mike here, please.
So I would like to
take the mic, please.
I would like to thank the first speaker very, very much, Professor Dr. Ziad, for his contribution to the clinic Saudi Arabia. I am from Heidelberg, from Germany, attached to Taif University. I have a concept, and this concept is proven during my publication 2020 after discovering COVID-19 and origins and before SARS and their groups. From different resources, I got the whole genome through the cDNA of these RNA viruses, and I found COVID-19 have a lot of mutations. And I published another paper about more than 300 mutations already. And this has come to me. Are you agree with me that most of RNA viruses, single-stranded RNA viruses, like influenza, SARS, MERS, and the others, they are transmitted from various animals to human, and human from China to Europe to different ancestral, different origins, would cause mutation, and this could be the question here. It is that could be the reason for what you have noticed us and conclude in your good talk about the inefficiency or inadequate for therapies around infections cannot be treated.
I agree with you. I think the concern is a continuous mix between animals and humans. We always bring China into the picture because if you go to China and you go to the animal market, you can see how intense the contact is between humans and animals. And that sort of promotes the spread of new viruses and the emergence of viruses that are mutating. I think this is going to be a continuous challenge. And I know that the WHO and other public health agencies have always called for sort of having that link between the animals and human sort of more clear and preventing sort of the mixing and all the illegal markets in China. I think some of it is happening, but a lot is still to be achieved. And I think this is going to continue to be a challenge with new viruses emerging, especially the RNA viruses, until this problem is resolved.
Okay, I think if you have more questions, Professor Ziad will be around, so you could catch him during the day. So the second question over there, please.
يعطيكم العافية. صباح جميل. أخوكم بروف شائع المالكي. شكرا للمركز بقيادة الدكتور عصام أزهر وجميع الفريق وأيضا الضيوف الكرام. بروف زياد ما يحتاج ملهم الجميع في المملكة العربية السعودية وأيضا أبرود. يعطيكم العافية. أبد فقط سؤالي للدكتور بندر كان بودي إنه نشوف أناليسيز على البايوانفورماتيك بحيث إنه نحن نشوف السبايكس للتو فيروسز الميرس وأيضا كورونا ميرس وكورونا كوفيد، وأتوقع إنه في فرق في الهومولوجي وفي السيميلاريتي. النقطة الثانية اللي كان بودي إنه قبل ما تتطرقون عمل جميل بكل المقاييس، ولكن إحنا دائما نبدأ بالتحليل بالبايوانفورماتيك أناليسيس. Sorry, the guys اللي ما يتكلمون. We just I'm asking if they initiated their conducted study with bioinformatic analysis to analyze the epitopes of the virus and also the cell receptor tropism. Before going in details for what you have doing with the great appreciations for that. Thank you so much.
Thank you very much. It's a really interesting question, and it's really important. But this study particularly focused on studying the immunity from blood samples. And the virus was not there in the blood samples, and they were recovered patients. So we mostly were interested in studying at two different levels, the cellular and humoral levels, studying the immunity and how long would it last. حشينا فيك by the way, Dr.
No, but also لا أبدا. The antibodies في monoclonal وفي polyclonal والcross reactivity أكيد موجودة، لكن بنسبة لأن في النهاية كلها كورونا فيروس.
أيوة.
لكن السيل ريسبتور تروبيزم بيختلف مع الإبيتوب، فالمناعة أيضا حتختلف accordingly.
I got your point. From a genomic point of view, it is a really interesting approach, and it is something that we are planning to do. We have actually an ongoing project now studying different types of samples, and we are trying to find throughout the years if there were any genetic changes, a drift or shift in the gene for the gene of MERS coronavirus. We have the samples. They are dating back to I think at least 2015, and we're going to study the different changes that happened from that time until now. We have samples from also the palm fluid from people who have been infected. So if your question about the virus itself and the different mutations that took place, it is something that's going on. If your question about the host itself, this is something we haven't done, but it's important.
Thanks, Dr. Bender, for this. I think this is the idea behind being here today. It's just to overlap, ideas and hypotheses and research. So what would be the future directions for our research? And I think collaboration is one of the most important, issues need to be undertaken out of this, conference and sessions. One, we need to take, if there is any questions from the female side. Prof. Alia, mic, please. Yes.
Hi, good morning, everybody. My question is to Dr. Ziad Memesh. It's an honor to have you today with us. May I ask a policy-oriented question for you, Prof. Ziad? As you mentioned in your talk about the significant impact of USA withdrawal from the WHO, I know this will be there's a direct and indirect impact on the world, but if there is any very significant impact on Saudi Arabia policy, health policy, and the health data, especially we are annually having we need to do the preparedness for the health season. We need a lot of data, projects, initiatives, everything. So if there is any direct impact, if there is a solution, any advice for the health authority? Thank you.
I think so I think the impact is mostly on countries that are low middle income countries. So because WHO works in all different regions, and the support that was coming from the US government is actually targeted to countries. Usually it comes as a direct funding. So they say we want to give such an amount of money to cover a topic or disease in certain countries. But the impact on KSA will not be relevant. What is interesting is that since the US has withdrawn a lot of support from WHO and also from the global public health efforts in Africa, a lot of the European countries have done the same. So this is going to have a huge impact on vaccination coverage, on follow-up on clinical diseases and vaccine application across the globe.
Any more? Take one more question from the female side and one more from the male. Then we'll leave the other questions to during the day. All the speakers will be around and
Hi, thanks for all the talks. My question is to Professor Bien Hong. I'm wondering if your team has considered looking for the presence of viruses in the aircraft's wastewater, and if so, please could you share any finding or challenge?
We don't have we are looking at the viruses, including also the bacteriophages. So I don't have the data right now to share, but we know that from our past studies, when we look at the wastewater, and then it is mainly looking for the viruses. Majority of the reads are bacteriophages. So the bacteriophages are basically viruses that infect bacteria. We will have to do very deep sequencing if we want to look at enteroviruses or infectious respiratory viruses. So those usually are very low abundance, but of course we know that they are low abundance, but yet can be very infectious. So there is a certain challenge looking at the viruses compared to what we have seen for the bacteria and the antibiotic resistance genes when we use wastewater as a medium to look for the antiviruses and especially the respiratory viruses, because you will need very deep sequencing. So those are the things that I can share right now, but we don't have the exact data for that particular set of aircraft sewage that is pertaining to viruses to share at this moment.
Thank you.
Prof, I'll come to you. Prof Hassan, say it.
So my question.
Mike, please. Sorry,
So thanks for this nice session. And actually, I have more than question, but due to time, I will have one question to Professor Mamir. So thanks again for your precise lessons that have turned out from this the worst pandemic over the last decades. The question is, after this challenge that faced WHO and I mean the Arabian country or the Middle East country regarding the vaccination invention and design. From your point of view, how could the Arabian country, in terms of cooperation, that even in each country for the national feedback that turned out from the beginning of pandemic till it turned out, how can we maximize benefit in each country regarding the clinical data, the feedback of vaccination, even the natural immunity, and between countries so that we can get ready to the next pandemic when it arrives. Thanks again.
Well, it's a difficult question, but I think we should start nationally and then regionally. I think nationally through encouraging collaborative research. I think this is what this meeting is all about. It's trying to sort of reinforce collaboration between academia and Ministry of Health and the Public Health Authority and different partners who are interested in research to sort of build capacity and have ready to go protocols for future emerging pathogens. What we've heard today from Saimi and from on what is being done in preparation for producing vaccine or for preparation for discovering new pathogens, especially doing wastewater testing, is really an example of what needs to be done by all countries in the region working collaboratively. But unless we can work in each country within different sectors, it's going to be difficult to work between countries. I think there's a lot of progress. We have the Research Development and Innovation Authority, which is trying to build capacity. Then we have the Saudi NIH, which is building capacity as well. And then we have all the academia and the healthcare sectors in Saudi, which is very robust that will support any research that can move it forward.
Okay. Thank you, Prof. We had the last question, but I cannot leave it. My boss.
Well, first of all, I would like to thank you all for being here, part of this conference. And I think the last point Prof. Ziad mentioned is the critical point. My question is related to that. Today we have the capacity to do surveillance, capacity to do surveillance and discovery of viruses, pathogen, AMR. We have, in my opinion, one of the top 5 leaders in mass gathering and pandemic preparedness, Prof. Ziad. We have capacity in virology and immunology. How can we capitalize on that? How can we unify what we're doing and work together instead of working independently and actually then talk to each other and we keep complaining that we don't work together, nobody is helping, nobody is collaborating. What do we need to do? And this is for everybody, even in the panel. Thank you very much.
No, I agree with you, and I think I've mentioned some of these, concerns and possible solutions in my presentations. At least seeing what has been done in Europe and the UK, and with the US, which is one of the leading research body on a global level, is trying to emulate what the UK has done, what Europe has done in dealing with future pandemics. And I think it's good because we have on this panel expertise from different, we have academia, we have public health authority, and we have Ministry of Health, and we have university. So I think it'd be good to hear from the rest of the panel what their suggestions for moving forward.
I guess maybe the funding ecosystem has to be a little bit more mature, I think, because a lot of the collaborations at the end, I think we all want to work together with each other. But I think a lot of the collaborations would not happen without a mature funding ecosystem that is robust and sustainable. So I think that's the first thing that definitely needs to be tackled, especially if the funding, the funding ecosystem can be formulated in a way that actually prefer proposals that across universities, across different stakeholders work together to collaborate, and these are the ones that are preferentially funded. That would be one way to push forward with collaborations with different stakeholders. So I think that's one approach to do that. And then, of course, collaborations have to be built based on trusts and based on organic, organic collaboration and trust, and also capacity building and transfer of knowledge. So those are the things that I think needs to be tackled.
So my question is going to be from my colleague, especially to Professor. I would like to
hear all of that.
We can. Okay.
Professor, actually, I saw your work, and I've done some work before with Red Sea Global, working with sediment. But the thing that I would have loved to see, is there any link between the antibiotic resistance that you found and possibly One Health? Because this directly links with Green Saudi initiatives for 2030. And is there something? Should we be concerned? That's what you didn't answer in the slides.
Thank you, Shadfat. Yeah, actually, of course, as the infection will increase, I think we should be of concern. At least we should keep eyes on it, because the data we shared with you was before this major development. So I think if you have continued something from those sides, particularly for these new developments, the contribution on those sides, I think there is a possibility that it might be. So it's important, I think, from the surveillance point of view, we should continuously monitoring those sites, especially if not in the Red Sea, but we check that human microbiome DNA in the southwestern Carolinas, because those were also we consider might be a pristine environment. But we see some human microbiome DNA as indicators as the human conditions on those sites. So I think if we look into the human microbiome DNA on the coastal side, maybe we will have some idea that if there is some information or something. So one to the Prof. Anu Hashim, I would suggest yes, really important, collaboration is very important. Particularly, I think if you have a consortium approach for a particular topic, like if you are talking about AMR, you know, a consortium, national consortium on AMR, having different sectors from the agriculture side, from the ministry, the environment, everyone. So I think we will have a national data rather than sporadic data that will be more helpful to the country to make the policy development, etc.
I think with all these, different researches and what we have heard from Professor Ziad, the message to take home today is we need to be active rather than to be proactive. But I think this is really important. I think what you have mentioned, Prof. Ziad, in your presentations, you touch base all the problems that we are facing, and you put the solutions for it. But now we need to have that as an action plan, because we need it to be on act rather than being on just presenting that. And I think this is one of the main issue that we are commencing this conference to be able, to link with each other and building our networks to be able, to be more active rather than proactive. And we have got a very, excellent lessons from the previous pandemic, so we have to build up on that one. So by this, we would like to conclude the sessions, and we'll go for a break for 10 minutes, and then we'll be back with the second sessions and Thank you. Yes,
Excuse me? Could anyone stay seated, please, for the honoring our speakers and leaders?
انا بس قاعد
ما كنت انا كان في موضوع عندي فرق.
ايوه دخلت
حق هذاك ولا حق
انا دخلت الدنجي لا هذولاك
انا بديت كونتاكت خلاص واتس
ما حيسوي لك ولا شيء.
جتك واتس. ودحين
أبوكم اليوم بالإجازة.
Hello again. So we will start now our second session for today. Our moderators are Dr. Ibrahim Al-Zahrani and Dr. Khalil Al-Kuwaiti. Dr. Ibrahim Al-Zahrani is an associate professor of microbiology in the Department of Medical Laboratory Sciences at King Abdulaziz University. He is the head of Medical Laboratory Sciences Department at Faculty of Applied Medical Sciences. He is also a member of the Special Infectious Agents Unit here at the center. And his research focuses on antimicrobial resistant molecular epidemiology of MDR bacteria and development of novel genotyping technique for nosocomial pathogens. And Dr. Khalil Kuwaiti is an assistant professor of microbiology in the Department of Applied Medical Sciences. He is a member of both EcoHealth Research Unit and the Vaccine and Immunotherapy Research Unit. His research focused on antimicrobial resistant pathogen detection and evolution of novel antimicrobial compound. He is also, Dr. Khalil is also the head of research and research affairs department at King 5 Medical Center. So with this, we could actually start our second session.
Just wait for speakers. Dr. Majid, can I help? Bismillahirrahmanirrahim. Assalamu alaikum. Welcome everybody in the second session of our conference today. Thank you for you all for joining us today. In this session, we have four distinguished speakers. Each one will deliver 15 minutes of presentation, followed that discussion and question will hold at the end of the session, inshallah. It's a great pleasure to introduce our first speaker, Dr. Majid Al-Arabi. Dr. Majid Al-Arabi is Director of Biotechnology Program at General Authority for Defense Development. He leads strategic initiative aimed at advancing national biotechnology capabilities with focusing of biosurveillance, pandemic preparedness, and integrating multi-omics technology into public health. Dr. Al-Arabi also holds PhD in infection and immunity, and also a master's degree in molecular and medical microbiology from Manchester University. Dr. Al-Arabi published more than 50 scientific publications in high-impact journals. Today, Dr. Al-Arabi will present his talk in important topic of metagenomic and diagnostic emerging infections. It's my pleasure to invite Dr. Majid to podiums and welcome.
Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning, everybody. It's my great pleasure today to be here, to be invited by the event organizers, Dr. Anwar Hashim, and also Dr. Sam Azhar for this great event, where significant minds and experts are gathering today to discuss very important topics. I think based on the first session of this morning, talking about the pandemic preparedness, and the many points, significant point has been discussed by the panelists, where we need to be prepared. I think today I'm gonna deep dive a little bit to make sure that we have the right technology, I think, where we can fish and look for the pandemics before it's happened. It needs a lot of infrastructure and human capability and capacity, where we can be able to react quickly and to have that early warning system to kind of save the globe from the next pandemic. As we reflect on the last couple of years and what will happen with the pandemics, we are sure that the next pandemic is not a matter of if, but it's when. It's going to happen. And the WHO has introduced the disease X in 2018, even before COVID happened. But also, we are still kind of wondering about when the next pandemic will happen in the near future. That's what we need to be ready for this pandemic. And in this presentation today, I will be diving into the technology. I might be able to introduce the concept of technology, where we can be able to build the infrastructure and capability and capacity and the facility that are well equipped with the important technology to be able to identify the pandemic agents in the near future. If you allow me, I will kind of define the technology in three categories. The first category, which is the core technology, the traditional technologies that are now deployed and implemented in the medical diagnostic facility. However, these technologies is not going to last long, because as we know, the technology usually has been replaced by other new technology, and that's the second category, which is the disruptive technology. I think there are many disruptive technology now with the potential to replace these technology. But do we have this technology ready, and do we have the right facility to accommodate this technology and to train the human capital to be able to use this technology? However, this technology needed to arrive in the clinical diagnostic lab. It is now in the research and reference lab, but it needs validity. It needs a regulation to be transferred to the clinical diagnostic lab. However, the third categories of technology, which is the emerging technology. These emerging technology is still not in a technology readiness level to be deployed. Still in the research facility, it's an ideation of the technology that requires the proof of concept to be implemented and deployed. As you can see here, most of the lab, the clinical lab now in Saudi Arabia and the Middle East, and globally, depend on the culture-based testing, multiplex PCR, and immunoassays-based diagnostic and multi-top MS. This technology is reliable. We can 100% reliable to this technology, but however, this is our technology able to diagnose specific panel of viruses and bacteria and fungi. If we're talking about the disease X, we cannot rely on this because this has a databases and panel that's already been defined before. And when we go to one of the most important disruptive technology, which is the next generation sequencing and the metagenomics, these are kind of have proven and have a proof of concept that most of the recent pandemic has been defined by this technology. But recently, as we can see in many global research centers, CRISPR-based detection is there, but it's not has been proven to be a kind of validated for medical diagnostic test. Also, it's a synthetic biosensor. This is a future technology where synthetic cells will be able to detect any biological threat and shows a signal. This is highly potential, but it's required a lot of proof of concept. We heard about lab on a chip, and that's going to be very reliable technology, fast and portable. And all these data generated by this technology required a computational power that will be able to handle this data and analysis, where artificial intelligence, AI, and quantum computing are going to be arriving to accommodate these data and then able to do a fast analysis and also to predict, because AI now, we want a kind of systematic-based databases that will be able to do a deep learning from this data, and we need a quantum computing which is still immature in real life, but that will do a fast analysis. if you look at these pictures and we think about microorganisms, everybody is carrying around 40 trillion cells of microorganism in our body. And also, if you think about the environment, everything is covered by microorganisms, but one of them, it could be, or its mutant could be the pandemic, the next pandemic. That's we need a technology that's able to define the microbial community fast and able to kind of identify a biomarker where this is going to be at risk. I think the evolution of sequencing, started in late 1990s, where the discovery of Sanger sequencing enabled Carl Woese to define the 3 domains of life. He kind of divided the prokaryotics and archaea by using 16S sequencing. This is the first introduction of genome sequencing that enabled us to have a high resolution to define different microorganisms. And he said in 1998, genomic sequencing has come of age, and genomic will become central to microbiology's future. To those who doesn't know sequencing, this is a technology where it's able to define the genome sequencing by a certain process of differentiation of the order of nucleotide in the DNA molecule. There are, as I said, 2 key technology are now, which is the Sanger sequencing, where it's sequences short of DNA sequencing, but also the next generation sequencing enable us to do high throughput sequencing to generate huge number of data. In this figure, it's illustrated a beautiful history of sequencing technology. It started in 1995 by sequencing the first genome of influenza, and then that's kind of shows the first generation of genome sequencing. Between 2005 and 2010, several sequencing technology and platform has been introduced, including 405 and Ion Torrent and Illumina, and that's also defined a certain short read sequencing. But between 2010 and the present, we have another generation, which is called the 3rd generation sequencing platform, where able to do long read sequencing. Also, if I want to deep dive, this chart shows the amount of data that has been generated over the years. If you allow me, I will skip the slide because I'll have a lot to scan and the time limitation. These the different sequencing platforms, Sanger sequencing, and the amplified molecular sequencing and singular molecular sequencing. However, there are many sequencing companies are evolving at the moment to have a kind of a promising sequencing technology to be able to sequence fast and with accuracy. What I'm talking today is a metagenomics. Metagenomics is very important technology. It has to be deployed and implemented in most of the reference lab to be able to identify the the microbiology community. Any sample types can be collected from human, from soil, from water, and this technology will be able to identify any genomic materials in this sample fast and quickly and with accuracy. It's culture independent approach. We don't need to capture the viruses and bacteria. Directly from the sample, we can sequence these genomic material, and we can identify the community, the taxonomy of these viruses and bacteria easily to be determined, and then we'll be able to figure out if these microbial community are harmful or good to the environment. I'm not going to go to the details in this slide, but there is 3 generations of metagenomics has been able to show a proof of concept that metagenomics is very important to identify diseases and infectious diseases and any pandemics that occur. And you can see here, for example, in 2000, SARS outbreak has been identified in 2001 in China by Metagenomics. And also the COVID-19, the SARS-CoV-2 was being identified with metagenomics. Without metagenomics, we'll be not able to identify any pandemic agents. There are two types of metagenomics, the targeted metagenomics and the shotgun sequencing. Targeted, that means we have a specific region of the DNA we need to amplify using high throughput sequencing. For example, if we want to identify a bacterial agent, then we need 16S metagenomics, and that will be able to determine only the 16S region in the bacteria, and then we'll be able to identify the bacterial taxonomy. However, if we need to know the rest of the genome, then we need the shotgun sequencing that will enable us to identify taxonomy. Also will be able to have data that will eventually allow us to identify the functional profile of these organisms. There are many there are many workflows that will enable us to identify either DNA or RNA. I'm not going to go into details, but you can define based on your requirements where to go, which workflow to define to identify your infectious agents. As I said, MERS, SARS, Ebola, and many agents has been identified by Metagenomics, and also there are many studies. Today, maybe I will take one of our collaboration with the King Faisal Specialist Hospital in Riyadh. We've been able to identify the cause of this this infection by Metagenomic. Without Metagenomic, we've been able to identify this agent. With Dr. Reem al-Maghrabi, there was a patient with 55 years old with a post-renal transplant patient with a multiple comorbidity, including hypertension, diabetes, and stage renal disease. We take a sample from that patient, and then we'll be able to run this sample through a pipeline. This is illustrate kind of the X-ray image of abscess in the brain, and then we'll be able to culture this bacteria. However, we've been able to take a sample directly from the patient and run it through our metagenomic pipeline, and we've been able to identify the cause of the disease. We used targeted sequencing metagenomics using 18S for fungal, because we looked for the 16S, there was no bacteria. By the 18S, we've been able to identify the cause of this pandemic. If you allow me, I will skip the next studies due to time, and I will go where we need to think about the challenging in metagenomic diagnosis. Been discussing with Prof. Arnab here early this morning about the challenging of metagenomics. Still immature, still required a lot of work. And the first, challenge, which is sample collection. The sample collection should be based on a systematic basis. So it needs a protocol that enables to collect the right sample without losing any microorganism in that sample. Any kind of storage problem that will allow to lose some of these microorganisms. Also, the sequencing. We need to have a reliable sequencing platform with the kind of standardization protocols to choose the right sequencing protocol, either 16S or 18S. Still, that has a lot of doubt, because the library preparation required a good skills technologist, and these required a lot of training. When you come to the bioinformatics, this is very important, as all these generated data require a reliable bioinformatic pipeline to be able to identify and interpret that data to meaningful results. Also, another important issue here, the systematic basis of database. We don't have a reliable database yet, and that's required a lot of initiative. Maybe Dr. Turkey today will talk about how we can build our own databases. We have to build our national database that will enable us to predict if there is a problem or any significant microorganism in our region. We need a regulation, we need a policy, and that's required a lot of effort from the government to kind of approve this technology for clinical diagnostic. All this will have an impact to rapidly respond to the outbreak. If we have this issue and challenges resolved and put in a systematic approach, then we'll be able to react quickly. And also, I will echo Dr. Ziad, that is not a one-man show. It's required a collaborative research from academia, from government, from private sector as well. All these need to work together to be able to build a kind of a reliable platform that will respond quickly to pandemic. Future, I think maybe AI and quantum computing and the computational power is an important element in the near future. We're able to solve a lot of problems. We need to build our capacity and capability in this domain to be able to react fast, because if we have a digital platform that enable to kind of obtain all the data from different sectors, then we'll be able to have a high resolution picture of what's happening at a national level, at a regional level, and also at a global level. Thank you so much.
Thank you, Dr. Majid, for your insightful thoughts, and this question will be all inshallah at the end of the session.
I'm honored to introduce our next speaker, Dr. Turki Ojali. Dr. Turki is an associate professor of microbiology in the Department of Medical Laboratory Sciences at KU. He also serves as the Deputy Director for Administrative and Financial Affairs at KFMRC. Dr. Turki leads the EcoHealth Research Unit, and he is also a PI at the Vaccines and Immunotherapy Research Unit. With over a decade of experience, his research focuses on the human microbiome, antimicrobial resistance, host microbiome interactions, and integrating bioinformatics with clinical applications. His talk today, Toward Building Saudi Microbiome Database, promises to be both insightful and thought-provoking. Please join me in welcoming Dr. Turki.
Assalamu alaikum. So my talk today actually is more into an invitation for collaboration to join forces actually in order to build a national Saudi database in microbiome and health and disease. And today I'm going to show our early stage in building this database and focusing on the dispiosis in pediatric inflammatory bowel disease. Working. It's freezing. Okay. So as Dr. Majid Al-Ghuraibi said, there is trillions of microbes present in our body. And actually, we are more microbes than human. If we count the number of microbes compared to our own human cells, they are actually outnumber our cells. And mostly these microbes composed of bacteria, viruses, and also there is archaea, fungi, and parasites. If we compare the composition of microbes in a cohort, we will find it divided into two main groups. The first group is a core microbiome that is shared between individuals, and the other one is a variable microbiome that is different. And the reason for this variation is many factors, including the mode of delivery, utilization of medications, aging, diet, genetics, lifestyle, and geography. These microbes present in our body, they perform very essential functions. One of the major functions is that they train the immune system. Also, they resist pathogen, promote epithelial cell repair, as well as many other beneficial functions. So we have in our body some certain symbionts and commensals that promotes health, and also along with those, there is pathobionts that promotes inflammation. So, and we live in a balance. When this balance is shift toward more pathobionts, will have inflammation that is associated with disease. There is a wide range of diseases that are associated with dysbiosis or differences in the composition of microbiota, including cardiovascular diseases, liver disease, diabetes, obesity, and arthritis. Also, it's linked to many other neurological disorders, including anxiety, depression, and autism. Actually, this tree is growing. There is more disease associated with the dysbiosis of microbiota. And not only with chronic infections or chronic diseases, they also associated with infections, where they found that dysbiosis in microbiota is associated with pneumonia, tuberculosis, hepatitis, Clostridium difficile infection, SARS-CoV infection. Because of their importance, the NIH in 2008 established a mega project, which is called the Human Microbiome Project. The aim of this project is to characterize the diversity of microbiota in different body parts. And since we don't know the exact requirement for culturing these microorganisms that are present in our body, they launched a culture-independent approach for the characterization of microbiota. So the main question they were trying to answer is, what are these microbes? And the second question, what are they doing in our body? The result of this project and other studies showed that there is key microbes that present in certain body parts that belongs to certain countries. And here we can see that the key microbes in different countries, including the GIT tract, skin, urogenital tract, and oral cavity. And here the countries are colored according to their degree of urbanization. So these microbes actually either in health and disease could be used as personalized medicine approach and could be used as a diagnostic biomarker. But if we look in Saudi Arabia, there is very limited amount of research that are done on the human microbiome. But one of the very interesting studies conducted here by Prof. Muhammad Yasser, where they actually compared the microbiome of Saudi and French, and the obese and lean Saudis, and compared them to obese and lean French. And here in the principal component analysis, we can see that in the green there, that there is similarities between the normal and normal French and normal Saudis. However, there is clustering in the Saudis and close together. But we can see the differences in the term of, in the red there, and the obese Saudi and obese French. So it means that there is unique definition of the Saudi normal, and there is a unique definition of Saudi obese, which means that the gut microbiota in the Saudi population is different than other parts of the world. So this actually incited us to start working on establishing the Saudi microbiome in health and disease in collaboration with Dr. Prof. Arnab Payne from KAUST, and funded by Smart Health Initiative, where we actually collected different samples from different parts of the body, including the respiratory tract, where we collected nasal swab samples from healthy individuals and people colonized with MRSA, also from the urogenital tract, from female, high vaginal swab from female infected with HPV and normal healthy female, as long as in addition to oral cavity samples from children that have dental caries and normal children, and got samples from healthy children and children affected by IBD and Syria. So in total, we have 1,862 samples. And today, for the focus of this work, I'm going to focus on the our finding or preliminary finding in the IBD work. So just a recap, IBD is an uncontrolled ongoing inflammation that affects the gastrointestinal tract, divided into two main subtypes, ulcerative colitis and Crohn's disease. Crohn's disease is a deep inflammation in the tissue, and it's patchy that could occur actually at any part of the gastrointestinal tract. But ulcerative colitis is a continuous superficial inflammation that is mainly restricted to the colon. The exact etiology of IBD is unknown, and the incidence of IBD is on the rise, especially here in Saudi Arabia. So the cohort, sorry, the alignment I think is not. But anyways, So in total, we have 77 samples in this study. 41 of them are healthy children, and 36 with IBD. 23 of them are CD patients, and 13 are UC patients. And here we can see the age range and male to female ratio, and we can see that it's similar between the groups. So what we have done, we have collected stool sample from those patients, and then extracted the DNA and did metagenomic sequencing. That was actually done at KAUST. And then we utilized our optimized pipeline to analyze these sequences in order to see the diversity and the taxa assignment and also the functional annotation. So in this figure here, we can see that the diversity of the sample compared between healthy and IBD, and on that side, healthy and IBD subtype. And we can see that the diversity where in between the alpha, the top part is the alpha diversity and the bottom part is the beta diversity. And we could see that there is a statistically significant difference, lower CHA1 index and Shannon compared between healthy and IBD. But when we look at the IBD subtype, we can see that the CHA1 only saw a statistical significance, or lower statistical significance in the case of CD. But beta diversity showed that a significant separation between IBD and healthy, and also CD and UC between healthy. And this actually is not a new finding. This actually has been reported in many studies before. Then we moved into the taxonomic profiling. We have identified more than 20,000 OTUs. Among these, 1,654 genera and 5,274 species. And here we're presenting the identified, or the top 20 identified genera, both on individual samples and the mean in each group. And if we look at the identified genera, we can see that clearly they are the major bacterial genera present in the gut, which validates actually our analysis pipeline. Now we compared the abundant genera and species between IBD and healthy individuals. And here we're presenting the linear discriminatory analysis that shows on the blue bar that these are microbial taxa enriched in healthy individuals, and the red bars show those enriched in IBD. And you can see that there is red stars beside the bacterial taxa, and this means that those are actually known to be associated with IBD, so either at the genus or the species level. And the ones with the orange star, it means that they have emerging, or they are being recently reported to be associated with IBD. But there is many others that has never been identified or reported in IBD. So what does that mean? Are they unique to the Saudi population? Maybe. Could they be used as a biomarker? Also maybe. But we have to confirm or these findings with much more higher number of samples. Then finally, we looked at the functional categorization for these microbiota, and we can see that here is the Cook functional categories for these taxa, and we can see that the top functional category that is assigned is the function unknown, and the ones with the stars are either differentially abundant between CD, IBD and healthy, or CD and UC are also been reported before in the literature, so there is overlapping. Here, and when we looked at the function, we found more data that actually goes with the literature, which means that although there might be a difference in the dysbiosis in terms of taxa, but the function in which they actually doing is consistent between the different literature. In different literature. Actually, right now, we are working on looking deeply in the virulence genes and the antibiotic resistance genes in order to see if there is a specific signature of antibiotic resistance and virus genes in the IBD cohort. So with that, I would like to conclude that certain bacterial taxa we have identified in our cohort that are similar to what is being published, and many others are actually unique to our cohort. There are several or 10 categories that were differentially abundant between the groups. So what could be the outcome of this? If we actually join forces and we actually build this UN database, we can actually have our own Saudi biomarkers. We could have our own Saudi personalized microbiome-based either therapeutics or probiotics. And by the end of this, I would like to acknowledge the people behind this work and the VIU unit. And of course, our collaborator, Prof. Arna Payne, Dr. Sagar Atroub, and my former PhD students, Shaima Al-Hazmi, Prof. Umar Saada from King Abdulaziz University Hospital. And most of the work or the work that I presented today was actually generated by Bashaira Attas, which is a PhD student in my lab. Thank you so much.
Thank you, Dr. Turki, for the informative presentation. It's my pleasure to introduce our next speaker, Dr. Hamza Tayyib. She's a clinical scientist. She is the head of functional genomics section at King Faisal Specialty Hospital in Riyadh. She's also the chair of the board of directors of the Metagenomics and Microbiome Association, MMA. She holds a PhD in molecular epidemiology from the University of Surrey, with postdoctoral work in multi-drug resistant pathogens and pharmacogenetics. Her work focuses on understanding infectious diseases as well as leading several national genomic initiatives. Today, she will be sharing her insights on the implementation of Metagenome as a diagnostic tool in infectious diseases. Please join me in welcoming Dr. Hamza.
السلام عليكم ورحمة الله وبركاته. مساء الخير جميعا وشكرا على الدعوة الكريمة. As Dr. Khalil said, the title of my talk is Implementation of Next Generation Sequencing. I'm going to give a brief actually about our experiment on having next generation sequencing, third generation sequencing to be diagnostic or clinical diagnostic tool in King Faisal Hospital Center of Genomic Medicine, exactly in the microbiome and metagenome section. So our vision is to reach what we call zero undiagnosed infection, starting with King Faisal Special Hospital. And that what guide us to the common question. Dr. Majid tried to answer some of that question in his talk, saying, why do some infectious remain undiagnosed? Why do we have to develop such kind of expensive, let's say, technology, whole genome sequencing or next generation sequencing? It's not a cheap one, or it's not an easy one to be performed in every lab. Well, the first reason is what we call one bug, one test. So we know it's in each lab, the significant diagnostic test is designed for certain bug. And this is not covering everything we may find in the sample from the infection and the co-infection and the antimicrobial resistance. The well-known sequence mutation, especially in viruses, and that's what caused the COVID-19 problem and still causing this. And it's actually present in every and a lot of viruses. It's a type of surviving mechanism, the lack of comprehensive multiplex tests. Some of you may say, well, there are a multiplex test in the microlab. Yeah, but it's really lacking comprehensive or sensitivity. So there are many family or subfamily may escape the detection on this test, the low sensitivity of existing tests, as we were saying before, especially in the low titer sample or uncultured sample. So many documented research document pneumonia, for example, 15 to 25% of pneumonia, meningitis, 60 to 80% of meningitis, diarrhea, up to 50% of diarrheal cases, and as we speak, up to 20% of sepsis remain with undiagnosed causes. All of these reasons take us to start thinking of developing whole genome sequencing. I'm saying again, whole genome sequencing to be the clinical approach for detecting, facing a lot of challenges. We are going to talk about it here. during this presentation. Why not 16S? Why not 18S, Hamza, in your lab? Oh, well, if I do 16S and it will focus on bacteria. If I do 18S, it will focus on fungus. And I may end up with negative result of both, and I will need to do the whole genome sequencing. Also, the same sample may have co-infections for bacteria and fungus. So after I treat the bacteria and I discharge the patient, he will come back again to me with a fungus infection along with the AMR profile. So I prefer to do it once for all with optimizing the time around time, the cost, and give the physician the right report to build his therapy plan for infection, core infection, and AMR profile. Because, you know, this kind of technology, you may detect everything built of nucleic acid material from bacterial, viruses, fungus, parasite, except of course for protein. And we don't do whole exome because for microbe they don't have an exome, so we do the whole genome sequencing, not the targeted panel. It doesn't mean I am against targeted panel, but it is not for metagenome. It can be adopted in the microlab. So after all the samples tested in the microlab and come back to me negative, then I can do the whole genome sequencing. So let me take you briefly and quickly on this step of the subject and the sample collection. And believe me, it is each one very important in giving a feedback on how to make this experiment or this experience successful everywhere as it has been successful at King Faisal Hospital. Because proudly I'm saying Metagenomic now is licensed by Saudi FDA, alhamdulillah, to be a clinical diagnostic system, not a routine one at King Faisal, only at King Faisal Hospital for a first panel. Inshallah, we are working to get the other license to be licensed clinical diagnostic tool in all the kingdom. So to evaluate first, we have to start with which platform. There are many sequencing and sequencer and different platform in the area. Which one we should use? Will we have to put in mind the price? We have to put in mind the coverage. We have to put in mind the time around time. We have to put in mind the quality, the quantity, and a lot of the purchasing, even the existing of logistic presenter for this company here in the kingdom. All of this, it's things we have to test it for each platform to know which one is suitable for our sample and our patient, especially that I'm doing this test for all different type of sample from all different type of body directly from the patient without culturing. So in sample selection for the first phase, because for the limited resources, as I told you, doing optimizing of this kind of test is not something easy or even cheap. So it was only for the admitted patient in the hospital with critical cases. To be admitted at King Faisal Hospital, you have to be in a critical cases. So if those patient was negative, their result, first of all, they have the symptoms of infection. Their symptoms due to infection, not due to some side effect of some therapy or chemotherapy. No, they have an infection and the results came negative from the lab, so and they are not responding, because some of the sample or some of the patient, they have negative results. However, they respond to the antimicrobial therapy. So this sample, I don't look at it. If the sample is negative and patient not responding, then the sample will be tested for metagenome. The other case scenario, if the sample are positive by the lab, so why should it be sent to the metagenome test? Well, if those patients are immunocompromised and their sample was positive and still not responding, so immediately on the spot I will do the metagenome. I'm not going to wait for the AMR profile from the microlab because it's immunocompromised patient. However, if those patients not immunocompromised, I will wait until the AMR results came from the microlab. Patient is still not responding, then I will do the whole profile for metagenome. This is for the sample selection, and using the inclusion and exclusion criteria, I'm trying to be very tight. Per year, I received like 300 samples with undiagnosed microbe. Only 50%. You will see in the result, only 50% per hand. Now the sample selection. It's very important to have the right dialogue with the physician, because some physician, for example, the patient have respiratory infection, or and all of a sudden I have a CSF sample from this patient, or they have leg infections. I have different type of samples. So we have to have a communication. All type of sample, it has to be guided by the infection site. For example, the CSF brain tissue is preferred for meningitis, while the pan and sputum is ideal for the lung disease, so on so forth. Of course, and the correct quantity of the sample for 1 ml or 800 microliter minimum. Now I am going to go immediately to the wet lab. So before starting working on the sample, again, let's not forget that my goal here is to use this technology for a clinical diagnosis of microbe or pathogen. I'm not using it for research. So I won't do the whole genome sequencing for everything in the sample, including the human. It will cost me a lot. I will be able to use this flow cell for 1 sample instead of using the flow cell for 5 samples, for example. So what I do, I demolish the human DNA. Yes, it can be a very good information helping in treating for some kind of patient, especially the oncology patient. But for me, I demolish the human DNA so I can focus on the pathogen or the microbe nucleic acid material. I can use the flow cell for more than 1 sample, for example 3 to 5 samples for Oxford nanopore or for Illumina, and I use for this some physical treatment. تقول لي 5 minutes. لا طيب. Okay, then of course the DNA and the RNA extraction and transferring the RNA to complementary DNA to double strand. No sequencer recognize the RNA. All the sequencer recognize only the DNA as a double strand material. Now let's go to the machine itself. We have I use, for example, for the long read and the short read. For the next generation, for the short read, I use Illumina. First, I use NovaSeq, and then I use MySeq. Why I change from NovaSeq to MySeq? For NovaSeq, I have to wait for 20 samples. The physician will not wait for me until 20 samples were collected, and then he get the result. The patient will pass away by that time. But for MySeq, I can do like 5 or 4 samples per the flow cell, so I go for MySeq. The expenses of both of them almost the same, but the around time here is 73 hours. Here I will optimize it to be 24 hours or 27 hour maximum, which is very essential for our work. So this is the comparison between the same platform, Illumina, MySeq, and NovaSeq. And here I am trying to skip for the time matter. This is where I try to summarize the time I use for extraction and demolishing and doing this and that, because as I told you, one of the things I try to optimize is the time around time to be able to give the result using Illumina, for example, platform to the physician within 2 to 2.5 days maximum. Let's go. And this is one of the papers we published during our work. Actually, we published 5 papers during this year. One of them was Barchivaris, Barchivaris causing infection in the gut, but it's I migrate to and cause some encephalus infection in one of the patients. She was in coma, not responding, and alhamdulillah, after she's been treated based on this result, she transferred to the war. She was from Bahrain actually, and then she moved to her family in a good health. Now this is the Oxford nanopore. Oxford nanopore, the 3rd generation sequencing. Now theoretically long read, so it gives me more coverage for pathogen. That each fragment it may cover the whole pathogen in one shot. So for me, for the same coverage of 25 million read of the flow cell, I can do, instead of 6 samples I use in Illumina, I can do up to 8 to 9 samples with Oxford nanoread. Along with they have, as they claim, now I'm telling you that the benefit, that why I used it. They say that they have the real-time analysis, meaning during the sequencing, I can see the outcome, I can see the results. Stop, show me this, stop, show me this timing. I can see the outcome, I can see the result, and I will be able to pick up the phone and tell the physician, okay, look, eventually your patient having this, I will come back to you with the whole report within a few days, because it's built in epitome third program party. And also it takes 24 hours and it's cheaper than Illumina. So what's happened after I try it on basic or on real pinch? Yes, it is very good. It is very significant. 24 hours, it cost me 2,000 real, or including the analysis, while Illumina cost me 5,000 real, including the analysis for the each sample. But the real time still we have to development in-house actually, because epitome, the real time or the third time program, it's have no viruses database, and it's work more with the human but not with the microbes, so we cannot rely on it. Here I am summarizing all the obstacles we have, how we go over it, and this I will send it to you. I have no time to go over it now. So let's go for the dry lab. The dry lab, it's really very important, because if I want to do this manual, as I did, I published in Metagenome actually first in 2017 with one of transplant patient. But for the analysis to be done manually, it took me 2 months. No, so it's a research study. For a clinical diagnostic, no, you need a third program party or a program that you can use and give you the result within a few hours, even half an hour or 1 hour. So I tried to test all the third program party and their significance, their filter provided on the market. The best one was the PFI from BGI and Cosmo ID, and then I decided to go with Cosmo ID because BGI have a lot of problem with their filter. I don't want to upset any company here, but I'm just telling what we find and what we use. So for Cosmo ID, they do the analysis for the metagenome and they do the analysis for microbiome. This is the manual thing, but let's go to the reporting and the result. It's more important. Here is the result. This result is actually old one before we collect the 300, but for the percentage, it's almost the same. 50% of the sample we received with negative outcome of microbe or pathogen, we've been able to identify 50% of this sample, and 50% of the positive sample was also positive for antimicrobial resistant profile. This is the sample we run both using Illumina and using, or let's say long read and short read. We compare between each of them, which one should be used and why. And let me tell you this. I'm closing. So for Oxford nanopore, actually it's good for all kind of sample, except for the sample with a low concentration, lower than 4 nanogram. You cannot go ahead with Oxford nanopore native sequencing. You should go with Illumina for example for CSF sample. It's more significant. This is all type of sample we are doing, and it's already published. You can see in details on the PubMed. As I say, up to globally successful of metagenome is 20 to 50%, and alhamdulillah we are reaching the 50%. Here is some reference using the whole genome sequencing and how they succeed to other findings. Also, I will skip this conclusion to some of the excited results. Actually, this is a patient who found to be positive for Klebsiella by the microlab and by us. However, the patient did not respond and he passed away. When we get the sample, we find that there are a co-infection with Shigella and even a resistant mechanism of Shigella that the patient not being treated with the right antimicrobial to it. There are another patient, this paper, it's already also been published for kidney transplant, infected with Malassezia restricta, and it escaped the detection also in the microlab. And we had to open the teenager again and remove the kidney after they've been positive for this fungus. Also, it's been published, as I said, urea plasma for a patient. He was in a coma, and alhamdulillah, now he's in a ward. Another patient with noroviruses also immigrate from the gut to the brain, causing encephalitis. And when we test the stool, yes, the patient was positive for noroviruses. Another study being done on outbreak infection with Pseudomonas in our hospital, and we find, alhamdulillah, the source. This is the case. We find the source of infections, the 0 sample infection, and we manage to help containing this outbreak. So with all of, as you can see, this is for the outbreak infection in the hospital. We can, with all different antimicrobial resistance that been detected in the microlab, we get all the information and details about this infection. Summarizing, metagenome, next generation sequencing, long and short read is the approach for the diagnosis of infectious diseases. The infectious bioinformatic pipeline is essential and very important, and no metagenome or microbiome will succeed without it. Next generation sequencing or third generation sequencing for pathogen detection and discovery is only the first step. Many regulatory and technical challenges remain. Thank you very much. I would like very much to thank my staff in the lab, Dr. Suzanne Azir, Dr. Razan Bakhid, and Ms. Amna Majdi. Thank you very much for your listening.
Thank you, Dr. Hamza, for your informative presentation. Next event, you should join us as a keynote speaker.
Yes.
Thank you.
Okay, thank you, Dr. Hamza, all previous speakers for insightful talks. It's now my pleasure to introduce our final speaker for this session, Dr. Sharif Ala. Dr. Sharif is Director of Biothreat Department at Public Health Authority in Riyadh. Is also General Manager and co-founder of Diagnostic and Discovery Company in Jeddah. He's visiting researcher at KAUST and also the head of the Infectious Disease Research Department at KMARC. Dr. Sharif holds PhD in Biological and Environmental Science from KAUST. His research focused primarily in genomic surveillance and diagnostic, addressing the critical issue in infectious disease, antimicrobial resistance, and preparedness for pandemic and biothreats. Today, Dr. Sharif will speak to us in topics advancing pathogen surveillance, tracking nosocomial and AMR in infectious disease and pandemic preparedness. Please join me in welcoming Dr. Sharif.
So thank you very much. It's kind of hard to come after Hamza because we do a lot of work together. Also, Dr. Majid. So it was kind of hard to pick the topic, but being the last speaker, I thought I'd give you an overview of what pathogen genomics, how does it work and what is the outcome of it. So because the reaction that I get from a lot of students and a lot of PIs who don't work on pathogen genomics per se, the reaction of what we call NIMBY, not in my backyard. So what? I want to show you here some of the data that we're working on. Not all of it, of course, because of the time. But I want to show you how can we make pathogen genomics simple. We would really know because in the past couple of years I have collaborated with multiple scientists and places that did the sequencing, collected the sample, but there was a flaw in the design itself because the question is what we want from it. And I want to convince you today, hopefully, that pathogen genomics can be used in public health, where I work, or it can be used in the clinic, like what Dr. Hamza said, or it can be used in defense, or in the environment, or in wastewater, in so many different places. So this is basically how I think about metagenomics or pathogen genomics. And by the way, I am a recent convert into the metatranscriptomics. So I really like to focus on the total RNA, even in diagnostic patients, because looking at the total RNA instead of the total DNA, you also cover the viruses. You also see the different expression. This is some of the work that we do in public health or in research. There is no one entity. It's not like other research. You do need the bioinfo lab. You do need statistics. You do need an actual consortium. We did successfully did that during COVID in our published papers, where we collaborated through several entities together, and that's what the message that I want to drive today. So I'm going to give two examples, just from the data that I know that what we've done with it, and this is only just a little tiny amount of the data. One I'll be talking about flu, the other one I'll be talking about AMR. And what I want to explain here is, and coming after Professor Mamish talking about anything related to WHO, it's just not gonna satisfy you. But I just want to show you that in Saudi, actually, even though we are blamed that we're not advertising a lot of the work, however, there is a strong foundation in there. Yes, there is a lot of points that we need to take from the talk of Dr. Ziad Mamish, where we need to share the data, we need to analyze the data better, we need to do benchmarking and so on. However, what I have seen in the past couple of years working on genomics and metagenomics is that actually the understanding and the urgency of using such thing for every single pathogen, not only flu or COVID or MERS, is actually has reached the top now. So we should expect in the next couple of years a lot of changes. I want to show you how we deal with it. So Dr. Hamza showed you how they do it in the clinic. Dr. Majid showed you how to do it in general. I want to show you what is the real life example of every day of what it takes to get the sample actually and to get the final result. Sometimes the results could be as testing the targeted kit that you have in the clinic, or sharing some data with the WHO. And by the way, I forgot here to add the new Roche machine, which is something that we're waiting for. It does, like, I don't know, 7 samples in 3 hours or something crazy. So to give you a quick example in here, this is some of the work that you do. You have to target specific places where you think that that pathogen is, in case of MERS or flu. There is a lot of work that goes into the logistics of getting the sample. This is something that we suffer from in the clinic, or even around the kingdom. The laboratories itself, would you go for PCR, would you go for sequencing, and so on. And then the data analysis itself is a complete department on its own. Similar work, we have applied it on the wastewater, which Professor Han spoke about in the morning. This is something that of urgency to the Ministry of Health, to public health, to researchers, and it comes directly from 2 different places: the urban sites and the aircraft wastewater. There is a lot of logistics and protocols that are open for students to work on and to develop, especially in Saudi Arabia here. Some of the data is extremely encouraging, and people who were here in the morning and attended Professor Hong could attest to that. The bioinformatics itself, many of and I was just speaking to my colleague, Dr. Naif, here. He's asking about what is the best bioinformatic tool for metagenomics. And I usually make it extremely simple for students. Almost all the tools that are made or given by, well, Professor Arnab would be mad at this, but it's based on Kraken, which is one tool that many of them based on it. However, the idea of metagenomics in general, and this supports Turkey's work, is that you have a puzzle and you want to compare it to a database. If I have a black piece, what is it here in the database, pink piece, and so on. This is the shape of it. So if we don't have our own Saudi database and collaborate to have it, no one will have advantage. Not in the clinic, not in the environment, nothing. So this is basically where I'm coming from here, and not even the public health. So what can I use pathogen genomics on? Yes, you can use pathogen genomics, and I mean it here. Not only metagenomics, just all sequencing, creating database right now. There's a project that we're working on prediction of using the data of 2021, 2022, and so on. Can we predict what 2026 will have in terms of pathogen or mutations of the virus? Would that help us in creating vaccine? So it's just in general, because I always hear this blame from a lot of scientists, and I understand this frustration comes from the love from how much they love their work, but the effort that I have seen being within the research area, with previously in National Guard in one of the big institutes, is that the effort is there. We just need to collaborate, and we need to talk to each other. I want to show you some of the results here. So I think, hopefully, by this time, I convinced you that pathogen genomics is important. I don't want you to get into the specific and the nitty-gritty data. This slide, probably I could discuss it with Prof. Anwar for hours upon hours, but I want to tell you that this data comes from public health, from the public health perspective, is that we want to know, is the kit that we're using to test during Hajj, during Umrah, within the hospital, does it actually capture what we see? That simple question. Are we looking, when we do the one bug, one test, when it's important, like flu, is it working? And this is only one part of the metagenome of the pathogen genomics that we do it continually. Someone else could take this data and come up with a whole new idea of it. So hopefully, by giving you these examples, like in here, we can understand what kind of flu A type that we have here, what lineage, what subtype we have. Does this work with the vaccine, instead of just relying on international data? No, we can actually ask what vaccine we have and does it apply to what we have. Moving on to AMR, I'm not going to convince you that AMR is the big, bad, ugly pathogen or pathogen agent, but it's just we can't really fight what we don't understand. We still, until now, this small effort, and I'm not saying small by the effort itself. I'm saying small by the number. This is something that Dr. Memish specifically said that we need to increase the number. So we thought about this. So I had two of my students here start this project about two years ago. Started collecting a lot of Klebsiella pneumoniae with collaboration with KAUST, Mohamed Malak in here, and Umnia Fallata. We tried to cover the majority of the kingdom, to just see what we see, to see what kind of Klebsiella we have. And just like what everybody says, yes, I can identify pathogen genomics, and the pathogen itself and the sequence type itself, but without the complete picture and the complete data, it's just not going to help. For the interest of time, I just want to tell you that yes, the thing that we when we use the right tool, when we use the exact data, we could understand that yes, there is a top strain that we see, and we did publish this, the ST20996, and the death rate associated with it is massive, as you can see. Just for the interest of time, I'm not going to get into the data, but if we look deeper, we see that this strain is actually only was at that time, at least it was reported in India and in Turkey. And not only this strain, when we dig deeper and look through pathogen genomics, not only this strain is resistant, like what everybody says, it's also hypervirulent, which is another whole problem that needs a lecture on its own. Of course, when you look at AMR, looking at plasmids is the most critical one, because that's how AMR goes from patient to patient. And this data, yes, this is the hypervirulence slide. So just a person who doesn't know what hypervirulent is, that's how it looks on the plate. We looked at it and we associated with the data, and we found that the strain itself gets a lot worse the longer the patient stays in the ICU. And it just actually clicked with me while Dr. Hamza was talking. The faster she can get the patient out of the ICU and out of the hospital, the better it is, not only for the patient but for the public. So understanding such data and focusing on it is no longer optional. We're thinking that pathogen genomics, it cost us 2,000 or 3,000. In my opinion, yes, maybe 5 years ago, if you see that the sequence costs 3,000 right now, some of the X-rays and some of the visit to the clinic cost that. So if it can save life and it can save the whole, it can give a lot of information to the public health, then sure. I want to talk about AMR is not only what we're looking at in general. This is the message that I want to say today. It's not only the carbapenem that what we know. Things are changing, and a lot of data we can see it in wastewater, and we can see it through patient sample. The profile is shifting, so I'm urging everybody who works in AMR to keep an open mind. Of course, I'm not going to talk a lot about the metagenomics, but people who are in the field, I want you to also look at the metatranscriptomics, where it also can give you a lot more info. And we wanna lay the groundwork basically to have an. I think this is our job right now, is to build an actual databases, good info. Yes, we can have short-term goals, but the long-term goal is to have an actual database that is shareable and accessible to everyone. Thank you so much. And I just wanted to thank some of the collaborators in here from the National Guard, from PAIA, from the Ministry of Health. These people have stuck with me through thick and thin since probably 2013. So I just want to give them their proper thanks. Thank you.
Sequencing, it is cost 5,000 or 3,000 each day of the admission of ICU patient in the hospital. It costs minimum 10,000. That if we want to talk economically, and rather than health-wise. So it's worth to use it to save life.
Candy, I would like to ask the speakers to come to the stage, please. Unfortunately, we don't have enough time for the discussions. Due to the preparations for the official opening ceremonies with the president of the university. Please. I would like to ask you 12:45, please. We would like you to be with us here in the main auditorium, just for the kickoff of the official opening ceremonies by the Master President of KFMRC. Thank you. We'll see you.
Excuse me, I just like to ask everyone to come back here at 1 for the opening ceremony. 12:50? 12:50.
هو يجي 50 احنا نجي 5.
يا دكتور طبعا قصدي وين كلهم والله طيب
كده وبعدين اطفاء
لا قلت المسؤولية تمام
انا نسمع عنك لكن نشوف الكلام والله كلك خير وبركه الله يبارك
كرسي جنبكم
ايوه المصورين بيجوا هنا وصور عن
محضورات في السيارة كانت السيارة
اللي كان في حادث
يلا لسه باقي ربع ساعة.
place now again. So it is assessment differences that if we see the deserve the good seeds, get the good seeds. We'll start in a few minutes, inshallah.
السلام الملكي. بسم الله الرحمن الرحيم، والصلاة والسلام على أشرف الأنبياء والمرسلين سيدنا محمد وعلى آله وصحبه أجمعين. سعادة رئيس جامعة الملك عبد العزيز الدكتور طريف بن يوسف الأعمى، أصحاب السعادة، السيدات والسادة، الباحثين والعلماء الكرام، ضيوفنا الأعزاء، السلام عليكم ورحمة الله وبركاته. يسرنا أن نرحب بكم جميعا في حفل افتتاح مؤتمر مركز الملك فهد للبحوث الطبية الأول. هذا الحدث العلمي الذي يجمع نخبة من الخبراء والباحثين لمناقشة أحدث ما توصلت إليه العلوم الطبية وتبادل المعرفة، إيمانا بدور البحث العلمي في دعم مستقبل الصحة وجودة الحياة. نفتتح لقاءنا اليوم بآيات من الذكر الحكيم يتلوها الطالب أنس بن يوسف عبد الجواد، فليتفضل مشكورا.
أعوذ بالله من الشيطان الرجيم. إن في خلق السماوات والأرض واختلاف الليل والنهار والفلك التي تجري في البحر بما ينفع الناس وما أنزل الله من السماء من ماء وتصريف الرياح والسحاب المسخر بين السماء والأرض لآيات لقوم يعقلون.
صدق الله العظيم. والآن ننتقل معكم لمشاهدة الفيديو الافتتاحي الذي سيعرض مسيرة وإنجازات المركز ورؤيته الطموحة نحو مستقبل مشرق في مجال البحوث الطبية.
نخلق بيئة جذابة وجيدة ورائعة في وطننا، كيف نكون فخورين في وطننا، كيف وطننا يكون جزء مساهم في تنمية وحراك العالم، سواء على المستوى الاقتصادي أو على المستوى البيئي أو على المستوى الحضاري أو الفكري إلى آخره. هذا طموحنا، وسنقدم كثير للسعودية وللعالم.
أنشئ مركز الملك فهد للبحوث الطبية بناء على الأمر السامي الكريم الموافق 1980. تتلخص مسؤولية المركز في تحسين ظروف المجتمع الصحية، وذلك بتطبيق عدد من الأبحاث الطبية الحائزة على امتيازات عالمية لتحسين ونشر الوعي الصحي. يخدم المركز مستشفى الجامعة بالإضافة إلى كليات ومراكز الجامعة والجامعات الأخرى، كما يخدم الجهات الحكومية والخاصة على حد سواء. الريادة في الأبحاث الانتقالية والتقنية الحيوية لمواجهة التحديات الصحية العالمية. ترجمة الابتكارات العلمية إلى حلول صحية فعالة من خلال الأبحاث الرائدة وتنمية الكفاءات العلمية. يضم المركز أكثر من 34 وحدة بحثية ومعمل مركزي، شراكات مع أكثر من 90 جهة محلية وعالمية، أكثر من 4000 ورقة بحثية، أكثر من 55 براءة اختراع. نشأت فكرة هذا المؤتمر لتستهدف بناء جسر متين يربط بين أحدث الأبحاث والتطبيقات العملية في المجالات الصحية، لتبقي الممارسين الصحيين على اطلاع بآخر التطورات والاستفادة منها في تطوير الخدمات الصحية. يستهدف المؤتمر المهتمين بآخر التطورات والأبحاث المنشورة المتعلقة في المجالات والمحاور التالية: أمراض الدم والأورام، الأمراض المعدية والتأهب للأوبئة، علم الأعصاب والشيخوخة والأمراض المزمنة، التقنية الحيوية والابتكار، السموم والعلوم الجدرية، الأشعة والتقنيات المتقدمة. أكثر من 100 متحدث، أكثر من 200 ملصق علمي. تحسين جودة الحياة من خلال البحث والابتكار.
حضورنا الكريم، نبقى الآن مع كلمة سعادة مدير مركز الملك فهد للبحوث الطبية الأستاذ الدكتور أنور هاشم، فليتفضل مشكورا.
السلام عليكم. بسم الله الرحمن الرحيم. الحمد لله رب العالمين، والصلاة والسلام على سيد الأنبياء والمرسلين نبينا محمد وعلى آله وصحبه أجمعين. أصحاب السعادة، رئيس جامعة الملك عبد العزيز الدكتور طريف بن يوسف الأعمى، الضيوف الكرام والزملاء الأعزاء، بالأصالة عن نفسي ونيابة عن مركز الملك فهد للبحوث الطبية، يسعدني ويشرفني أن أرحب بكم جميعا في مؤتمر مركز الملك فهد للبحوث الطبية الأول 2025، والذي يعقد برعاية كريمة من سعادة رئيس الجامعة وبدعم من سعادة نائب رئيس الجامعة للدراسات العليا والبحث العلمي، وذلك في رحاب جامعتنا الحبيبة جامعة الملك عبد العزيز تحت شعار تحسين جودة الحياة من خلال البحث العلمي والابتكار، وذلك بالشراكة مع شريكنا الاستراتيجي الجمعية السعودية لأمراض الدم ومع معهد الجينوم الطبي ومركز الابتكار في الطب الشخصي. ينعقد هذا المؤتمر في ظل التحولات الوطنية الطموحة التي تقودها مملكتنا الحبيبة من خلال رؤيتها 2030، حيث أولت قيادتنا الرشيدة بقيادة خادم الحرمين الشريفين وسمو ولي عهده الأمين حفظهم الله اهتماما بالغا بتطوير منظومة الرعاية الصحية والبحث العلمي والابتكار والتقنيات الحيوية. ونحن هنا اليوم لتجسيد هذه الرؤية على أرض الواقع من خلال منصة علمية تجمع بين التميز الأكاديمي والتقدم الطبي والريادة البحثية والعلمية. فخلال 5 أيام سيستعرض هذا المؤتمر أحدث المستجدات في محاور بحثية عدة تشمل الأمراض المعدية والاستعداد للأوبئة، التصوير الطبي وعلوم الأشعة، علوم السموم والعلوم الجنائية، أمراض الدم والأورام، الشيخوخة والأمراض العصبية والأمراض المزمنة، التقنية الحيوية والابتكار. لقد حرصت اللجنة العلمية على دعوة واختيار نخبة من الخبراء والعلماء من داخل وخارج المملكة لتقديم محاضرات ومشاركة أبحاثهم الرائدة في جلسات حوارية وعلمية وتفاعلية تهدف إلى نقل المعرفة وتعزيز التكامل البحثي وتحفيز فرص التعاون المشترك. To the esteemed guests and colleagues, as the director of King Fahd Medical Research Center, I deeply believe that innovation flourishes where minds meet and ideas are shared. This conference is not just a showcase of scientific progress, it's a call to action. I encourage each of you to listen actively, ask boldly, collaborate openly, and envision together how we can translate scientific discoveries into better care, health, and future. Thank you very much.
حضورنا الكريم، في هذه اللحظات لا يسعنا إلا أن نعبر عن بالغ شكرنا وامتناننا للرعاة الكرام الذين كان لدعمهم السخي دورا أساسيا في إقامة هذا المؤتمر. نستأذنكم سعادة رئيس الجامعة الدكتور طريف بن يوسف الأعمى بالتفضل بالصعود على خشبة المسرح لتسليم دروع التكريم. الراعي الذهبي شركة GSK. الراعي الفضي شركة AGBL. شركة دكتور لابراتوري. شركة توريدات الخليج للأنظمة الأحيائية. شركة عبد الله فؤاد للوازم والخدمات الطبية. شركة غندور. وكما لا يكتمل النجاح إلا بتكافل الجهود، لا يسعنا في هذا المقام إلا أن نكرم شركائنا الأعزاء الذين كان لتعاونهم المثمر بصمة واضحة في دعم مسيرة المركز وتحقيق أهداف هذا المؤتمر. فليتفضل شركاؤنا الكرام للصعود إلى المنصة لتكريمهم. الشريك الاستراتيجي الجمعية السعودية لأمراض الدم، معهد علوم الجينوم الطبي، مركز الابتكار في الطب الشخصي، مركز الاتصال المؤسسي،
يعطيكم العافيه ما شاء الله مبدعين. لا تقول لهم لا تفرحوا معليش. يعطيكم العافيه. شكرا لك.
وبهذا نعلن معكم رسميا وبكل فخر انطلاق فعاليات مؤتمر مركز الملك فهد للبحوث الطبية الأول، متمنين للجميع مؤتمرا حافلا بالنقاشات المثمرة والحوارات العلمية الراقية التي تليق بطموحاتنا ومسؤولياتنا تجاه صحة الإنسان والمجتمع. الحضور الكريم، نستأذنكم بالوقوف في أماكنكم استعدادا لالتقاط الصورة الجماعية. شكرا للجميع، كان معكم صهيب بن يوسف عبد الجواد، والسلام عليكم ورحمة الله وبركاته.
Now we're starting our third session for today. Our moderators. So our moderators for this session are Dr. Ahmed Mahmoud, who is an associate professor and director of Health and Life Research Center at Taiwan University. His research focuses on cancer immunotherapies, vaccine development, and oncolytic viral therapy. And our next moderator is Dr. Abdullah Al-Qaisi, who is also an associate professor of virology at Jazan University, where he also serves as a director of Health Research Center and lead the emerging and endemic infectious disease research unit. His work focuses on the epidemiology and pathogenesis of emerging infections, infectious diseases with extensive experience in vaccine development for coronavirus like MERS-CoV and SARS-CoV. So right now, I'd like to ask our moderator to lead this, the lead in moderating this session.
السلام عليكم ومساء الخير عليكم جميعا. So we're going to continue our discussion with session number 3 in infectious disease and pandemic. And our keynote speaker for the first talk is by Prof. Arnold Payne from Pathogen Genomic Laboratory at KAUST. So Prof. Payne received his PhD from the University of Cambridge, UK, working on microbial pathogenesis in bacterial pathogen of cultivated mushroom. He had his postdoctoral training in Oxford, and then he did his postdoctoral training also at Wellcome at Sanger Institute, at Wellcome Trust Sanger Institute, and he is well known globally for his research in microbial pathogenesis, especially in malaria, Plasmodium falciparum. So today he's going to talk about his work at KAUST. His research group uses a combination of high throughput sequencing, comparative and functional genomics, protocol and bioinformatics analysis tools to study pathogenicity determinant in selected parasite and bacterial and fungal, and more recently with SARS-CoV-2 and Dengue, as well as emerging infections. So please join me to welcome Prof. Arnab for his talk beyond COVID-omic. We're going to talk about omics in other vector-borne disease in Saudi Arabia. Welcome.
Assalamu alaikum and good afternoon to you all. And I first, before I start, I thank the organizers, particularly Professor Anwar, Professor Assam, and Professor Turki, and also other colleagues here to give me an opportunity to come and present some of the work that we are doing. And as it has been said time and time again, that the idea is to set up new collaborations or even strengthen the existing collaborations. And also thank you for the introduction. So I am let me see how I move. Yes. So I have actually divided the topic into some that is already we have been undertaking over the years, and that relates to various pathogen genomic driven work. And I'm going to just introduce you what we generally do. We have been doing over the last 15 years. It's just about next month I will be finishing 15 years in KAUST. I'm one of those founding members of the founding faculty there. And then, of course, what I will share with you what we did during the COVID pandemic, and then eventually a brief overview of the surveillance that we are doing. That's the main topic of the day, if you like. And finally, it's already been very nicely put together by various speakers earlier about metagenomics-driven work on discovery of pathogens in clinical and also in more recently in disease vectors. So how do I? Okay, the first one. So our group has been working on various aspects of disease, as already been alluded to. Malaria, tuberculosis, and non-tuberculous mycobacterial diseases have been a significant focus since I moved, and also before moving to KAUST. More recently, COVID-19 and dengue. I'll show you some of the results that we have been generating over the years and in the last few months, if you like. Of course, nosocomial infections, AMR, is a very major problem, not only this part of the world, but globally, and major, it's very important aspects of health-related research. Metagenomes, again, Professor Turki Abu Zamel talked about our collaborations. I hope to make it even much, with his leadership, it would be, I hope that this will really go into a much bigger scale in future. And also finally, some work that we have just recently done, and it is showing a lot of promise about how we can detect pathogens in any type of samples. But in this case, I'll show you how we can detect pathogens in mosquitoes, for example. So basically, they are like flying syringes when they have something to inject you on you when they bite. So we work on various aspects in the group, but for the purposes of today's talk, I just stick to two areas, which is I just told you about, and then of course what we routinely do, we use really the power of next generation sequencing technologies, various types of it, Illumina, ONT, and also sometimes Pacific Biosciences platforms that we have access to in KAUST. And then, of course, we have a few bioinformaticians in the group. I also work with other expert bioinformaticians as collaborators. And all of these things that we study, they have a specific footprint, if you like. And then studying those footprints, we get some clues, and then we use them like functional genomics and cell biology tools. Hopefully, during the course of my talk, you will know how we can, starting from genomes, all the way we are going into mechanisms for some areas. So, of course, we are extremely fortunate to have major collaborative network and includes many in Saudi Arabia, and I'm very happy and proud to say many of my collaborators are actually sitting in this audience here. And also we are now planning to develop some training workshops. So anything over the years we have learned while dealing with pathogen genomics, from our success, also from our mistakes, we want to share with you in forms of some training workshops. I hope to be able to come with this before end of this year for sure, maybe earlier, next summer. So, I'm slightly stuck here. It's not working. Okay, so the COVID. Now, so of course, genomic surveillance, why we need, not only for coronaviruses, but for any pathogens. Of course, we need to track the viral mutations and their recombination rates. We identify the transmission pathways, whether it's animal to human or human to human, or even intercontinental spreads. Of course, emergence of variants with increased transmissibility of virulence is very important. Again, by systematically surveying the genomes of these pathogens, whether it's DNA containing genome or RNA viruses, for example, it's extremely important. And of course, you know, the results that you get out of it, they are important for public health interventions and also vaccine development. For example, the last COVID vaccine I received, the spike protein is Omicron spike, not the original one. And I think many of you actually have received the same. So what we did, basically, at the start of the pandemic, again, these are mostly published, so I'll go very quickly on that. So we basically collaborated with various organizations within the kingdom, basically 9 hospitals in Saudi Arabia. We collected nasopharyngeal samples. Ultimately, we just did the sequencing, and eventually the sequences ended up in GIS AID databases. Of course, this is a massive program, you can see, and I particularly want to refer to Dr. Sharif Hala, who used to be part of my team at that time. He played an absolutely critical role here. So thank you, Sharif, if you're still in the audience somewhere here. And of course, Sarah Mentorez, she at that time was lab manager, one of my PhD students. We collected large number of samples, we sequenced them. We also looked at the RNA copy numbers in all these samples, and these are all Saudi samples. And then we have a very talented bioinformatician, Tobias Moore here. He ultimately found, by doing a lot of data crunching, that actually if there was a particular mutation in the nucleic acid gene of the virus, those patients suffered more severe disease, and it actually resulted in higher virus copy number. So that resulted in a series of, I proposed a large number of projects. That's why I put the COVIDomics in the title. Again, for the purposes of today's talk, I will not be talking about it for most of my talk, but just to say that what we actually developed a large volume of collaborators, many of them actually present in this audience, and we ultimately ended up in large omics-driven projects, and then again, with support from various organizations, as I have listed here. And then we found, as I told you, that some particular specific mutations were associated with higher copy number of the virus and severity. So this is a very important part of the nucleocapsid protein, which is the most expressed protein in the virus. And then we wanted to see why, just 2 changes in the amino acid makes such a big difference in the disease outcome. So then we wanted to, I have a talented research scientist in the group, Muhammad Shoaib. He really wanted to do a lot of functional work that he ended up in doing large-scale analysis. Eventually, all the work was put together, and this is the first big-scale publication with all this work summarized here, where we show the effect of this mutation with higher RNA binding, more phosphorylation at particular sites, higher level of transcriptional response in cells, if just the virus has this particular mutation in its nucleocapsid gene. And of course, this also gives me a time to particularly acknowledge all my collaborators, colleagues who played critical role here as part of the work. Of course, this was just a beginning of the work. And then we investigated, okay, we already knew which patient had seen which virus, which mutation they had. So now the idea is, could we then look into how the patients, individual patients reacted at the transcriptome level? So this is what exactly we, somehow this is not listening to my command. Okay, so it's taking some time. So we used the patient cohorts, basically the same. We knew which viruses they had seen. Now we wanted to do how the patients reacted. This is nasopharyngeal swabs, by the way, and we call it RG patients that had seen the normal nucleocapsid, and the KR patients which had seen the virus with that mutated form. And what we saw, actually lot of experiments, these are all again published. We saw that the patients who actually had these KR mutations, they had shown significant amount of inflammatory response. And that was a significant finding at that time, that how a viral mutation driven how we reacted to it. So we basically then also went ahead, and this is not part of today's talk, but I wanted to show what we are doing here. We actually now have been collaborating with Liverpool, Cambridge, and also getting this reverse engineered virus, and we have been successful doing that. We have done the mice experiments to show exact results that we showed initially in the patient samples, then in the transcriptome, but now in mice experiments with reverse engineered virus. So now going to what we are doing on the SARS-CoV-2 and the surveillance. So literally genome trapping tracking part of the work, and we didn't want to just restrict ourselves to Saudi Arabia, so we went in the whole region, Middle East and North Africa. So I wrote basically a grant, but why I wrote that grant is that this is as of the 14th or so, end of last year, you see the overall global deposition of sequences. So what you see is our part of the world is extremely this is the global, 17 million sequences, genomes were sequenced, and our part of the world, UK really led the whole lot with about 13% of their whole reported cases sequenced to full genome level, but in KSA only 0.36%. So the idea, I wrote quite a big grant by setting up a big consortium here to increase that number to 2%. And basically, we actually wanted us to count in the genome variants global map. Again, this is just COVID I'm talking about, but it's also true for other bacterial pathogens, AMR, or also even dengue, you'll see in a moment. So what we did was, I called it, COVID Genomics MENA. This was very generously funded by the Wellcome Trust, UK Aid and Foreign Commonwealth Office. And of course, I got a large number of institutions working under the umbrella of this consortium. This was formed in July 2022, just finished the last part of funding on December 31st, 2024. And we actually, the idea was to form consortium researchers, COVID as a pre-example, but we are now working also for other pathogens, for example, dengue and other mpox and other other pathogens of regional and global importance. And of course, one particular thing is the last point. We are trying to build a training component so that whatever we learn together, we then spread the, and I am actually hosting a training workshop of this particular consortium. It was supposed to be next month, but due to some visa restrictions, due to Hajj, it has now been temporarily postponed. So what, oops, so now I have some, okay. So we have been actually and these are the components in Algeria, Kuwait, Oman, Egypt, Morocco, and Qatar. Again, the data you see, it's not only just us, but what has been submitted as part of the global activity in this part of the world. And you can see since about 2020, and the last one is about October 2024, how different variants have been mapped and been submitted. Basically, we are exactly the patterns. If you just follow the color codes here, instead of going into various specific types of Omicron and subvariants, you can see that the patterns almost follow the same across these countries. Let's see what is happening. Okay, another thing is also that these countries have been represented by various cities. So it's actually quite a representative data, if you like, from all these countries here. And finally, what happened in Saudi, exactly the follow pattern, the same as we have seen. Of course, these are again what's been submitted there, and you can see we have significant representation from Riyadh and Jeddah, and also some other, Medina, few from Eastern Province and Qatif, and also some from Mecca. And then, of course, there have been some major effects happening within the kingdom. I'm just referring to what is available in the GIS IID global database. The last on the right side here, the green is our lab actually has done over half of them. And then also King Faisal Specialist Hospital, their effort led by Dr. Fatima Al-Hamlan, they have also submitted a significant number of isolates. Now moving to MERS-CoV. Again, MERS-CoV has already been introduced, and I will be spending very little time on the occurrence. Of course, there was an outbreak, it continues, and in 2024, last year, there were several cases, and I just heard that this year about 2, 3, 4 cases, right? So, so what happened was that we wanted to have a genomic surveillance tool developed. One is targeted, one is untargeted, so that you can develop, you can see not only MERS-CoV, but anything that's there is could be a co-infection, could be any other infection that could be suspected of a MERS-CoV infection. So we use this metagenomic CISPA protocol. We really optimized it pretty well. And again, this work has been led by Mohammad Shoaib and Shabir Adra. And we also came up with our own amplicon-based approach for generating the genomes. And what we did was we did a metagenomic ONT, Oxford Nanopore technology. And then, of course, we used the amplicon-based using 2 sets of primers of 400 base pair each, amplicon if you like. And then what we had was, there was one particular fatal case, which was a reinfection in King Faisal Hospital in Jeddah. And sorry, I don't know how it is moving so fast. So, okay. Okay, so then Professor Ashraf Dada, he contacted us, we collaborated with him, and this was a case where we have received a little bit of sample, and this is the first human, if you like, recent human case of that MERS-CoV in Jeddah, and we actually using this protocol, we managed to sequence both of in 2 complete sequence at 99.5% coverage. So basically the amplicon-based average CISPA method, and you can see CISPA is sequence-independent method. So we can actually, if it was not MERS-CoV, something else, or even SARS-CoV, we would have detected it. So we actually came up with all this optimized protocol. And then this is also Chang Li, one of my PhD student, she also did this analysis where we actually saw in normal metagenomics data, you can see here that, sorry, it should come up, yeah, that Middle East respiratory symptom in the metagenomics data, because this is, you can see, viral reads are seen in the sea of other types of reads, including human reads. So we also did because we have the whole genome, we also analyzed the SNPs, single nucleotide polymorphisms. You can see there are a lot of synonymous and non-synonymous SNPs across the genome of this human case. And then, and again, that SNP was done by Chang. And then Kainat Sheikh, one of the first year PhD students in my group, she has a lot of interest in structural biology. So she actually mapped all these variations in the MERS-CoV molecule, particularly the spike protein. And actually, I forgot to tell the one that I showed you with red here. There are actually two non-synonymous substitutions in the RBD, receptor binding domain, and very quite satisfyingly, what we also saw that these are we then mapped with camel. We downloaded all the isolates that ever been sequenced and deposited to full length, and then we divided them into camel-derived isolates and human-derived isolates, and actually placed them in the clade P. And the very reassuring thing was there has been really seminal study published by Assam Hazar and colleagues that they actually sampled large number, like 40 plus samples from camel and some other coronaviruses, and they actually found a lot of mutations that we find the human case also, which is not unexpected. But if you see, I've particularly highlighted that this is also this, say, that this is very important to do some active surveillance of this virus. So what's happening with dengue? Now, dengue do not need introduction, but as you know, we are going through a major outbreak here. And just to tell you give you some numbers, we took some over 11 million dengue cases and 7,000 dengue million. This is from the European CDC data. So it's very important, and this is a major importance also when we are about to, Saudi Arabia is about to have the Hajj and also whole yearly Umrah and other religious activities. So this is very important. And of course, dengue comes in various flavors. This is just a schematic representation of its genome, and it comes in 4 serotypes, main serotypes, and then within serotype, various genotypes. So also, this is just a global at the moment for between 1 year period. Really recently, you see that Saudi Arabia has large number of cases here. Of course, it's happening everywhere now with global warming. The mosquitoes are also spreading. So this is the real data from Ministry of Health. You can see that it's from January to December, and if you look at 2023, it's a large number of cases happened over here. Of course, there needs to be a major genomics-driven surveillance program. So we have initiated with collaborators within the MMOH. But this is, again, another case. This is the global data sets. And you see our area is grossly. There are 7 genomes as of that day. It was taken around October, November last year, available from Saudi. Not a single from Oman as well. I'll tell you why it will become relevant. So you can see that we are, again, there is a crying need to do some genomics-driven surveillance of dengue in the Kingdom of Saudi Arabia and in the region wherever it is endemic. And we actually have been collaborating with Ministry of Health laboratories. We have collected large number of samples, and you can see where they come from within the kingdom. And then, of course, we have been using our routine qPCR and the whole genome approach. The idea is to basically do it quick, do it accurate, do it cheaper if possible. And we have achieved all, and also analyze them, come up with tools, automated tools to analyze them. So we actually have come up with a complete set of protocols from serum sample all the way to the genotype and serotype, we can do it within a day of 96 samples. It took a lot of time to automate the process, to come up with some tools, and this is exactly what we managed to do. Of course, the credit goes to the team, Sabir, Lamia, and Rohit on the bioinformatics side of things. So now what we have got, this is the first view of large number of, you can see we have all, I think by end of this month, we'll be reaching like 1,600 genomes within Saudi Arabia. We have also, there was a major outbreak in Oman. So we have been collaborating with SQU, Sultan Qaboos University. So we've got all of their samples too. So we've analyzed all of them. And you can see, while the dengue 1 is the main serotype 1 in Saudi, there are some dengue 3 and dengue 2, but in Oman, the situation is quite different. Dengue 2 is the main circulating strain there. Again, I particularly thank Dr. Walid and also all his team and other members of the Ministry of Health, and of course Mr. Mewa also, and also Sultan Qaboos University, our main collaborators for this. Again, this work is continuing, and we want to sort of have a global picture of what's happening in this within the next few months or so. So now last part, which is last but not least, metagenomics driven pathogen discovery. Okay, what we are trying to do, maybe this follows very nicely from some of the previous presentations. Dr. Majid Al-Gharibi and also Hamza Taib gave a very good presentations on this. So our idea is to do rapid pathogen discovery or produce pathogen alerts in data in real time or near real time, or as fast as possible. Again, this work has been happening in the group for some time now, and we have established a really good protocol that involves both Oxford Nanopore and Illumina. Of course, our collaborators have been extremely helpful and supportive from King Fahd Medical City, and also Dr. Reem Al-Maghrabi, Maya Al-Maghrab from King. We have been working with them quite long, and we have many projects, ongoing projects with them. We have been already published work related to that on with King Fahd Medical City on finding multivector leprosy in skin biopsy samples, Naegleria fowleri. Now these are published work. Then unpublished work remains. We are working on them, sort of publishing them on some zoonotic brucellosis, also some brain-infecting fungus, and also I have a major project with Dr. Reem Al-Maghrabi and her team on solid organ transplant. So these solid organ transplant projects where we are actually using metagenomics to call the organs pathogen-free. And that work has, and we are particularly working on lung transplant and liver transplant patients. Donor as well as. The final part of my talk is, as I mentioned, we are looking at dengue, we are looking at other chikungunya, looking at other things, but important thing is can we actually, the mosquitoes are the culprit in this case, can we actually identify, because they are basically flying syringes, right? So can you identify them using some protocol? And I'm very happy to show you some, again, this is not published, literally data from last month or last week, if you like. So we have been working on this just as on a pilot project in North Jeddah, South Jeddah, and just few from cows. And I show you the results now. So basically, again, Chang Li is doing the Bioinformatics part. So she has a full protocol in there. So you see, we have collected anything that looks like mosquito. So we have actually these mosquitoes in our pools. That came up quite clear. And then now is the real data. You see that on those, we actually see in one of the, if you follow the red arrow, it actually finds dengue. So we actually now in a pooled mosquito pool, we can see dengue even before it caused disease. It's in the mosquito. And also another thing, if you follow the blue arrow, it actually finds Wolbachia and Racemia Aedes. As you know, CAXT has a big project on releasing Wolbachia for dengue control. This way, actually, we can figure out whether the mosquitoes here are carrying Wolbachia. If they are, if we sort of make it a big, like a nationwide effort in this. So this is just the first. And again, I thank Chang Chang is supposed to be in the audience, but I don't see her at the moment. And then finally, oops, again, stop. Okay, so actually from these methods, we can do the whole genome of dengue just from this sequence-independent method. You can see that it's low coverage, but we find this. But also, we have used the amplicon-based method that we have been doing the main genome projects. Again, full genome of this. So basically, we can even now identify not only presence, but also the whole genome and the genotype and serotype from even a pool of mosquitoes. So now we can reliably detect fully assembled dengue genomes from the disease vectors, which are so-called the flying syringes. Now we said dengue, but the methods are used. If they're carrying malaria, if they're causing chikungunya, if they are carrying Zika, we should be able to detect. And also the methods are actually irrespective. So it is wastewater, if it is something else, farm water, any sample we can now detect quite reliably using the techniques that we have over the months, over the years optimized in the group. So this is my summary slide. So basically, I have shown you. This is my last slide. So I have shown you with NGS-driven analytical technologies and track how you can track. Basically, you start with genome sequencing, identify some specific areas of interest that may be associated with severe disease, which happened with the COVID case. Then we took them all the way to human transcriptome. So I did not talk about we have a major proteomic study also actually available in bioarchive from the same sets of samples. And of course, then you can take it into more like, reverse engineering the virus, look into more like biological, cellular kind of mechanistic effects of how you are seeing, you identify the mechanistic sites of why you are seeing a phenotype. You can also, we are doing COVID and MENA in the MENA, but now we can also do any pathogen surveillance, which of course with many collaborators, many of you sitting here in the audience, we are working with. But most importantly, the pandemic preparedness, that's the main theme of this, at least sub-theme of this meeting. Yes, we have optimized various types of SAM over the years, and we can work on any organism now that are thrown back at us. Using recent clinical and mosquito samples, of course, on clinical metagenomics, which Dr. Hamza particularly pointed out, this is something we do quite routinely, and I think it's a great way to help the clinicians sometimes in decision making. It doesn't solve something problem, but when, as she very clearly said, the cases which are unidentifiable, there is no etiology, doctors get some benefit. We also are very happy when we're able to show, for example, the MDR leprosy case I told you about in KFMC some years ago. I was working with Dr. Faisal Al-Asmari there, and he was so happy when we came up with all the diagnostics, and also we told from the genomic data where exactly it came from and what to the level, what are the various antibiotics that it is MDR-like resistant to, based on the SNPs. And the last point, which is basically we are prepared to roll out the NGS-guided genomic tracking of major viral and other pathogens, including coronaviruses, dengue, or other novel or re-emerging viruses in KSA and region. Of course, we'd be happy to collaborate with some of you if you have, we don't have our in-house, we have the technology, we have the know-how, but we don't have a hospital. And you have all this, so together we can actually be a very winning combination here. And the final one is really to thank all the team, all the collaborators, all the organizations who have patiently sometimes, waited for us to deliver some science, and we'd like to extend this. And of course, the funders, they really need a mention here. And thanks for your attention, and I'd be very happy to take questions during my question time. Yes.
Thank you very much. Our next speaker is Dr. Naif Harbi. Dr. Naif Harbi is a distinguished expert in microbiology, vaccinology, and epidemic preparedness. Dr. Harbi currently serves as the Director of Public Health Emergency at the Gulf CDC, where he has recently led the establishment of epidemic intelligence and pandemic preparedness efforts across the GCC region. Previously, he played an important role in establishing the vaccine development unit at KMR. Among his notable achievement, he co-developed the Children's MERS vaccine candidate and has made a significant contribution to a national initiative in translational research, biotechnology, and in the biosecurity section. Dr. Harbi actually earned his PhD in molecular biology and vaccinology from University of Oxford. Please join us in welcoming Dr. Naif Harbi.
السلام عليكم ورحمة الله وبركاته. صباح الخير جميعا أو مساء الخير. I would like to thank the organizer, Dr. Anwar Hashim, Dr. Assam Azhar, and Dr. Turki, and the rest of their team for inviting us as speakers today. We'd like to be part of this and enjoy being here. My presentation is a bit difficult now after the Prof. Arnabil, but I think you did what I call like a core science that will take you back to the public health and the picture of pandemic and pandemic preparedness. And the always lesson when it comes to pandemic preparedness is to look back and to learn from the history of public health emergencies and the history of public health intervention. You see here in the picture the smallpox vaccination campaign in Riyadh in the 60s or 70s. A disclaimer here that I'm not presenting on behalf of the Gulf CDC, but most of the material are presenting from Gulf CDC and they are available online. So a public health emergency is focusing on a number of hazards: CBRNE, chemical, biological, radionuclear, environmental. Most countries are putting their national plans to what we call all hazard or cross-sectoral or multi-sectoral sometimes. And the Gulf CDC has published its regional plan or a multi-country preparedness plan for public health emergencies with much focus on biological hazards. Here is the picture of the emerging epidemics and pandemics in the last 20 years. We've seen a number of them, and we lived through a number of them. And now we're calling disease X or pathogen X for whatever potential or coming up pandemic or epidemic. And one of the preparedness plan activities or initiatives, and that's also included in the Gulf CDC or GCC preparedness plan, is the epidemic intelligence. Epidemic intelligence is where you search for information in an early phase of the epidemic using multiple sources, whether AI-based or manual, searching for signals, what we call signals. And then you alert, you send your early warning to the public health professionals and to healthcare workers and practitioners, including what we call social listening and focusing. And this has been in practice in the Gulf CDC during mass gathering like the FIFA in Qatar or the Hajj, annual Hajj. And the way it works, you gather your online sources. In the annual report published by Gulf CDC, we did more than 1,000 signals a year in 2024. You filter them through expert opinion and through criterion system to signals, and then you discuss with the public health expert if they are qualified for threats or not, and then you have another criteria if they qualify for events or not. So if it's event by Gulf CDC, it means it has a relevant to the 6 countries, and we call it the event of public health interest to the GCC. Online, you will find annual report, monthly reports of the epidemic intelligence and a number of risk assessments, but the weekly report are sent to the focal point directly. You can appreciate here from 2023 and 2024, most of the signals we detected are infectious and human. And the event of regional interest in last year were diphtheria, measles, yellow fever, avian influenza, mpox, and Marburg. Now, if we look at this, these are the location of the first signal coming from which country, but it doesn't reflect the epidemiological status or situation of these diseases. To give you a snapshot of each of or most of these diseases, the human and highly pathogenic avian influenza, H5N1, it was reported in 6 countries: United States, Cambodia, Australia, Canada, China, and Vietnam. In the States, more than 10 states, I think by now, are reporting human cases. The new thing about this outbreak is it has affected or infected cattle, dairy cattle. There are more than 700 dairy herds that have been infected in the United States, and the annual number of their cases are more than, it is the second largest after the Egyptian outbreak in 2015. There is no evidence of human-to-human transmission. However, if you look at the United States map, if you look at the wild bird and poultry, every state has reported wild bird and poultry, and European countries millions and millions of poultry farms have been killing birds. And there was an issue of the dairy milk tested positive for the avian influenza, and for the lab or the researcher, we need to be able to detect avian influenza from milk products. But also, this is a regulation for the livestock authority, the veterinary. So if you think about this issue, it's affecting environmental sector, agriculture sector, health sector, public health, and scientific community. Mpox, since 2022, the Mpox have been a public health emergency of international concern, FIC. However, last year it has re-emerged again as a clade 1B. The clade 1B was first detected in DRC in Africa, and it appears to be affecting children as well as adult and sex workers. The table here and the map are showing the African country reporting Mpox in general, but the bottom two small tables there cut out there are the countries that have received Mpox clade 1B by importation from African countries. I think this is not updated because this year there has been some countries like Oman who reported to the WHO officially that there is clade 1B, and the United Kingdom have shown local transmission of or local acquired clade 1B Mpox. Marburg, on the other hand, last year we saw Marburg for the first time in Rwanda, with 80% in healthcare worker, with a high mortality rate, 23% of Marburg in Rwanda last year. However, this mortality rate is below the average for this virus. Since 2017, there has been 6 or 7 outbreaks: Rwanda, Equatorial Guinea, Tanzania, Ghana, Guinea, and Uganda. The reason or some of the factors for Marburg virus re-emergence are hypothesized to be due to more human contact with animals and change in climate. Diphtheria became a big issue last year in Nigeria and the year before. Nigeria reported more than 16,000 cases with high number of deaths. This increase also into other African countries, but Nigeria was the main epicenter, if you like. And this is a message for public health professionals, and we were discussing this. Are you gonna, for example, trying to relate this information to reality? Are you gonna test people coming from Nigeria for diphtheria? Are you gonna mandate certification of vaccine for people coming from Nigeria? And so on and so forth. Measles are increasing in almost every country in the globe since 2 or 3 years ago. And we are no different in the region. The factors for that are proposed to be missed measles vaccine or delay in the vaccination. The better now is it's a historical average cases of measles globally. You can think of the first 4 months of last year. There was more than 50,000 cases globally. GCC countries also reported more than 1,700 cases. However, the rate of infection of measles in 2014 was lower than the rate in 2023 in the GCC countries, and that's mainly due to the drop of cases in Saudi Arabia. Yemen is suffering from a number of epidemics: malaria, diphtheria, cholera, dengue, while vaccine-derived polio and many others, but measles is among one of them. Pakistan and Iraq also has shown an increase of the cases in measles. Now, cholera as well is increasing mainly in the EMRO region and in the Afro region. And in the neighboring, what we call in the Gulf CDC, we have the neighboring countries, including Yemen, Sudan, Egypt, and just geographically linked to the GCC. And cholera was reported in Eastern Africa as well as in Yemen. Poliovirus, so poliovirus is wild. Poliovirus is coming back again, especially in Afghanistan and Pakistan. Vaccine-derived poliovirus is reported in many countries. In European countries, they detected it through wastewater surveillance, the poliovirus. but with no cases in humans. I'm going to skip dengue, although it's very interesting that the local acquired cases are happening in Italy, France, and United States. I think Dr. Anna mentioned dengue, but dengue is emerging. Now you see local acquired cases of dengue in Europe, which tells a lot about the climate change and the expansion of the vectors. Here, what I call exotic or rare virus, my colleagues, they're calling it emerging pathogens. There are a number. If you do epidemic intelligence, you come across some viruses that you never know before. So, Orborche virus, I'm going to come back to that. Uzi virus in Japan was isolated from a tick, and there was a lethal case in human. Ikarat virus also is endemic in Peru with some cases, and then Chandipura virus. So Chandipura virus is in central India, in Gujarat state. It has a high mortality rate, 50 to 60% of Chandipura virus in India. However, the risk of getting the virus into the GCC was determined to be low or very low. But again, it's a vector-borne virus, and it's quite serious. Also, Usutu virus that was identified last century in South Africa. There has been some cases in France in the last 2 or 3 years. Jamestown Canyon virus, again, that was reported in Michigan, and the herpes virus, CME, which is a monkey virus, get through a very close contact with monkeys. And going back to the Orborche virus, it started in Peru, mainly Peru, but recently it has been increased in Brazil, Cuba, and Bolivia. More cases also detected in the United States and in Europe. And it needs a special attention and special reading, basically. And the reason why I'm trying to put a highlight on these epidemics and viruses is because I was doing science, full-time science, and moved to public health, and I know our colleagues in the clinic. And what I learned working in public health is you need to be aware. And one of the recommendations that we always put in risk assessment is to increase awareness. I thought in the beginning it's nonsense. Why you increase the awareness? Like everybody's aware of it. But if you are doing the clinical work, you may not be aware of Chandipura virus happening or the increase in measles in many countries. And especially people doing triage, people attending to patients, people doing the travel clinic. For the scientists also is very, very important because you see where the need is happening. Because whatever you spend doing prioritization for your research, which disease you want to focus on, sometimes you have to be aware of the reality happening around you. With that, I would like to thank colleagues back in the Gulf CDC who are a very ambitious team, very new center, established 2 years ago, and they're doing an amazing job. Thank you so much for listening.
Thank you tonight for really shedding the light on this important organization, the Gulf CDC, and the work they're doing. Toward your last point, so the next speaker is actually a physician, and he's gonna show us the clinical insight into the infectious disease and pandemic preparedness. So our next speaker is Dr. Mohammed Boussaid. He is infectious disease consultant at the Department of Medicine at King Abdulaziz Medical City in Riyadh. He is associate professor at College of Medicine also in King Abdulaziz University for Health Sciences. He is also the director of clinical efficiency at the Clinical Strategy Department at the Ministry of National Guard and Health Affairs. He is also the chairman of Department of Infectious Disease Research at KFMRC. Please join me to welcoming Dr. Mohammed Boussaid, and he's gonna talk to us about their experience for doing the first clinical trial for a vaccine for MERS-CoV.
Thank you, Dr. Abdullah. I will not be talking about Chandipura virus or Aussie virus. So we'll talk about something we know. I would like first to thank Prof. Anwar, Sam, Dr. Turki for the invitation. And it is a pleasure to be back more than 15 years ago, to be back to my university, visiting and seeing familiar faces. Okay, so we will talk about clinical trials, basically. I think I'm having the same problem that Dr. Arnold. Yeah. Which one? Okay, so basically I will take you through the story of the phase 1 clinical trial that we conducted in the last few years, and then we will go through some challenges that usually faces clinical trials, early phases clinical trials, and what is the future direction of this. So the clinical trial was about MERS-CoV, so it's something that we know, and we had multiple outbreaks in the last 10 years, where we had hospital closed, we had a lot of patients, high mortality, and it was a health care concern at the time. So basically, MERS-CoV is still circulating. I'm gonna, maybe when the slides are here, we'll try to show it. Currently, as we talk now, we have 2 cases here who are in the critical care unit and due to respiratory infection secondary to MERS-CoV. Are we going to have the slide? So I'll maybe talk a little bit about the virus itself. So it's a respiratory virus. It's called Middle East respiratory virus. Most of the cases in Saudi Arabia, as I said, case fatality rate were very high, and yeah, here's good. Can you do the screen, full screen mode? So as of this year, also we do have new cases circulating. So it is expected at any point in the future that we might have a problem with this virus. And we need to have preparedness, we need to be aware of this and ready. Okay. I think we're good. Wait. Okay, so I think we went through this because of the time, so let's just go through the clinical trial. So MERS-002 is a collaboration between King Abdullah International Medical Research Center and the University of Oxford. It took a few years to conduct such a trial, from the start of the discussion till the final conclusion of the trial. It is actually started. Okay, so the discussion started in 2015 with ambitious scientists working on the Chadwick's MERS vaccine, who's sitting with us here, and he is aiming to take this lab work all the way to clinical trial. And after that, it was actually followed by a couple of years of further discussion, requesting for fund, obtaining the fund, and then the real discussion was started with the scientific merits of this vaccine and how we should do this trial. So MERS-001 was conducted on the University of Oxford in Engineering Institute, followed by MERS-002. So in 2017, a discussion with Saudi FDA started, and then 2018, a lot of actually work has been done, a lot of activities, including writing the proposal, going through the clinical trial agreement, discussing with Saudi FDA with multiple meetings, and also meeting with the ethical committees. And at the end of the 2018, we submitted the full proposal to the Saudi FDA. Of course, to have phase 1 clinical trial, you need to have a unit that is very well equipped to do a phase 1 clinical trial. So we did have multiple visits and inspections from the Saudi FDA to the unit. And eventually, by October 2019, we obtained the approval for the unit to be able to conduct a phase 1 clinical trial and also the approval of the study itself to be conducted. So I will not take you through all of that, but it is a lot of work. It's a lot of documents. It's a very big dossier that you are going to send to Saudi FDA. All of that has been done from scratch. Specifically, that this is an investigator-initiated trial. This is not sponsored. It's not a drug company who took this and did all of the logistic work. So all of that has been done over 2 years, and eventually we adjusted the trial before we started it as the policy in the clinical trial that goes. So in terms of the infrastructure, in the hospital, we created a board that has all the stakeholders who can support a phase 1 clinical trial, and that includes medical services, operation, nursing, and all of that. And then also the unit was very well equipped unit. So we have a 10-bed ward in the hospital, in King Abdullah Specialist Hospital within the National Guard. And it is fully equipped with nursing, pharmacists. Also, it has a lab and a pharmacy within the unit. So it's an independent unit that has been only utilized for clinical trial. And we made sure that this is patient-centered because the phase 1 clinical trial usually will have healthy volunteers. So this unit should be very friendly, having a lot of areas where you can make sure that the volunteers are having a good journey and experience during the clinical trial. Of course, also we needed an independent data safety and monitoring board. So we did have a senior expert from the States, from UK, also from Saudi, who served as an independent committee observing the safety of the clinical trial. And then we started our advertisement. Of course, again, the advertisement is very well done and very well actually observed by the Saudi FDA and the ethical committee. We went to malls, we went to the universities, we did it also through social media as well. And the aim here is to recruit eligible patients who are going to be screened and eventually be part of the trial. And then also, again, doing a phase 1 clinical trial, which is one of a kind in the country, it's not an easy thing. So we made sure that our team is very well trained. We did a startup meeting, we did mock trials, our nurses has been trained for the trial, and then also they have been trained for all emergency scenarios that might happen. Now it's stop moving. Okay. Okay, so we are here. So this one in the green box is the vaccine. It arrived in December 2019. And also we had a team from Jenner Institute joining our team for the first patient in. So late December, we were able to report our first patient and we had the integration for the unit. And that was after screening more than 38 healthy volunteers, going through the study itself, doing physical exam, history, and lab workup, and making sure that these patients are actually eligible for the trial. Okay, so this is the journey for the patient. They usually go through a vaccine. And then they have multiple visits, up to 6 months. Some of these visits are clinical only, just a clinical checkup, and some are for immunological testing as well. So again, safety and immunogenicity for the trial. And then okay, so it was a dose escalation study. I don't want to take you through the science much here, but it is 3 doses, 24 participants, low, intermediate, and high dosing. And the first 3 patients in every group, we have admitted them to the hospital for 48 hours to make sure that they are eligible. So basically, when you start with the low dose. If the patient is the healthy volunteer is doing fine, you can escalate to the higher dose after the data safety and monitoring committee approved. I'm sorry for the slides, but so this is the result of the trial. So basically, unfortunately, this was continued during the pandemic, and we were able, alhamdulillah, to continue the trial during the curfew, and we managed to recruit all the patients. So we have young population, 21 to 46 years old as an average, and then the 24 that were recruited were between males and females. And as I mentioned, so 6 for the low dose, 9 for the intermediate dose, and 9 in the high dose. Regarding safety, there were no major concern. A lot of mild reactions as expected with any vaccine. But regarding severe, it was only in 2 volunteers in the intermediate group, and it was basically headache that they had for 1 day. So there were no major safety concerns in the trial. And regarding immunogenicity, actually the signs were really good, having a very good response in terms of antibodies all the way till 6 months of the vaccination time. And then also the cellular immunity showed good response, and even if we compared our population with Oxford population, our population did very well and maintaining immune response in cellular level up to 6 months. Okay, so here we wanted to talk about the challenges. So basically, in early phases clinical trial, and it's something that we are still lacking in the country. And in these trials, it's really challenging to conduct because of many things. So these are the main things that I think that are preventing many of the clinical trials worldwide to be conducted, especially for early phases. Funding is one thing, especially that it does cost a lot. You need a lot of infrastructure that will also cost a lot. You don't usually have in phase 1 clinical trial and like direct outcome or direct product that you are going to have. So when you go to phase 1 clinical trial, you need to have a strong case to get people funding you. Second is the culture and belief system, and I think that the people had sometimes bad experience with vaccination, especially with what happened with the COVID-19 vaccination. So we need to make sure that the culture accepts that. We need to make sure that we have transparency with people. We need to address the system and the public with clearance, with direct and clear messages instead of just trying to record without building this rapport with them. And then recruiting, it's sometimes really very difficult to recruit volunteers because of multiple things, including also ethical issues. So for us, as an example, we're prevented to recruit students from the university. They might be a vulnerable population. You cannot recruit soldiers. We are National Guard, so we couldn't recruit soldiers. That would be an easy access for people to recruit, but we were not able to do that. So also, it's not as easy as it looks. Infrastructure, as you saw, that we had a unit in the hospital that is very well equipped. So that's not available for everyone. And I think it's really important to have places that are unique and very well equipped for phase 1 clinical trial. And then conduction and monitoring, again, you need to be able to train your team and also the investigators to do a phase 1 clinical trial. And this also required a lot. We needed to send our people even to Oxford University to do some training. What's unique about the vaccine, I will just go through it quickly. So vaccine clinical trials are not therapeutic clinical trials. So you need to make sure that you have a public trust. We need to make sure that every platform being understood very well because it's different from platform to platform. The population diversity in terms of genetics, in terms of ethnicity, in terms of ages, have a high safety expectation. If you are doing clinical trial in healthy people, you should be able to provide high safety instead of having, I know it's not having. So healthy people will not come if they know that they will have it first. So again, immune response and the risk of immune enhancement. I think you know the story of dengue and RSV before the vaccination. The future direction, we expect that we are going to have personalized vaccination strategy where we're able to have awareness of every patient response, every volunteer response, instead of universal vaccination. We should have one platform that can cover a lot, like what we have with influenza vaccine, where we are going to have coronavirus vaccine for all coronaviruses. Biosecurity, we should be prepared. As I mentioned, MERS, we should have a MERS vaccine ready for any potential outbreak. And then eventually, I'm expecting that AI is going to take over in terms of recruiting patients, in terms of analyzing, and sometimes real-time analysis of the immune response and the safety profile of the vaccine. Thank you.
Thank you, Dr. Hussein. Our next speaker is Dr. Tamer Andijani. Dr. Andijani is an associate professor of virology and consultant in laboratory medicine. He currently leads the scientific research unit at the Faculty of Applied Medical Sciences, King Abdulaziz University, and also affiliated with Special Infectious Agent unit in the King Fahd Medical Research Center, with over 45 publications in peer-reviewed journals. Dr. research focused on respiratory and re-emerging viruses. His work contributes to the field of virology, immunology, epidemiology, and development of therapeutic antiviral agents. Please join me in welcoming Dr. Tamer Andijani.
Right. What a heavy scientific meal from our great scientists, Prof. Arnav, Dr. Harbi, Dr. Hussein. Speaking of meals, eggs is among my favorite food. Scrambled, boiled, sunny side up, shakshuka, egg and cheese, some people call it omelet to make you pay 10 times more, right? And my favorite is egg Benedict. But surely I'm not here today to tell you that, to talk about my favorite food or tell you that eggs actually is a great source of nutrition. What I'm going to talk about today, hopefully to show you that eggs can be a great source of producing therapeutic and diagnostic antibodies. The whole process starts by immunizing the chicken, collecting the eggs, and from the egg yolk, we purify our IgY. It's distinct, it's different than mammalian immunoglobulin. In terms of structure, it's close to IgG, but it has 4 constant domain. It also lacks the hinge region. That makes it more rigid, more stable. It's actually more resistant to pH, to temperature, to proteolysis. And the lack of hinge region makes it less flexible, but that also means less cross-reactivity with mammalian proteins. That actually, it also doesn't interact with Fc receptors, and it does not activate complement. Collectively, this makes IgY an attractive target for oral and mucosal immunotherapy development. Our success stories started with these 2 coronaviruses. If you're a virologist, you already know this. If you're not, maybe you should know this now. The main viral protein that we'd like to target when we develop immunotherapy or we develop therapeutic antibodies is the viral proteins that's actually important for viral attachment and viral fusion and entry into the cell. So we can actually block the viral replication from the initial steps. In case of coronavirus, here we're talking about spike protein, which is actually composed of two subunits. S1 that has a receptor binding domain that's important for attachment. Also the S2, which is important for viral fusion. Our work with MERS-CoV, we have used the full length S to immunize the chicken with, and there are other work that we have used only S1, right? Both works show similar results, so I'm going to focus on only one of them. The chicken were immunized, the eggs were collected, and from the egg yolk, we have purified the antibodies using the PEG standard protocol. This is our purified products, shown by SDS phase and Western blotting. We also monitor the kinetic of IgY production over weeks following the immunization. Next, we wanted to determine whether IgY can specifically detect our viral antigen. So we've done Western blotting and also a dot blotting. With both of them, we've used our produced IgY as a primary antibody. And in both cases, we can show that our produced antibody can specifically detect the viral antigen of interest. More importantly, we have done an in vitro serum neutralization assay to assess the capacity of the produced IgY to inhibit viral replication, to combat the infection, right? And we got positive results with the in vitro assay that actually allowed us or motivated us to do some in vivo work. We've done the in vivo work in the transgenic mice that expressing the human receptors. We can see that the presence of the IgY that we have produced it actually preserves the normal histological features of the. Can you hear me? Okay, the normal histological features of the lung of the infected mice. That also coincides with a reduction in the number of infected cells in the presence of therapeutic IgY. Again, we got similar results when we used only the full-length S protein or we used S1 only, and both work were published and it was also patented. With regard to SARS-CoV-2, we've also used a different set of antigen, a list of antigen, but I'm going to focus on the receptor binding domain, which is again part of the S1 subunit, that's part of the full-length S, that's important for viral attachment and injury. Again, our purified product is this stage, and we monitor the production of IgY weeks following the immunization. Also, the produced IgY can specifically detect the RBD antigen, as shown here by Western blotting. Most importantly, when we've done the in vitro neutralization assay, we can show that our IgY, our RBD IgY could neutralize the wild type local isolate to lesser efficiency, but it still can also inhibit the replication of the Delta variants, while the Omicron variant escaped from this neutralization. We proceeded with in vivo work. However, the wild type SARS-CoV-2 is not infectious, or let's say the wild type mice are not permissive to wild type virus. So to overcome this obstacle, we have used two animal models. In the first animal model, we've used the wild type virus, but we have transfused the mice with the human receptors. In the second animal model, we've used the wild type mice, but we've used a mouse adapted strain, right? So hopefully in the first case, we can mimic the normal entry into human, normal viral entry to the human. Well, in the second case, we have a wild type mice, so that probably will produce more natural and more native host immunity. In the first animal model, as days after infection, all mice actually started to lose weight as a response to the infection, but those were treated with RBD IgY quickly and rapidly recovered. The number of the viral plaque forming efficiency was also reduced in the presence of RBD IgY. Also, the histopathological features in terms of pneumonitis, immune cell infiltration, edema, and hemorrhages, all of them were reduced in the presence of RBD IgY. In the second animal model, the presence of RBD IgY nearly 100% led to nearly 100% survival of the infected animals. That's also correlated with a significant and substantial reduction of the viral titer in the lung of the infected mice. And that's true whether these mice were male or female. This work was also published and patented. Moving to a more recent work that's currently actually conducted in our lab, we're focused on RSV. It's a respiratory syncytial virus. It's one of the leading causes of acute lower respiratory infections in infants. 1 in every 30 deaths among infants globally is linked to RSV. More than 3 million hospitalizations every year. Among the elderly, the burden of RSV is very comparable to seasonal influenza with over 14,000 cases death annually. In late 2023, there was 2 vaccines have been approved for use to those age 60 and above. And both vaccines were based on the viral fusion protein. The viral fusion protein exists into 2 conformational status, pre-fusion and post-fusion. The pre-fusion one exhibits potent neutralizing epitopes. The one I'm referring to is the null and the 5, while actually it's hidden in the post-fusion states. That's why most of the vaccines actually rely on this fusion protein in a pre-fusion state. Actually, also the currently used monoclonal antibody therapy is based on this vaccine. The problem is how to get this pre-fusion vaccine stable in its pre-fusion state. Luckily, we could get it, and we had planned our experiment and immunized our chicken with different doses of pre-fusion antigen. Again, the eggs were collected, and we purified IgY against pre-fusion protein from the egg yolk. In the top right corner, we can see that anti-RSV pre-fusion protein by Western blotting and SDS phage. We've also monitored the production, the kinetic of production over weeks by ELISA. In this ELISA, we use different type of fusion antigen in different states from different subtypes. Because the fusion is highly observed among RSV subtypes, the produced IgY could interact with both of them, and that was also confirmed by Western blotting. In the last 2 weeks, we had the first set of data with the in vitro neutralization assay, and our preliminary data indicates that the produced IgY could neutralize RSV subtype A, A2, and B as well in vitro, which covered the clinical isolate and the laboratory fixed strain. What I would like to say is that we have a well-established platform, well-established protocol for producing IgY. Our work is expanding, covering too many viral agents. In fact, we are starting to do some work actually with other infectious agents, including some bacteria and some fungal as well. Thanks goes to the incredible team in the Special Infectious Agent Unit. Special thanks goes to the key player of this project, our respected Rob and our hardworking, enthusiastic national and international students, also to our collaborators. I'd like to leave it here by saying as a child, I have grown up reading lots of stories, folk tales, fairy tales, and actually some fables, right? About Cinderella, about the tortoise and the hare, or the rabbit and the turtle were in a race. Also about this goose, this chicken, who can lay golden eggs. I think with a little help, they can do it. Thank you all for listening.
Thank you so much, Dr. Samer, for this really insightful effort to use very efficient therapeutics for emerging diseases. So we are continuing with this very informative session. With the final talk for today is from different perspectives, from the pharmaceuticals. We have the final speaker for today is Dr. Khalid Suleiman from JSK. He is a clinical pharmacist and also public health specialist, and he's going to talk to us about Listeria meningitis serogroup B vaccine global experience. Please join me to welcoming Mr. Khalid.
Assalamu alaikum wa rahmatullah. Thank you, Dr. Abdullah Muhammad and Dr. Anwar Razan for this invitation. I'm Khalid Sulaiman. I am the medical manager of this area portfolio in GSK. My disclosure that I am full-time GSK employee, and this is a medical session, no promotion in it. Allow me to walk through these topics. Today we'll talk about the meningococcal disease, some characteristics, and then walk through global and regional and Saudi epidemiological data, and closing by the vaccines available for this severe disease. As you know that the invasive meningococcal disease is one of the most serious diseases worldwide and in Saudi Arabia. Why? Because the early signs and symptoms of this disease is easily misdiagnosed. The signs and symptoms may resemble that of common viral infection, but unfortunately within only 24 hours or 48 hours, the case may go for death. With acute serious illness of IMD, the septicemia or meningococemia may lead to high case fatality rate reaching up to 15% with the proper treatment in place. Without the proper treatment in place, the case fatality rate reaching up to 50%. Not only this, but the survivor after acute cases of meningitis are suffering from severe sequelae. These sequelae ranging from physical, psychological, neurological sequelae, as you are seeing here, maybe bilateral amputation or deafness, or this complication as a lifelong complication. When we are looking for the different serogroups of Neisseria meningitis causing the IMD, we are seeing 6 serogroups of Neisseria meningitis, the most common 6 serogroup causing about 90% of the cases of IMD worldwide. For our region and in Saudi Arabia, as we are seeing here, that the most common 3 serogroups causing IMD, serogroup W, about 40% of the cases, followed by serogroup A, 36% of the cases, and coming number 3, serogroup B, representing 17% of the cases. Another factor regarding the different serogroups of Neisseria meningitis, when we are seeing these different serogroups, we are seeing that there is a dynamicity and unpredictability of different serogroups of Neisseria meningitis. Here in US, for example, in 2010, serogroup Y in green color was the most dominant serogroup, representing the majority of cases. After only 6 years, the serogroup Y started to decrease and serogroup B started to increase. The dynamicity and the unpredictability of different serogroups push us for a different vaccination and immunization strategies. Another example from UK, here in 2006, serogroup B was the dominant serogroup. After 8 years, the serogroup B started to decrease, and the other serogroups started to increase, like serogroup Y and serogroup W. Different factors make the different vaccination strategies should be in place for Neisseria meningitis, as you are seeing here, the unpredictability, and at the same time, the transmission of the different serogroup from one region to another region as a global travel to endemic regions. For example, meningitis built in Africa, or here, huge mass gathering happening during Hajj and Umrah. And another factor, which is natural genetic shift from different serogroups. As you are seeing here, serogroups are always shifting from one serogroup to another. What about the epidemiological data from different countries worldwide? As you are seeing here, this is data from WHO in 2019, showing that serogroup B is the dominant serogroup in Europe, in North and South America, and in the meningitis African belt, the most dominant serogroup is serogroup A, then C, and serogroup W started to increase. In Europe, in 2018, the most dominant serogroup is serogroup B, and the age group with a high incidence of cases of IMPD was in infant below 1 year of age, followed by the toddlers until 5 years of age, and then adolescent in between 15 to 24 years, the second peak of incidence. Not only the infant and the adolescent, there is age group of adult also with a high risk of infection, like military individual, traveler to endemic region like for Hajj and Umrah, and healthcare workers who are dealing with the isolates of Neisseria meningitis. The vaccination strategies is successful when we are implementing a vaccine. For example, in Europe, since 2000, they implemented vaccine against serogroup C, and the reduction of cases of serogroup C started to appear in blue color. But for serogroup B, as you are seeing here, the most dominant serogroup, there was no vaccine worldwide till 2015. And the cases of serogroup B is the highest. After implementation of the vaccine, started the case to decrease in Europe starting from 2015 till now. What about our region also when we are talking about the epidemiological data and the risk during Hajj and Umrah, and Saudi Arabia has a lot of examples of outbreaks happened during Hajj and Umrah. Till 2024, there is some cases of meningitis appeared for the pilgrim when they are returning back to their home after performing Umrah. We are seeing here data from North African country. The country you are receiving a huge number of pilgrims coming to perform Umrah every year, and from Islamic country like Turkey and Kuwait and Iraq, we are seeing the contribution of serogroup B is the highest between the cases of IMPD. Also from Asian country, we are receiving a huge number of workers coming every year, returning back to their country. The contribution of serogroup B is the highest. Serogroup B representing more than 60% of the cases in Philippines and the majority of cases in Bangladesh. This is the clear situation of Saudi Arabia, the epidemiological data here in Saudi Arabia. We faced two outbreaks of Neisseria meningitis happened during Hajj. In 1987, the first outbreak happened during Hajj. It was serogroup A. And in 2001, the second outbreak happened by serogroup W. Two different serogroups caused two different outbreaks in Saudi Arabia. The data here in Saudi Arabia focused on the surveillance, and we needed to activate surveillance since this time to identify which serogroup is the dominant serogroup in Saudi Arabia. This one of the study have been conducted by Dr. Asiri, Dr. Memish, published in Vaccine 2017, talking about the carriage between pilgrims when they are coming to perform Hajj and after departure after performing Hajj as a nasopharyngeal carriage. They identified the carriage between the pilgrim that it is in the normal range, but the contribution of serogroup B is the highest, about 66% of the carriage between the pilgrim. In 2021, a narrative review and literature review has been published talking about the epidemiology of invasive meningococcal disease in the kingdom, collecting more than 15 articles regarding the IMD, plus the data from annual health statistic book from MOH. It is highlighting that the incidence of IMD is still high after implementing vaccination for the infant. We started implementing the vaccine in 2010 for the infant at 9 and 12 months. Although that we are facing some increase in the cases, for example, in 2017, we have reported 10 confirmed IMD cases. Not only this is the confirmed cases, but the reported unspecified cases of meningitis was huge in 2017. About 358 cases has been reported in 2017. So the outcome of this publication that the immunization strategy should be strengthened and considering to add MenB as one of the recommended vaccine. Another narrative review has been published in 2022 talking about the meningococcal disease immunization activities in Hajj in Saudi Arabia. As I mentioned, we are receiving about 75% of the pilgrim from 13 Islamic countries. This is the distribution of these Islamic countries. And this is the timeline of IMD outbreaks in Saudi Arabia since the immunization started. The first outbreak in 1987. After that, we recommended MERS vaccine as one of the required vaccine available at this time, with a chemoprophylaxis in 1988. After an outbreak happened during Umrah in 1992, the recommendation went for AC vaccination, one of the recommended vaccine available at this time in worldwide and in Saudi. Came serogroup W, making another outbreak during Hajj in 2000 and 2001. Outbreak happened and the recommendation went for polysaccharide vaccine in 2002 for all the residents in the Western region and for the individual performing Hajj in Saudi Arabia. In 2010, the recommendation went for MERS-CoV as a conjugated vaccine by names available now. You are knowing it for the infant starting at 9 months and 12 months of age, and all individual requiring to perform Hajj. What else? What we can expect that serogroup B need to be tackled by vaccination, as the vaccine has been registered in 2021, and the pentavalent vaccine either for ABCWY and at the same time ABCWX will be available in near future. GSK has invented this vaccine for controlling serogroup B after about 20 years of R&D, because the polysaccharide capsule of serogroup B was poorly immunogenic and it wasn't resemble that of other serogroups like ACWY. The polysaccharide capsule was poorly immunogenic, and there is some structure in the polysaccharide capsule resemble that present in human body, so there was a fear of autoimmune response. This vaccine, called the 4C-MB vaccine, 4-component meningococcal B vaccine to tackle only serogroup B. The first country implemented in their national immunization program was UK in 2015. After 3 years of implementation, they reduced the cases of IMD due to serogroup B for the infant and the children till 5 years of age up to 75%. Other countries worldwide implemented the vaccine for their national immunization program, like Italy, and at the same time Portugal implemented as endemic setting control, and Australia and Quebec in Canada, they implemented as outbreak control, and the result reduction of the cases by more than 70%, and in Canada more than 90%. Other countries worldwide went for the control for high-risk population. On top of them, the sickle cell patient with asplenia or splenectomy, or patient on biological treatment like eculizumab, raflizumab, and HIV patient. These high-risk population, they recommended the vaccine for them, and the vaccine started to be available. This is the vaccine. It's available in Saudi Arabia by this recommendation, starting from 2 months of age. It's called 4C-MB vaccine. It is recommended as 2 primary doses, 1 booster dose, and from 2 years of age and above, only 2 doses. Thank you. Any question more than?
Thank you to all the speakers. Please join us on the stage for the panel discussion.
So it's open for question now. We have a question over there. Arnold, please join us.
شكرا للجميع على المحاضرات الرائعة. عندي سؤال لدكتور دكتور بوجعيد على clinical case trial number 1, phase 1, sorry. So it's actually wonderful. I was very excited to see this work and how much it's been building infrastructure. I'm pretty sure it's not easy. I worked before with a group who has a center of vaccination in their center for a few things like a clinical as a clinician. And I saw their hard work, so I can imagine what you put into it. Now, my question is, yes, you have this phase 1 clinical trial. Moving forward, how can you look at the expertise, the funding, all of this for the center, given that you don't have that number of trials happening here? Are you able to recruit any industry or other people around the world to do that stuff? And the second question, sorry, is for samples that you gather from this trial that you have, do you have some sort of a protocol or a pathway to keep the samples in Saudi for future use? For example, with the Israeli, for example, to see if there's any cross activity from the vaccination with SARS-CoV-2 and other stuff. Thank you.
Okay, thank you for your question. I agree that it's not easy to build an infrastructure, and you need to make sure that the higher management are very well convinced to spend that much efforts and investment on building this one clinical trial. In terms of the skills and expertise, I totally agree. Actually, maintaining this is not easy, and you need to make sure that the unit keeps recruiting phase 1 clinical trial. So it's not easy because you need to partner with the startups, you need to have a lab that can take the vaccine from the bench all the way to phase 1 clinical trial. So it's not easy. We are doing a lot of efforts to maintain this, make sure that it is operational. It's not just like one-time story and that's it. So we are trying, and we do have at least 2 phase 1 clinical trials coming for non-infectious diseases that they are going to utilize in this year. And for infectious diseases, we have a lot of discussions. So potentially, we think that we are going to record some of these trials to be done here in Saudi. Regarding the second question is, so all the lab work has been conducted by Dr. Naif and his team in KMR. And as per the IRB, we should keep the samples for 10 years. So we do have the samples, of course.
I have a question. I need if one of you will answer. So we have the hypersocial expertise from sequencing to antibody development, vaccine development, clinical trial. How you guys can work all together to from pathogen detection to vaccine antibody development to clinical trial? I know many scientists has no one mentioned that many scientists have antibody in the lab, have developed several antibodies or developed several vaccines, and they never go to clinical trials. So how we can work all together to have our products from the bench to bedside, starting from Dr. Pink's.
Do you hear me? Yeah. So that's a very pertinent question, and thanks for asking. So in the interest of time, I'll be giving my part of the answer very short. So actually, we should learn from what, for example, some of the very successful organizations did during as part of the pandemic response. I can say UK as an example, where usually they used to work together, but also quite fragmented, but really went together, like the way the vaccines were developed, the way sequencing, as you know, even
globally speaking, UK led the way of sequencing, virus tracking. Oxford really led the way, on very quickly making the vaccine. So I think for us, we have plenty of core expertise. For example, I can see King Abdulaziz University. You guys already have been working hugely on its immunological aspects. We have also I've seen work that's been done in KFMC by Dr. Banda and colleagues. So I think we actually probably should do something like a consortium, for example, could be directed towards pandemic preparedness for coronaviruses, or could be just particular type of pathogens or escape pathogens, and then work towards our, no single organization will do it all. I can tell you from my experience. Let's put our strengths under one pot and work together.
Thank you, Dr. Ahmed, for this question. From GSK perspective, as a pharma company, we have initiated this collaboration with the MOH here and PHE. At the same time, we have more than 5 memorandum of understanding last year, and at the same time included some high specialized research and development from GSK and MOH. I expect that has started to begin last 2 years, and we are on the track to evaluate what we are reaching and making another strength point in our way to develop this memorandum of understanding to enhance the research and development in South area.
Thank you, Dr. Ahmed, for the question. Well, I think if you look at organization, we're in different organizations. There are so many organizations in Saudi Arabia. There are so many infrastructure. I worked in Mawhiba, evaluating students. You hear about different equipment, different research in so many universities. And so there is no lack of equipment. There is no lack of hardware. And if you look at the organization, what are organizations? They are made of people, right? If you go to KMR, you talk to Dr. Busaid, another person, these are the organizations. I think we have to show example to the next generation. We are not young anymore, right? There are another generation coming up with science, right? And, yeah, except somebody who's middle age. And I mean it. I think I expect some scientists to be very protective of their work, to publish their own paper, if it's the first paper, or the second paper, or the 10th paper. But after 10 paper, you should open up, you prove yourself, you put your name in there and comment, everybody knows you, you should now give away. If you look at our labs, there are so many samples that have existed for years. And there are so many protocols that have never been optimized or validated or put into action. I think that's my message, is to move away from just publishing for the sake of publishing. Trying to find a good idea it's a national idea, or it's a global idea where you need to earn up for something, you need to say for something, to found something, and work together. Nobody's going to do it alone. And I think we have a responsibility, honestly, to the community, to the history, to show example. So one day they will say, this person, he was very good. So I think we're all convinced, but we need to put it into action. That means the collaboration. Thank you.
Yeah, first of all, I can talk about myself and my research group. I think we're very open for collaboration. Actually, if somebody can find something interesting that we can do in our lab, or something that actually we can share our experience with, somebody can offer his experience as something to complete our work, that we would love to do all the time. But if you ask me, actually, we're all scientists. We all have, it's a competitive environment, right? So to be honest, if you ask me what is the best solution, it should come from from those higher authority, in which like we should be forced to work together. I think that's the solution for it.
Okay, I will not take much, but this is a very difficult question. It's not as easy as it looks. But I'll just take it from my side. I'm a clinician. I can do clinical trials. So if you have something ready for me, I'll start working on it tomorrow. We need a project that it's promising, GMP manufactured, and we can start working with that. So I think we have the pieces, as my colleague mentioned, but we need to put them together. We need to put one goal, one vision, and everyone should work toward it.
We'll queue by your door.
ها؟
We'll queue by your door.
الله يحييك. أنا بس خايف من الوقت بس أخاف من ذيك. والله لا شيء.
So I think, thank you everyone. I think what I'm going to say, I think one big consortium and actually this, and I think Dr. Anwar Hashim. So when Dr. Anwar Hashim maybe can lead this consortium, I think as Dr. Naif said, we really need to set an example. You need to share your protocols with others. Thank you very much. Thank you very much.
I have a comment. I think it's a very important question. Again, it goes back to what I said in the beginning. I think we all say that, but in action, we don't do it. That's the reality. So we need to be bold enough to do it. I think, of course, having organic collaboration is very important, and it's the actual collaboration. Otherwise, you cannot enforce people. You'll have an agreement at the higher level, but I think people need to understand that it's important to work together. Otherwise, I'll put obstacles all the way, hurdles all the way. I always say it's easy to come up with excuses rather than executing and doing the job required. So I think we need actually to agree that admit the problem first. We say this on stages, we say it in conferences and meetings, but when it comes to reality, we don't do it. Why? I have no idea. So we need to tackle that and think about today in Saudi in particular, or in our region. If we don't collaborate, we're not going to compete. We're not going to be on the map. So hopefully people understand that, especially the youngsters. It's our job, as Naif said. It's his job. I'm still a youngster. He left the scientific community, some kind of. But people who are doing research, clinicians, PhDers, they need to, especially the ones with experience, they need to help the new generation and teach them that rather than teach them how to work, to be selfish and work in silos instead of working together. Thank you.
We have one question over there.
Thank you very much. Thank you very much for the audience. So I'm Professor Moisrini from Paris-Saclay University from France, and I'm so happy to be here with you. And I have one question and one comment. The first question is, from your experience, I see that all presentations are talking about infection viruses. Did you have analyzed the incidence of non-communicable diseases like, for example, inflammatory bowel disease and the infection? And the comment that we are now organizing the World Microbiome Congress in Paris during August, so you'll be welcome to participate in such event. Thank you very much.
Yeah, thank you. I think this is very interesting. But I think there is, I think third day or fourth day, we're gonna tackle the non-communicable diseases in one of the days, right?
Yeah, but maybe Arnab and Turkey, that's also.
And also, Dr. Turkey Arnab can also add to the microbiome.
can also talk about my work actually. It's like all of the and with this platform that we're used to be honest, I don't see any problem why not tackle non-communicable diseases. We need a immunotherapy for it. Anything that can function as an immunogen, as an allergen, I think it will produce that the antibody that we need. I think so, but we have never tried that.
Okay, so one of the, it's a very important topic that you raised, and again, when it comes to non-communicable disease, but there are, for example, a lot of cancers that are microbially induced cancers, right? So I mean, I'm not just restricting my comment on Helicobacter pylori and colorectal, but I'm talking about others also. That's coming increasingly common, and that's because we can now do a lot of this kind of work. So yeah, I mean, at the moment we are not working directly on that. I had a project some time back, but sometimes you need to focus. So we are focusing on communicable diseases and applications of NGS. But then Dr. Turki talked about the microbiome part. Again, that's a massive. In this part of the world, microbiome research is happening, but very scattered. And I think, as he also mentioned, that he's very keen to get everybody under the same umbrella. And let us do our own bit. We have a lot of access to technologies and bioinformatics analysis algorithms. Many people here in these rooms have actually working with clinicians or actually work directly in hospitals, have particular access to material samples and can start new projects. But first we have to standardize our analysis protocols, sample collection protocols, inclusion, exclusion criteria, and so on. But yeah, I mean, that's a very important thing. And I would love to receive some more information about this meeting in Paris that you're talking about. Thank you.
Assalamu alaikum. Thank you so much for your talk. Dr. Tamir, I have a question actually for you. Your talk was fascinating. Thank you so much. You mentioned the IgY, they have neutralization function. What about other functions of the antibody, like ADCC or antigen-immune complex? Will the missing part lead to more precipitation, for example, in joints or other organs leading to autoimmune disorder or something like that? Have you checked that on the mice?
That's an excellent question. To be honest, no, we haven't checked that. But then I would say that when we talk about the structure of IUI, so like it has a four constant, meaning it also like the hinge region that makes it like less flexible and less cross-reactive with mammalian proteins. Okay, so even when we wanna purify it, like we cannot use this protein G, protein A because it doesn't interact with the bacterial proteins as well. So it's also well known that it does not activate the complement system. It does not activate the receptor. Yeah. So that's why it's very desirable when it comes to mucosal and oral immunity, where actually say systematic immunity activation is less preferred. Okay, so that's what makes them an attractive target for that purpose. But did we look actually reconfirm this? That's a very interesting thing, and we have. To be honest, looking into that, but we haven't ourselves.
All right, so time's up. I think we can take one more very brief question.
Thank you very much. I had one question for Dr. Arnaud. You mentioned about surveillance and the models that you mentioned were primarily on the collection of patient samples and in Dengue's case, vector samples, which is an improvement from sentinel chickens and so on. My question was that COVID, SARS, MERS before that, these are basically airborne respiratory viruses. So how would you expand surveillance, persistent surveillance, particularly at schools and airports, from aerial samples?
Thanks for your question. Actually, I didn't mention because something that I actually had a project initially funded by the some time back by one of the, it's actually Lockheed Martin and CAX joint project, which we were systematically collecting air samples and then looking into pathogens. So that's something we can check further because we need more firepower and all this. But now that the technology has improved so much, we can now detect, as you see, from single mosquito, any virus that you look for. and to a very high degree of confidence. So I just what I showed you is as long as you have good starting material, whether it is DNA or RNA, whether those RNA or DNA have been collected from the air or from a bunch of mosquitoes or ticks or other vectors, or clinical samples, or from patient biopsy samples or CSF samples, they usually you have the power to assess presence of unexpected organisms or viral particles there. Challenge is the amount of material. Challenge is how can it is RNA, how can you keep quality intact? Otherwise, you might not get the answer. So we have now at least part of the the protocols, and it is easier, relatively easier. I do it in a lab, controlled environment, everything works beautifully. But how you can reproduce the whole thing in a if I have to do the same thing in Jeddah Airport, how do you do this? How do you make sure that your material is collected safely with enough metadata and then ultimately transported to a regional lab? Could be RRL, MOH lab, or could be any other university lab, including in KU here. But I think now, what was not possible 5 years ago is now possible. We are kind of at a technological level. It's just a sample collection and storage. These two remains to be a little bit of a challenge. But I think with a bit of design and maybe involving some design engineers, we should be able to overcome.
Okay, so with that, we conclude our session. Again, we thank all the speakers for their presentation. Thank you so much. Stay with us for a while. Who's next?
It was nice to meet you. So let's catch up. I think Aviv is continuous discussion with him. So he told me everything he's done.
That's something that we doesn't work on.
So we do have one sample.
Now we will have a short break. We'll start our session at 3:30. So in 10 minutes. Thank you.
Yeah. Yeah.
That means it's like the information for the
Yeah.
Oh, right now, I think.
شوف تامر بس وين؟
ايوه طلع المسلم Prof. Tamir? May I have your attention, please? We will start the last session today. Hello, everybody, please. So we'll start the last sessions today with the introductions for the moderators. We have Dr. Turki Abu Jamil. Dr. Turki Abu Jamil is an associate professor. Am I right?
Yeah.
Yes, Dr. Turki Abu Jamil is an associate professor of microbiology in the Department of Medical Laboratory Sciences at KU, Faculty of Applied Medical Science. He also served as a deputy director for administrative and financial affairs at KFMRC. Dr. Abu Jamil leads the EcoHealth Research Unit, which is recently established in our King Fahd Medical Research Centers. and is the principal investigator in the vaccine and immunotherapy unit. So he's with over a decade of experience. His research focuses on the human microbiome and antimicrobial resistance and host microbiome interactions, integrating bioinformatics with clinical applications. Please, Prof. Turki. And second, sharing the moderations of these sessions is Professor Thamer Al-Andijani, Abdulaziz Al-Andijani. He's an associate professor of virology and consultant of laboratory medicine with over 45 publications in peer-reviewed journals. His recent work focuses on respiratory and re-emerging viruses, and his research highlights contributions in viral immunology and epidemiology, surveillance, development of diagnostic tool, and immunotherapeutic antiviral agents. Please, Prof.
Right, thank you, Professor, for the intro. For the sake of time, I'm going to start very quickly with Dr. Nabil Al-Zahrani. Dr. Nabil is an assistant professor and the chair of the Department of Clinical Laboratory Sciences at King Saud University. He also serves as a consultant and clinical scientist in microbiology and virology in the microbiology lab at the Ministry of National Guard. Dr. Zahrani research work bridges clinical diagnostic and academic research, particularly in the area of diagnostic and public health microbiology. Please welcome Dr. Zahrani.
Bismillah ar-Rahman ar-Rahim. First of all, I'd like to thank the organizing and scientific committee, Prof. Anwar, Prof. Assam, and our moderators here for their kind invitation. Today, I will be talking about epidemiology and seasonal trends of respiratory infections in Saudi Arabia. Okay. Here's an outline of my lecture. So I'll be talking about respiratory infections, their disease burden, their economic burden, and their global epidemiology. And then I'm going to dive in some data from the Ministry of National Guard Health Affairs in Riyadh, showing some respiratory pathogen trends. And then I'm going to wrap up with some concluding remarks. So as probably all of us know, respiratory tract infections are one of the leading causes of morbidity and mortality worldwide. In fact, 3 of the leading causes of death list published by the WHO in 2021 has 3 respiratory infections at the top 10 causes of death. These infections can range from anywhere from an asymptomatic infection to a life-threatening infection. They are extremely common. In 2021, there are about 12.8 billion episodes of respiratory infections. About half of those were in children less than 5 years old, and then about 800 million in the elderly individuals above 65 years old. These infections carry a high burden of disease and high mortality rates. In 2023, the estimated death toll caused by respiratory infections was 2.4 million deaths worldwide. And the death rate will depend actually on where you live, depending on the socioeconomic status and many other factors as well. These data were taken from the Global Burden of Disease study, showing that the incidence and prevalence of these infections in Saudi Arabia continues to rise, where these infections peaked around 2020, where we had COVID and then kind of normalized at 2021, but they continue to rise over the years. If we want to look at the economic burden of these infections, one very elegant study by Dr. Abdullah Asiri from MOH and his colleagues studied this, and what they found is the total economic burden of viral severe acute respiratory illness will vary by whether it's early diagnosed or delayed diagnosed. So the direct cost for these patients, if they're diagnosed early, is about $46,000 per patient. and that adds up to $3.8 billion on an annual basis of direct costs. If you factor in indirect costs, that means days off work and stuff like that, that adds about 5% or something like that. It goes up to $4 billion US dollars per year. Now, if the diagnosis is delayed, the number goes about an extra billion, almost an extra billion, $4.7 billion per year. And if you add up the indirect costs, it goes all the way to $5 billion US dollars per year. So a huge disease burden, a huge mortality burden, as well as an economic burden. So what type of respiratory infections are there around the world? What are the most common infections? This systematic review looked at the etiology of respiratory infections in adults on the global level from 2019 to 2022. And as expected, SARS-CoV dominated the respiratory infections during this period, as it was the peak pandemic period, followed by enteroviruses and rhinoviruses, and human RSV was also prevalent in adults unexpectedly. And in this study, they looked, the total number of samples cumulative was about 421,000 samples. Now, if you move to pediatrics, they looked at this in this systematic review an extra year, 5 years, and you see in pediatrics, SARS-CoV-2 is not actually the most common etiology of respiratory infections in those. Actually, it's enterorhinoviruses and respiratory syncytial virus, where about cumulative pooled prevalence of 25%. So, and SARS-CoV came in third place in terms of pediatric patients. So, moving on to talk about data from our hospital, I'm gonna talk about the respiratory patterns from 2021 to 2025 up to March. So I'm going to show you how these respiratory infection dynamics change over the years. So first off, 2021, this is a figure showing the number of cases that we had from infection on the x-axis, and then on the y-axis is the etiological agent. So the highest number of cases in that year, where we analyzed about 7,500 samples, human rhino-enteroviruses really dominated with around 1,600 cases, followed by SARS-CoV-2. And unlike what we saw on the global level, we had a lot of cases of adenovirus in our institution. And if we look at the seasonal pattern, here, what I'm showing is over the x-axis is the months of the year and then the number of samples. You see that with the green line in this rainbow chart, this is showing the human rhino-enterovirus, and you can see they're prevalent all year long. When you look at COVID, which is the orange line, it stops right around August, and then in September, and the fall and the winter months, influenza kind of takes over the large portion of infections during these months. Moving to 2022, with 2022, we still have human rhino-enteroviruses as the most common. The number of cases went from 1,500 to almost double, 2,800 cases. SARS-CoV was number 2, respiratory syncytial virus was number 3, and you can see almost all of the pathogens doubled in the number of cases that we have seen in 2022. I'll come to talk about the reason for that in just a minute. So here, again, you see rhinovirus, rhino-enteroviruses. This is their typical pattern all over the year. They're dominating. And then you see here in this figure, SARS-CoV really dominated during this period. One significant thing that I should mention here is where COVID-19 restrictions were lifted in Saudi Arabia. So during the July-August period, people were on summer vacation, traveling, so the number of cases was down. And then when school came back at August and September, you start seeing this really high surge of infections, mainly rhino-entero and RSV really dominated this year. Moving to 2023, in 2023, we had a huge shift in dynamics. human rhinoenteroviruses always dominates, but here we saw a huge surge of influenza A that took over SARS-CoV-2 in 2023, and that was followed also by adenovirus. And number 3, and SARS-CoV went all the way to number 4 in terms of the number of respiratory illness cases. And if you look at the seasonal pattern, again, dominated by rhinoenteroviruses, and you can see in the light blue where it started September, you can see the influenza cases jump really high. So the light blue is the influenza A non-subtyped, and then in the purple right below it, below the green, is the H3 variant, and then the H1N1 is in the light orange afterwards. So it's all the different types of influenza dominating that season in 2023. Moving to 2024, again with the human rhino as the most dominant virus, and then SARS-CoV kind of made a comeback as the second most common pathogen in that season, followed by RSV. You can see the number of samples are increasing with each year, so we started off with RSV at the beginning of January, very high cases, the blue color. And then COVID around March and April in 2024. I believe that's when the JN1 subvariant was circulating around the world, so that would cause that surge. And we had a kind of a mild winter during that year in 2024. Now moving to the current year, up to March, what we're seeing now is, again, with the rhinoentero as the dominant virus, a respiratory syncytial virus, and then human metapneumovirus really dominated this winter season with a lot of cases, about 370 cases so far up to March. And then also influenza B, we had a lot of cases of influenza B during this season. So as you can see here in the blue color, this is the RSV, the green is rhinoentero, and then in the pink color, that's influenza B, followed by influenza A, H3, H1N1, and then human metapneumovirus. And you can see the pattern really shifting throughout these months compared to the previous years. Okay, so notable trends in the past years that I mentioned. One of the very notable trends that I have seen is the RSV, where we had a very high surge of cases in 2022, with around 1,367 cases during that year, and kind of started to normalize afterwards. MERS-CoV, at least in the data that I'm seeing, seems to have a biannual peak, where we had 16 cases in 2021, 19 cases in 2023, and almost no cases in 24, and only 5 cases in 2022. Bordetella pertussis and Mycoplasma pneumoniae, we have seen these pathogens re-emerge in 2023 and 2024, and these pathogens are not really exclusive to our institution, but this is really a worldwide phenomenon that everybody is seeing a resurgence of Bordetella pertussis and Mycoplasma pneumoniae as well. So to conclude, these viruses and respiratory pathogens in general continue to pose a public health concern, especially among young children, elderly populations, and immunocompromised hosts, which we're having an expansion of transplant programs around the country, and this causes an increase in immunocompromised patients as well. The circulation patterns, as you can see, kind of differ from year to year, and there is what we call this immunity debt that we had, that we were really had all the restrictions and everything, and we were not really exposed to any pathogens, and now they're returning with a vengeance. Finally, I want to say that the investment in improved diagnostic tools can really help you map out your epidemiological landscape in your country. Although these multiplex assays are expensive, but on the long term, they will help you understand the patterns and how to prevent them and how to undertake them in terms of research and other support. With that, I'd like to thank you all, and I would be happy to take any questions.
Professor, because Dr. Nabil Hanani has to fly back to Riyadh, I think.
Yes.
Yeah. So we're just gonna take the opportunity to have him. Thank you, Dr. Nabil. Right, moving to the next talk. Our next speaker is from Egypt. Dr. Hassan Al-Sayed. He's an associate professor and a group leader at the Biotechnology Research Institute in the National Research Center in Cairo, Egypt. He obtained his PhD in molecular and medical virology from Friedrich Alexander University in Langen-Nuremberg, Germany. I'm pronouncing this right. Dr. Said has extensive expertise in infectious disease, cancer, and nanoparticle applications. He nominated several national and international grants and developed innovative vaccine strategies against SARS-CoV-2, HIV, and HCV. Dr. Said developed an ELISA assay for COVID antibody detection in both vaccinee and patients. Please welcome Dr. Said. Thank you.
So thanks for this kind introduction. And first of all, I'd like to thank the organizing committee, especially Professor Assam Azhar and Professor Anwar Hashim for such kind invitation. So my talk today will harness how can we harness the natural infection, not only to have a safer vaccination strategy, but also we'll discuss more deeply the interplay between the host genetic and disease severity. So in the next 15 minutes, my talk will be structured as follows. First, I'm going to give a brief introduction about SARS-CoV-2. Then we will more deeply answer the question, how does vaccination work, before moving toward the rationale of our vaccination strategy, showing you the preclinical study that we already have, and we will end my talk with host genetics and disease severity. So when we think about SARS-CoV-2, we all agree that SARS-CoV-2 was the worst pandemic resized over the last decades that lead to the coronavirus disease of 2019, which is called COVID-19. So as you probably know, SARS-CoV-2 is a respiratory virus that mainly targets the lung. Nevertheless, it could also target many other organs, like the heart, the brain, the lung, because of ACE receptors. I just want to bring your attention here to this slide 14, because this is the key player in our talk today. Despite the presence of various emergency licensed vaccination strategies like that for Moderna, like for Pfizer RNA vaccines, and etc., there is an urgent need to have an efficacious, safe vaccination strategy that would indeed overcome the newly mutated SARS-CoV-2 mutation or virus. But how does vaccination work? Ideally, if you want to have a vaccine that completely prevents infection, this is called sterilizing immunity. In general, sterilizing immunity is conferred by antibodies that binds to whatever the pathogen is and prevent even the initial round of infection from occurring. This is caused by antibodies. On the other hand, there is much more common kind of vaccines, and these vaccines indeed don't prevent disease, but don't prevent infection, but prevent disease. And this is called non-sterilizing immunity. Generally, non-sterilizing immunity is conferred either by antibodies or cytotoxic T lymphocyte. To our knowledge, there is much more common strategies that vaccinologists can develop to have a vaccination strategy, like the killed virus, like we've already got polio, hepatitis E, and rabies, live attenuated virus, for instance, the measles, rubella, and mumps. We could also play around the genetic makeup of the virus, that you have a recombinant protein like the licensed vaccine for hepatitis B or hepatitis B. Nowadays, you could also have a recombinant even DNA vaccine, and was in the revolution of this pandemic side, the mRNA vaccines. So what about the rationale of our vaccination strategy? As I mentioned in the beginning of my talk, that SARS-CoV-2 mainly targets the lung. So our colleagues, Ahmed and Sheikh, started a pan-immunoinformatic analysis, thereby studying all the isolate or strains that have been developed since the beginning of pandemic 2020 by identifying the spike protein, so that they were able to have the most consensus sequence of the spike protein. Subsequently, they provide a pan-immunoinformatic analysis, thereby they were able to define the B-cell epitope CD4, CD8. Side by side, we were curious to define also the proboscis CD4 epitope from the licensed vaccine like polio, like hepatitis, etc. Then we use a nano particle delivery system to deliver our candidate vaccine in people. So this is example for one of our antigen, our candidate peptide, short amino acid, like for example, this is only a 5 amino acid. The other peptide, as you can see, around 9 amino acids. The rationale actually based on that we would conjugate both peptide with what is called cathepsin cleavage site. This cathepsin cleavage site has a capacity that could be undertaken by macrophage, and then each peptide have been released to express the functionality. We would also use each peptide in octamer, which what we called multiple antigenic peptide. Thereby, we improving the surface area of absorbance and therefore induction more and more with antibodies response. So our colleagues, Professor Shireen Khattab and Professor Mona Agua, the team of peptide synthesis and conjugation nicely conjugated or coupled peptides, what you can see, peptide A, for example, peptide B, peptide C, D, and E, onto the lactoferrin, our delivery machine, which was lactoferrin. After characterization of such conjugation, we went further for in vivo experiment. But the first question we asked is, which candidate of these peptides that I gave example for you from A, B, C, D, E, that would be the ideal candidate that could induce immune response once we went for further in vivo experiment? And to answer this question, we established a collaboration with general organization for teaching hospitals and institutions from the Ministry of Health in Egypt, with Professor Abdul Ghaffar and Professor Sheikh, that they were able to provide us with samples from IC unit from more than 14 hospitals belong to the GOTHI in the Ministry of Health. Thereby, we have samples from those patients, and those patients have been confirmed to be already infected with SARS-CoV-2, not only by a CT scan, but also with a PCR, to be sure that these samples from those patients already hospitalized in the IC unit have already got the SARS-CoV-2 infection. I'm showing you the NL ratio besides the CT scan. To make the long story short, we established an in-house ELISA, and this assay was performed by Dr. Reem and Dr. Rehab. Thereby, we make a panel screening for more than 50 peptides to see which of these peptides would indeed recognize antibody response in the serum of hospitalized patient. What you can nicely see here is that peptide 3, peptide 5, peptide 13, and 15 exhibited immune response. What I'm showing you, only the peptide which give us a positive response. And actually this was from the beginning of the pandemic, I think by the end of 2020 in Egypt, till the mid of 2022. Recently, we have, once that there was a new variant or new strain, we were curious to see if this antigen would indeed recognize with the newly mutated virus. Interestingly, what you can see here, that we still have also antibody detected in the serum from hospitalized patients. Actually, this would mean that if we selected such epitope and we went for in vivo immunization, that would make sense that once you immunize the animals with such peptide, you would have actually antibody response which mimic these response that I've shown you in tissues. To make the long story short, mice received 2 immunization experiments within a 3 weeks interval with only 1 microgram from our, which we named Picovac, and the control PBS. What you can nicely see here, and this was done by Dr. Reem, that for instance, the reactivity for the mice sera anti-spike antibody was very important, statistically significance induced only in immunized animal versus control. The same picture was observed also against the second candidate peptide D5. As you probably know, antibody recruit different similar effector mechanisms beside their neutralization. Therefore, to dig deeper into the characteristic profile of such antibody response, we were curious to see such antibody response of any relevant functionality, like for instance, antibody ability. So the antibody ability is a key surrogate for the efficacy of a vaccine. Therefore, we established an ELISA assay whereby we could determine if this antibody of relevant functionality. What you can see in this cartoon that we treated the antigen-antibody interaction with a ketotropic agent, which was sodium cisphenate. Interestingly, you would have 2 scenarios. If the binding is very strong, this means that you will have a strong immune response. On the other scenario, if this binding is very weak, then it will be a leakage, a new antibody of a weak activity. This was done with Dr. Rahab, and what you can nicely see here is that she calculated the avidity indices by dividing the response to cisphenate by the response with PBS, and to have the index we have to multiply with 100. What you can nicely see here is that Picovac induced strong antibody with high avidity indices compared to the PBS in response to B3. The same picture was observed also for the B5. Interestingly, when we compared the response that we have in vaccinees using our candidate peptide, you can nicely see here that the natural infection actually induced antibody with avidity indices close to what we induced, like in the upper figure, compared to the response induced in mRNA vaccinees versus AstraZeneca, which was very supporting to our vaccination strategy. Then we were interested to see if our vaccine would induce a strong T-cell response. What you can nicely see here, we compared our Picovac with the Chinese vaccine, the Sinopharm. Mice received only single immunization. 2 weeks later, we explored the T-cell response via ELISpot and cytokine ELISA. What you can nicely see here, that our Picovac vaccine induced a strong induction for IL-2 in the immunized spleen and lung in both animals, compared in both intranasal immunization as well as intramuscular. Interestingly, the response induced by Picovac was much better than that induced by the Sinopharm vaccine. The same picture was observed for other cytokines, IL-6, IL-8, and IL-10. We were curious to see if we will have the same picture, not only via Th2 cytokine, but also for the Th cytokine, Th1. Therefore, we performed ELISpot ELISA in both lung and spleen. So interestingly, we got a very nice result with all route of administration for the Picovac vaccine for the intranasal, intramuscular, and subcutaneous is also compared to the Sinopharm. We were curious to see if immunizing animals with our Picovac vaccine would alter the biochemical lab, because if you know already that once you get the infection, you have alteration on all liver, kidney, and even the cholesterol CBC picture. EMAN and WM nicely performed a complete blood picture, liver and kidney. What you can nicely see here, there is no differences in the complete blood picture in the liver and kidney. Then we were curious to see if such immunization strategy would indeed induce any changes in the liver, kidney, spleen, lung, or brain. Therefore, Professor Mais Hanafi performed the histopathological examination 2 weeks and 2 months, and there was no differences between immunized and non-immunized organ. In a visit last December, last year, Muriel and Dr. Hab, in collaboration with John Bibison and Bastille Institute, they performed an immunization experiment. Interestingly, this is by seed particles, but we have already seen some kind of positive response. like this is in red column, our vaccine as compared to avian. Just to show you that how patients differ, some patients already got mild symptoms, others have severe, and this is actually because of host genetics. In this slides, I'm going to show you some published and unpublished data. This was done with Dr. Rihab and Dr. Noha Gamal. What you can see on your right hand, this is the OE's response. There was the distribution of genotype GGG with mild cases was much higher prevalence than in severe. The same picture was observed for also the IL-28B. That the distribution of CC genotype was much higher than in mild cases. Just to conclude, I've showed you a platform for a vaccination strategy against SARS-CoV-2. This vaccination strategy have the B-cell epitope CD4, CD8, and Promiscuous CD4, which actually have been prolonged preclinical study and clinical one study in Bioinformatics Institute in Germany. By that, I would like to thank our working group, some collaborator, clinician, like John Dibiso at the Steele Institute, Dr. Al-Assas, Dr. Muhammad Mustafa for having sampled, and of course, Dr. Ali Zaki. Some of you know him. He had more than 20 years here in Jeddah. And thank you for your attention. We're happy to answer your question.
Thank you, Dr. Hassan, for this really nice presentation. And now it's my pleasure to introduce our next speaker, which is Dr. Aisha Al-Na'mi, who is an assistant professor at King Abdulaziz University, specializing in pharmaceutical chemistry and drug discovery. She holds a PharmD degree from the King Abdulaziz University and MSc and PhD degree in drug discovery and development from University College London. Her research focus on FTSZ inhibitor for Mycobacterium tuberculosis with expertise in protein crystallography and small molecule screening. Please join me to welcome Dr. Aisha.
First of all, let me thank you, the organizer, to having me today here as a speaker, and then I'll start to take you through my journey. It's a drug discovery. It's a pure drug discovery project. It's a pure drug discovery project. It's a collaborative project between University College of London and King Abdulaziz University. Our drug discovery project looked at humanine-based inhibitor as inhibitors for bacterial cell-deficient protein FTSZ, which is promising target for developing broad-spectrum antibacterial agents. As I said, it's a drug discovery project, so I'll start my story by choosing by selection a disease target. First of all, we choose disease target. We focusing on multi-drug resistance bacteria. Why we choose it? Simply because the multi-drug resistance bacteria it is an urgent unmet medical need, with the rise in the worldwide resistance. It's well known that infection with multi-drug resistance bacteria is usually associated with high morbidity and mortality. It's also estimated that multi-drug resistant infection could result in 10 million deaths yearly by 2050. And there is some remarkable examples for multi-drug resistance bacteria like MRSA or MDR-TB. Okay, then, as I said, next step to the select novel drug target. Novel drug targets, we do evaluation all the available bacterial drug targets, and finally we choose the FTSZ. Why we choose an FTSZ? We look to the function, as I said, is essential bacterial diffusion protein. In the presence of GTP, FTSZ polymerize into protofilament, and during the bacterial cell division, FTSZ move to the direction side and recruit other accessory protein essential for cell division. And it's like here, like the FTZ, it's like monomer in the blue dots, and forms what says is contractile Z ring. This contractile Z ring contraction, and then finally leading to the cell division. Okay, when we do the inhibition study, we see that inhibition of this FTZ will proper formation of the Z ring, leading to the filamentation, and the bacterial cells continue to grow, elongate, but it cannot divide and eventually cell death. So, if we inhibition the FTZ, we expect it to have bacteriostatic, not bactericidal. And when we do, like, we try to research if there is a possibility of resistance, and we see if you do the mutation studies, we will see even single change in one amino acid could result in reduced in FTZ activity and also bacterial viability. We assume that the resistance via mutation may be difficult due to the fact that FTZ is essential rule and conserved nature. As I said, FTZ is essential for all bacterial cell division and they're present in almost all prokaryotics. So, when we take FTZ from different bacterial strains and we're trying to align the amino acid sequences, we will find that there is a strong similarity between the amino acid sequences. So, there is a high possibility if we end with good FTZ inhibitor, we end with broad spectrum antibacterial agents. When we look at the drug target, we have to look at selectivity and safety. Selectivity and safety, it's meaning that we have the drug safe and have lower side effect. FTZ, as here see, there is the structure similarity between the normal tubulin protein present in eukaryotic, but the similarity only in three-dimensional structure. But when we align or try to align the two amino acid sequence from the tubulin from eukaryotic with the amino acid from prokaryotic, we find that the similarity between amino acid sequence is only 17%. And also we will try to see from the classic tubulin inhibitors, which already validated drug target like paclitaxel or albendazole don't inhibit the FTZ. The third step in our story of a drug discovery journey is to hit identification. Challenge in the drug discovery, several groups over the world have acknowledged the importance of FTZ as protein drug target and antibacterial drug discovery since 1990. However, the identified inhibitor, usually from natural product or synthetic molecule or nucleotide, usually false positive or weak binder, because they use the traditional method. Traditional method is meaning that they use whole-based assay or use the target-based screening. All of them may be end by the scaffold already discovered or it's false positive. However, when they try to use the modern drug discovery, like structure-based drug design, or even to go further to their studies, they hampered because there is a lack of crystallographic data, especially FTZ in the presence of inhibitor. The native structure, they managed to solve it, but with inhibitor, no. So we try to take a challenge and try to fill the gap by do some crystallographic, extensive crystallographic study to we go to the literature and try to find number of validated or what they said it's inhibit FTSZ and try to crystallize it, like 4-hydroxycoumarine and some benzoimidazole derivative. This study were followed by structural activity relationship analysis by catalog with goal of advancing research in the field of structure-based drug design, which one of the modern drug discovery techniques that we use today. First, before starting to find a hit, we express and purify the protein with high yield and with high purity as well. As you can see here in the gel, it's more than 95. So we are ready to characterize the protein. We characterize the protein if FTSZ have two functions, GT-based functions and polymerization functions. For GT-based function, we set up two assay, and we set up light scattering assay to look at polymerization activity. We will use this two assay later on to study the inhibitory effect of the potential FTZ inhibitor. After that, we do structural characterization of Mycobacterium tuberculosis FTZ. We managed to solve the native structure of FTZ as a native structure. Here, this table shows the collection data and the statistic data. And we will see that the FTZ, it's crystallized at dimer composed of two monomers, free nucleotide monomer and monomer binding to the nucleotide. And the monomer oil contain two domains, N-terminal domain and C-terminal domain is separated by helix 7. After we characterization our drug targets, now we are ready to do our screening for the published FTZ inhibitor using the X-ray. I do X-ray crystallography, do co-crystallization, and I do soaking crystallization, and both of them, we managed to have the data sets, we managed to have do refinement, but we didn't find the inhibitors binding to the enzyme, except the 4-hydroxycoumarine complex, while other inhibitor, we couldn't be resolved. So for FTZ 4-hydroxycoumarine, this is the first binding pocket solved for FTZ from Mycobacterium tuberculosis, and the second one for FTZ in general. We managed to solve the crystal structure at high resolution is 1.68 angstrom, and we can see hydroxycoumarine bind exclusively to only the monomer that it doesn't bind to the nucleotide. It's bind to two binding pockets. This is allosterical binding pocket. And we can see here, this is the binding pocket number 1. We can see here that 4-hydroxycoumarine binding to the binding pocket 1 with full occupancy, and can see here electron density map is covering the 4-hydroxycoumarine entirely. And this is the interaction between the 4-hydroxycoumarine and the residue in the binding pocket. Is for number of hydrogen bond and number of hydrophobic interaction. Here, the second binding pocket, we can see here as well, the electron density map is fully covered the 4-hydroxycoumarine and bind fully to the binding pocket number 1. The resolution was very good, so we could even saw that the water mediating hydrogen bond and hydrophobic interaction. Now, as we said before, that there was the problem that to start structure-based drug design is the absence of the FTZ in complex with the inhibitor. I need the structural data. Why do we choose to work with the coumarine, scaffoldidovol, a lead compound, among the other available inhibitors? Simple, because the antibacterial effect of several coumarine analogs have been quantified in different bacterial strains, including Mycobacterium tuberculosis, Staphylococcus aureus, and whatever. And literature shows that the 4-hydroxycoumarine inhibit polymerization activity in 2 filaments and GT-based activity with IC50 value around 200 micromolar. And And we just obtained the crystal structure of FTZ in complex with 4-hydroxycoumarine was obtained. So there is nothing to stop us from going to structure-based drug design. As we know, the coumarine, there is a lots of analogs of the coumarine is commercially available. So we're trying to do like a research for commercially available coumarine by using some sub-database like PubChem or A molecule, and we found more than 1,500 coumarine analogs, and we create a library of coumarine analogs to prepare it for docking studies to trying to ranking the analogs. After that, we docking the coumarine into two binding buckets, and the top 100 analogs with the best docking score were selected for purchase, and then we'll do biochemical, binding affinity data, and structural data could be given for analog, and the SAR would be established in the progress. This is step, it's in progress. Until this moment, we tested 38 coumarine analogs. Among 38, we got 4 coumarine-based inhibitors hits result from our screening. 2 of them named 3-methylchrominol and 3-aminocoumarine. The estimated IC value for 3-aminocoumarine is 0.9 millimolar in coupled assay enzyme, and also in malachite green assay is 0.3 millimolar respectively, while 3-methylchrominol showed no inhibitory effect. We tried to explain why this is the phenomena. If we look to the chemical structure, we will see here we have coumarine scaffolds. Here we have chrominol scaffold. We think that this is different in scaffolds may result in the loss of the activity of FTZ inhibitor. And we managed also to crystallize 3-aminocoumarine and 3-methylchrominol in the presence of FTZ, which further help us to justify what happens. We will see here 3-aminocoumarine, it's just like 4-hydroxycoumarine. It's bind to 2 binding pockets. And also with full occupancy, full occupancy at resolution 2.5, and also this binding to another binding pocket, as you can see here. While 3-methylchrominine, it's binding only binding pocket. This is maybe suggest that the coumarine scaffold is not only important for binding to the binding pocket number 1, but it's also important to bind to binding pocket 1, and maybe the coumarine FTZ inhibitory effect is linked to the ability of compound to bind to binding pocket 2. Our story is not ended, it's still in progress. Thank you very much.
Thank you, Dr. Aisha, for this interesting talk, and inshallah ختامها مسك. Now we have the last session or the last talk for today, and it's my pleasure to introduce our next speaker, Dr. Abdullah Bawazir, who holds a PhD and MSc along with advanced credential, including a doctorate in healthcare administration, project management professional, and lean certification. He currently serves as the executive director of the Diana Medical Factory. He's a consultant and a head of the microbiology department at Asir Central Hospital. His research interests spans viral pathogenesis, immunology, and antimicrobial resistance. He's also actively involved in the nanoparticle-based drug delivery and clinical microbiology, biotechnology, and bioinformatics. Please join me to welcome Dr. Abdullah Bawazir.
السلام عليكم ورحمة الله. أنا باخذ هذا أدفانتج يعني باخذها واحد من وشيء ثاني. شكرا لاستضافتي، وكان هذا صراحة من أميز المؤتمرات اللي حضرتها. خلينا نبدأ عشان نعمل. فخاطر. طيب we will talk about the novel potential therapeutic targeting the reduced pulmonary inflammation during severe influenza virus infection. Also, if I have time, I will go through brain multidrug resistant nanoparticles. As everyone mentioned, a lot of people talking about respiratory disease, including influenza virus. And we know that for the respiratory disease, influenza virus is very critical, and we see that some mostly affect the epithelial cell from airway, but some strain can infect in the macrophage. Also, I will be quick for introduction because a lot of people talk about that. And we know since 20th century it's influenza pandemic. Currently, the vaccine is mostly have working effect approximately 15 to 60%. However, 2017 as low as 16%. For there to have a limitation for that. Also, antiviral, there's some limitation for antiviral needed to take within 2 days, but the patient mostly go to hospital after 5 days. We go through for hyperinflammation during influenza virus, when the virus infect body epithelial cell secrete the cytokines such as TNF-alpha, IL-16, IL-1 beta, etc. Then continue the activation in monocyte and neutrophils to also produce cytokines, conjugate together to cytokine storm, hyperinflammation. Then when the epithelial did the leaking from plasma to escalate and developing to pulmonary edema and out-like disease. Then after that, I will talk about inflammasomes. The inflammasome is the multiple organism enzyme complex consists of sensor. There's a lot of kind of inflammasome. And ask as adapter and enzyme caspase 1, caspase 11, etc. The main function of inflammasome is to process member of inflammatory cytokines and activate the cytokines, such as interleukin-1 beta and IL-18. In literature review, they show that NRB3 inflammasomes knockout mice were more susceptible to influenza infection. Important introduce of NAT and adapter also. So the mechanism when the influenza affect the body, influenza viruses, we have two signals. First signal activate bilateral recreation receptors, then activate NF-kappa-B to go through the nucleus, then reduce pro-cytokines such as IL-1 beta, IL-18, then goes outside by outer nucleus, then this signal after that activated by cytokines. The cytokines required some ROS and protein lysosome to activate it. After the inflammasomes activation, it's make pro cytokines from inactive and immature cytokines to mature, then leads to apoptosis of this inflammation. We have an Australian Monash University work with some drugs before to affect body vivo for mice by influenza, then use MCC950 as a potential small molecule inhibitor from RB3. And we found there's a good result for positive and survival. Then also for later and earlier stage. And also we found the first lab and show that NRB3 inflammasomes have two stages, protective and detrimental. So the for protective is early stage, and for detrimental is later stage. And we also found the infiltration macrophage to infiltration So coming after, that's why we saw this peaking and increasing. So based on before, we hypothesized that NRP3 responses in infiltrating myeloid cells play a determinant role in promoting I1 beta mediated hyperinflammation and disease during influenza A virus infection. So I have two aims: examine NRP3 in different cell types in vivo, also characterize the contribution of I1 beta as the potential treatment. So currently, the NRP3 chromosomes timing is an affection. It's not well defined. I infected mice with Hong Kong H3N2 and collect the whole lung tissue to examine RNA and protein or cytokines and NRP3 inflammation component. Then second step for response for immunocells by first epithelial cells, isolate CD45 positive and CD45 negative. This is designed for diagram shown for experiment. Then the result for whole lung tissue found the NRP3 inflammations upregulating and for peaking to third day, NRP3 caspase 1, also the important I1 beta. As we expected, ask as a doctor, it's the same level. So when I do it, this is for RT-PCR. For about protein for Western blot, we found and detected pro-I1 beta and IL-1 beta after maturation, and all the inflammation component upregulating NRP3 caspase and comparing to thyroid. Then I do that for second step for the first to understanding the immune cell for CD45 and CD45 positive and negative. And we found for genes the comparing CD4 positive and negative is speaking to the one for NRP3 caspase 1. For caspase 1 upregulating for epithelial cells also and myeloid cells. And IL beta, it's detected on 4 genes for upregulating and also for both cell types, epithelial cells and immunocells. And we detected here, detected after Western blot for I1 beta for mature and pro-inflammatory cytokines. And you see here comparing for whole lung tissue with versus to epithelial cells and immunocells. So second aid, before second aid, we found the upregulating and activating for inflammations, and the result showed I1 beta activated both cell type, myeloid and amyloid. So the second thing characterized the contribution I1 beta disease during influenza virus infection. So I show you have previously for MCC 950 work for day 1 increasing successfully for influenza infection as potentially treatment. Then I do it, I did the I1 beta in vivo different stage of infection for antibody and examine the impact of population, weight loss, viral load, etc. So, in fact, 2 groups of mice with influenza virus. Then one of them treated by early stage from day 1, 3, and 5. And the second one, I treatment by later stage from day 3 to and day 5. Then we found this survival mice we found to early stage and later stage it worked very well, but significant good result for mice is working the early stage, but the better for later stage after treatment mice after day 3. Also examine the impact of anti-I1 beta antibody treatment on the pathogens for day 0 as a control, day 3, day 4. Then we'll see the antibody treatment model, modulated information on the lung following the infection, examine the cellular inflammatory unbound. So we saw the viral load and total cell autophagy, and the significant increase inflammatory macrophages here and dendritic cells after anti-IL-1 beta inhibitor use. Also, I examined the antibody treatment modulation information for bulk cytokines such as IL-1 beta and serum, and we have a significant result found the increase anti-inflammatory IL-10. So the conclusion, anti-IL-1 beta is potentially treatment an early stage, but the later stage is survival of the mice variable. This last so actually, because I saw a lot of people working nanoparticles, so I go through very fast. I will not talking. I acknowledge my professor and my team in Monash, Dr. Michelle Tate and Ashley Benson, Dr. Sarah Rosalie and Chair from Monash University and Hudson Institute. So I will go through for introduction for multi-drug resistance. This is another project, or another project for talking multi-drug resistance. Everyone or before Dr. Nami talking about the important and for found some drug for antimicrobial resistance. The novel project for there we found in researcher nanoparticles without antibody, it's kill bacteria and some fun. So found the researcher Duda and Tuta. So I go through, I synthesized the nanoparticles in lab just for 2 days because nanoparticle is very cheap and very fast. So this is good opportunity. So, and measuring the size and zeta potential, because we have some behind, from that is hypothesis, we found a few scientists highly positive charge for nanoparticles and conjugate with negative charge for cell membrane maybe damage and kill the bacteria. So I make this nanoparticle, 4 nanoparticles, mix it with monoline, and to make it high positive charge, then make each one have different concentration, 1 millimole, 2 millimole, 5 millimole. Then I take the negative, take all these 13 nanoparticles, examine to gram negative, and found this result by effective inhibition and elimination, by broth and by BBS, and found very good result for that. Also, after I put it just nanoparticles with monoline, take the same nanoparticles, synthesis, and added oxalized acid as organic. So we found good result and make nanoparticles work very well. Then I take the same nanoparticles and use it on MERS-A, and we found also some nanoparticles it works to kill MERS-A. And also I comparing that, take nanoparticles and put it folic acid again for all of them, and comparing I found good result also. Take all of them to Streptococcus uniformis by 3. One of them is sensitive and 2 of them is resistant. It's worked very well for sensitive and worked very well for resistant, for some of resistant, and some another for fungi not work for another one. So that's the conclusion for fungi. Is for sensitive for inhibition by BBS, it's work with monoline and sometimes some others, and for broth work actually, and for amphotericin be resistant, it's work very well, but it does not work. I think we have limitation for growth, the last one resistant with after it's not growth, but also this that was not work. Also for behind this not work with our candida also, but I think we have limitation for growth forestry. Thank you for my knowledge, my team, work with them, Dr. James Chapman and Maggie Vikan and Shiana Govendi for RMIT University from Australia. Thanks so much.
Thank you so much, Dr. Abdullah. Now I'd like to ask all of our speakers to join me in the stage for opening the panel session. Maybe we'll open the floor for the audience to ask questions.
Thanks for all the speakers on their work. I have a question for Dr. Bawati. Regarding the molecule that, well, I have two questions. So regarding the molecule that you used it's like a monoclonal antibody against the inflammasome or the NLRP, something that blocked it, I suppose. Because you just said it's a molecule, I wasn't sure what the nature of that molecule that you've been using in the influenza infection. So my question is about the nature of the molecule you're using. Is it an antibody or what is it?
The nature for I1 beta?
No, the molecule you've been using to block that.
As a treatment?
Yes.
It's I1 beta. Actually, when I work for so when I showed you before for mechanism from when fit the body, already so activate through both cytokines, I1 beta and IL-18. It's already on the body from genes to developing as a protein. So this I1 beta leads to bioprocess, to cell death and inflammation. So this I1 taken as a chemical and yes, and block the I1 beta, anti-I1 beta.
IL-1 beta. So my question for you, because I noticed that the survival curve showed whether you used it early on at day 1 to, or you used it maybe later on day 3 to 5. The survival curves were not necessarily, there wasn't, maybe you used a very significantly high dose of infection, and because there was still very low survival in the treated mice in the later days. So my question is, do you think, and this comes from the data that we showed that was emerging from COVID-19 during the pandemic, most of the monoclonal antibodies that we use, especially the biologics, that there's like a sweet spot at which if you use these molecules, especially these anti-cytokines, if you use that, you might be able to actually prevent the progression of the infection. But if that was too late, then you can't actually help that much. And if you use it too early, you might actually lead to increase in the viral pathogenesis in a different way, because it's quite a balance. So have you thought, or maybe before you do this data, did you look at using it prophylactically? So those mice that got infected received it at the same time, which would resemble in clinical scenario people who got exposed, high-risk people who got exposed to the virus, but haven't yet presented with symptoms.
Actually, the inflammasomes, most people, this inflammasomes is responsible for more than 80% for disease. So yeah, so when used as a blocked, some of cytokines secreted from the inflammasomes, it helped for. We believe that it helped not only for influenza, because this has just for example for influenza virus, it can use for. We believe it's responsible for a lot of disease. And if you block that element, it can be helpful element. And there's some question behind that, maybe it's compared to another. Also, I show you that the survival from early stage is still significant. But when comparing with later stage, it's better. I show early stage, maybe without later stage, this question that can happen. When comparing, you found this question.
Yes, that was a question. I mean, I was just wondering if you can actually improve the survival overall, whether it was later. But
overall, how long?
Because if I'm not mistaken, was the survival at 20 or 10% survival in your group?
Yes, 18%.
18%. I was hoping to see survival even higher, if that was possible, but it also can be related to how much virus you use to infect the mice.
Actually, after we treated the mice, this is your question, I understand. We can't take leave the mice long time. So after 17 days, they leave after 17 days, and I was.
Yeah, I'm sorry for the mice. My second question, sorry, because you just raised important stuff. When you use the nanoparticles to kill the bacteria or the fungi, the crypto, for example, these are non-specific mechanism by which you're killing it, right? So my question is, so when you do this as in vivo, how do you envision to deliver this nanoparticle? Because, for example, I envision it as if you have, for example, a prosthetic joint infection or a valve infection, you can do a targeted delivery to the where there's a higher pathogen load. Yep. This is how you envision it, because it's quite non-specific. You can't really give it systemically.
When I do this experiment, but until now we don't know what what understand when you measuring size of nanoparticles. As I said, for hypothesis, we leave it as a chart, hypothesis chart for nanoparticles. But also, when we measure the size of nanoparticles, we found this change to cubic nanoparticles, and this cubic makes us advantage with high dose charge for nanoparticles to kill the organism. And for the future, we should be doing the because I only did
Assalamu alaikum. First of all, I would like to thank the organizer and the speakers for today. My questions for Dr. Hassan. Would you please elaborate more on the controls that you have used in your study? Because I saw -7 and 0, and then you started to 2, and then you go on or went on for the others. And also the control, is it enough to use just from the beginning, or you should add like a solvent that you use or something that could affect the result? I mean, if the vaccine was dissolved in something, as a solvent, would it affect, or have same effect when we have the vaccine on the mice? Thank you so much.
Thanks for this point. Actually, due to time limitation, I didn't give much more details about the vaccination dose and the control. But for in vivo experiment, what would be important that we deliver the Vicovac, which actually a cocktail of peptide and these B-cell epitopes plus CD4 and CD8, but we have more than one vaccination strategy. So we have a vaccination strategy to induce only antibodies, which mimics the antibodies that have been induced during natural infection. This is a vaccination strategy which would be fit for kids, for pregnant women and who out. Another vaccination strategy, which would actually combine both antibody response and T-cell response. And in this, we deliver not only B-cell epitope, but also CD4 specific to the spike protein, as well as CD8 plus promiscuous CD4 epitopes. And this CD4 epitope we've already got, like for instance, the tetanus vaccination, the polio vaccine, the measles vaccine. So we focused on some specific epitopes from the CD4 just to induce much more stress to the antibody to have more burst reactivity spikes. And for us, it was important to deliver these in as a saline PBS steroid. Therefore, we already immunized a group of mice with only PBS, and we didn't observe any response as expected.
Okay, we have time for one last question.
Okay, Assalamu alaikum. So this question is for Dr. Aisha. So you showed this complex between the protein and the covalent. Have you tried to measure like the affinity of these interactions?
Thank you for your question. Yeah, we're trying to use to measure the affinity using the microscale thermophoresis, and we're using like thermal shift as well. But the data, because if you look to the structure of the coumarine, it's look like fragments. For the fragments, usually the affinity is low. So this is why like we're working like to have a full SAR for coumarine to start growing the coumarine for larger molecules. We end by larger molecule, we will like starting to measure the affinity using this techniques, but we already like set up this toolset.
Okay, now we announce the end of our last session for today, and please, some of you should join me.
تكريم تركي هذا شيء قريب حقيقة الناس أبليوا بلاء حسن كثير الفريق كله الحقيقة ما شاء الله تبارك الله شكرا.
Now actually we we have the poster award.
طب هل في سلايد مكانين ولا لا؟ ايوه استنى شوي.
يعطيكم العافية جميعا. شكرا على الحضور وشكرا على المشاركة. أتمنى everyone enjoyed today's talks. Hopefully we build use it as a starting point for building more not starting, but capitalize on it to make more collaboration and work together. Are we ready? Huh? So now we're I think announcing the winners for a poster session. Who's gonna announce? Your feedback is highly appreciated. خلاص؟ يلا. من الثيرد صح؟ الثيرد المفروض. عادة احنا من الثيرد. خلاص جو اهيد. خلوا قولوا مع بعض كلهم ثلاثة. ها؟ يلا. يطلعوا سوا. يطلعوا سوا كلهم مع بعض بسرعة. يلا.
Okay, so the first place goes to MERS-CoV. المركز الثاني لأفنان السلمي. والمركز الثالث goes to سلمى السيد.
طبعا اخر وحده تخرج يوميا عن المركز بعد انجاز اعمالها في اللاب هي ميسة يونس. عيون ما تبان الله عليها يفتح المركز في الصباح وتقفل المركز ذاك. فما شاء الله انهم متميزين جدا الحقيقه، ونسال الله عز وجل التوفيق. تعمل ايش دكتوره؟